Section 1, Part 2. The Case Against Universal Common Descent and for a Unique Human Origin. 10. The Fossil Record and Universal Common Ancestry. Günter Beckley and Stephen C. Meyer. Summary. This chapter is the first of 3 examining the strength of the case for universal common descent, the second, historical, part of contemporary evolutionary theory and the part of evolutionary theory that theistic evolutionists most commonly defend. We begin in this chapter by examining the logical structure of the argument for universal common descent. Taking that structure into account, we then assess what the fossil record can tell us about whether all forms of life do, or do not, share a common ancestor. Theistic evolutionists often claim that the alleged common ancestry of all forms of life is a fact, even as they may acknowledge doubts about the creative power of the neo-Darwinian mechanism. Nevertheless, we have become skeptical about universal common descent. In this chapter, we explain why using the fossil evidence to illustrate how a scientifically informed person might reasonably come to doubt the arguments for universal common descent, or universal common ancestry. After first describing the aspects of the fossil evidence that the theory of universal common descent explains well, we then examine other aspects of the fossil record that the theory does not explain as well, or at all. We especially highlight the many discontinuous or abrupt appearances of new forms of life in the fossil record, a pattern that contradicts the continuous branching tree pattern of biological history postulated by proponents of universal common descent. Introduction. Contemporary neo-Darwinian theory has 2 main parts. The first part, which we have critiqued at length in the preceding chapters, asserts that the mechanism of natural selection and random genetic mutation has the capacity to generate major innovations, macroevolutionary change, in the history of life. The second part of neo-Darwinism, the theory of universal common descent, concerns the pattern of change through biological history. Indeed, the theory of universal common descent is a theory about what happened in the history of life. The theory affirms that all known living organisms are descended from a single common ancestor somewhere long ago. Biology textbooks today often depict this idea, just as Darwin did, using a great branching tree. The bottom of the trunk represents the first primordial organism, or organisms. The limbs and branches of the tree represent the many new forms of life that developed from it. The vertical axis of the tree represents the arrow of time. The horizontal axis represents changes in biological form, or what biologists call morphological distance. Whereas the mechanism of natural selection and random mutation describes how major evolutionary change allegedly happened, the process by which change occurs, the theory of universal common descent asserts that such major change did occur, and occurred in a completely connected, rather than disconnected or discontinuous, way, the historical pattern of change. Thus, the theory of universal common descent, UCD, depicts a monophyletic view of the history of life because it portrays all organisms as ultimately related as a single connected family. Darwin argued that the theory of universal common descent, or what he called descent with modification, best explained a variety of lines of biological evidence, including the succession of fossil forms, the geographical distribution of various species, and the anatomical and embryological similarities among otherwise different types of organisms. Modern evolutionary biologists have added the genetic similarities, or molecular homologies, of otherwise different organisms to this list of evidence supporting common ancestry. Proponents of Darwinism have often heralded the fossil record as the most decisive evidence for common descent with modification. Philip Gingerich even claimed that morphological continuity in the fossil record is the principal evidence favoring evolution as a historical explanation for the diversity of life. On the other hand, those who doubt UCD have argued that the fossil record poses a severe challenge to the theory. Still other proponents of universal common descent, including Richard Dawkins, have sought to foreclose any such criticism by arguing that we don't need fossils. The case for evolution is watertight without them, so it is paradoxical to use gaps in the fossil record as though they were evidence against evolution. So what do fossils tell us about the history of life? And how strong is the case for the theory of universal common descent, the historical part of Darwinian theory? This chapter will examine the logical structure of the arguments for universal common descent, with a particular focus on what the fossil record, and the argument from fossil progression, can tell us about whether all forms of life do, or do not, share a common ancestor. Though many theistic evolutionists portray this part of evolutionary theory as a well-established fact or theory, even as they may acknowledge doubts about the creative power of the neo-Darwinian mechanism, we have become skeptical about universal common descent. In this chapter we will explain why, and we will use the fossil evidence to illustrate how a scientifically informed person might reasonably come to doubt the arguments for universal common descent, whether those arguments are based on the fossil record or on other classes of evidence that have been marshaled in their favor. 2. Logical structure of the argument. Yet, before we look at any of evidence for, or against, universal common descent, it might be a good idea to examine the logical structure of the argument for it. Despite the presumed consensus in favor of universal common descent, there are good reasons for doubting the argument in its favor, reasons that are well illustrated by the fossil record and the competing possible interpretations of it. In particular, the argument for UCD depends on an often inconclusive or weak form of inference known as abduction. In abductive reasoning, scientists, or detectives, reason from effects, or clues, in the present back to causes in the past. To see the difference between abductive and deductive inference, consider the following argument schemata. Deduction. Data, A is given and plainly true. Logic, but if A is true, then B is a matter of course. Conclusion, hence, B must be true as well. Abduction. Data, the surprising fact B is observed. Logic, but if A were true, then B would be a matter of course. Conclusion, hence, there is reason to suspect that A is true. In deductive reasoning, if the premises are true, the conclusion follows with certainty. Abduction, however, does not produce certainty, but only plausibility or possibility. Unlike deduction, in which the minor premise affirms the antecedent variable, A, abductive reasoning affirms the consequent variable, B. In deductive logic, affirming the consequent variable, with certainty, constitutes a fallacy. The error derives from failing to acknowledge that more than one cause might explain the same evidence. To see why, consider this deductive fallacy. If it rained, the streets would get wet. The streets are wet. Therefore, it rained. Or symbolically. If R, then WW. Therefore R. Obviously, this argument has a problem. It does not follow that because the streets are wet, it necessarily rained. The streets may have gotten wet in some other way. A fire hydrant may have burst, a snow bank may have melted, or a street sweeper may have doused the streets before cleaning them. Nevertheless, that the streets are wet might indicate that it rained. Oddly, abductive arguments have the same logic structure as this fallacious form of deductive argument, they too affirm the consequent. For this reason, unless these inferences are strengthened using a process of elimination showing alternative hypotheses to be implausible, they remain weak or inconclusive. In PhD work at Cambridge, one of us, Meyer, showed that the case for universal common descent is based on several abductive inferences from various classes of biological evidence such as fossil succession, anatomical and molecular homology, embryological similarity, and biogeographical distribution. Consequently, as we have studied the case for universal common descent, we have both become gradually more skeptical about the theory because we find that the circumstantial evidence in favor of the theory is inconclusive at best. Moreover, we have found that the arguments for UCD were inconclusive for exactly the reason that abductive arguments often are. For each class of evidence allegedly favoring the theory, more than one explanation, or picture of biological history, could account for it. As we will show, the fossil record illustrates this problem in spades. 3. The case for universal common descent from paleontology. Even so, the theory of universal common descent offers an elegant explanation of several features of the fossil record. Thus, those features, at least when considered in isolation from other evidential considerations, seem to provide support the theory of universal common descent. Consider, for example, the evidence of fossil progression or succession. The fossil forms preserved in the layers of sedimentary rock progress from simple organisms in older strata, layers, to more and more complex organisms in successively younger strata. According to proponents of UCD, this stratigraphic progression in the fossil record from less to more complex forms of life supports the theory of common descent because this pattern of evidence is exactly what paleontologists should expect to find if all organisms did in fact descend from earlier less complex ancestral forms. And though there are exceptions to it, the simple to simple to complex rule is roughly true. Thus, the general pattern of successive temporal appearances agrees nicely with the Darwinian picture of the history of life. Moreover, the theory of universal common descent also explains other aspects of fossil evidence, since, again, the observed patterns are precisely what one would expect if all organisms had descended from earlier ancestral forms extending back to one universal common ancestor. For example, morphologically intermediate fossils, possible missing links. Paleontologists have discovered many fossils, such as Archaeopteryx, that appear morphologically intermediate between the putative ancestors and their descendants. By morphologically intermediate, paleontologists mean that the fossil form in question displays some, but not all, of the primitive characteristics of a putative ancestor group while exhibiting some, but not all, of the derived characteristics of a putative descendant group. A derived character is a novel or changed genetic or anatomical feature not present in a putative ancestral form or a more primitive state. Morphologically intermediate groups are not necessarily temporally intermediate, that is, they may not have been found in strata that lie in between putative ancestors and descendants. Similarly, such fossil forms may not have been found in the same geographical region as possible ancestors and descendants. Nevertheless, their similarities in form suggest the possibility of being transitional in time and space. Even though there are still groups for which such forms are lacking, many morphologically intermediate forms do exist, such as the numerous morphologically intermediate forms between land mammals and whales that have been discovered in recent decades. Since the theory of universal common descent entails the existence of temporally transitional intermediate forms, it would also predict the existence of many such, at least, morphologically intermediate forms in the fossil record. That such forms exist is, therefore, readily explained by universal common descent. Morphologically intermediate and temporally transitional series. In addition to morphologically intermediate fossils, paleontologists would also expect, based on the theory of universal common descent, that the fossil record would document some detailed transitional sequences where several intermediate forms lie temporally in between the presumed ancestors and descendants in the sedimentary strata. Some examples of such sequences have been found in the fossil record. Famous examples are the horse series illustrating the successive transformation of the primitive 3- to 4-toed legs of Eocene Hyracotherium, formerly known as Eohippus, into the single-hooved legs of modern horses. Another is the mammal-like reptile series that illustrates the transition from the primary to the secondary jaw articulation with detachment of the 3 auditory ossicles. 4. The evidence against universal common descent from paleontology. Notwithstanding the above evidences in support of universal common descent, there is also strong paleontological evidence that does not easily square with the Darwinian notion of descent with modification via a gradual series of successive transformations from ancestral to descendant forms of life. In particular, the fossil record also manifests large morphological gaps and discontinuities between different groups of organisms, especially at the higher taxonomic levels of phyla, classes, and orders, representing the major morphological differences between different forms of life. With very few exceptions, the major groups of organisms come into the fossil record abruptly, without discernible connection to earlier, and generally simpler, alleged ancestors in the fossil record. Indeed, leading evolutionary biologists and paleontologists have long acknowledged this pattern of discontinuity. Evolutionary biologist Ernst Meyer, one of the fathers of the modern neo-Darwinian synthesis, famously noted that, however we look at the living biota, discontinuities are overwhelmingly frequent. The discontinuities are even more striking in the fossil record. Moreover, since the publication of The Origin of Species in the late 19th century, our knowledge of the fossil record has greatly increased. Consequently, in most cases, fossil discontinuities can no longer be explained away as the result of alleged incomplete sampling of the fossil record. In fact, paleontologist Michael Foot of the University of Chicago has noted that, as more and more fossil discoveries have been made, the new forms that these discoveries document consistently fall within existing higher taxonomic groups, example, phyla, subphyla, and classes. In other words, these new discoveries have repeatedly failed to document the rainbow of intermediate forms expected in the Darwinian view of the history of life, especially at the higher taxonomic levels. Foote has shown, using statistical sampling analysis, that as this pattern has become more and more pronounced, it has become ever more improbable that the absence of intermediate forms reflects a sampling bias, that is, an artifact of either incomplete sampling or incomplete preservation. Increasingly, paleontologists accept that fossil discontinuities are real and need to be explained, not explained away. As Cleveland Hickman et al. Note, most major groups of animals appear abruptly in the fossil record, fully formed, and with no fossils yet discovered that form a transition from their parent group. Indeed, numerous fossil radiations, or explosions, of new forms of life are characterized by such abrupt appearances. To get a sense of how pervasive this discontinuous pattern is, and how significant these events are in the history of life, consider the following short descriptions of several of the salient examples of the abrupt appearance of new forms of life in the fossil record. The origin of life. Evidence suggests that the first living cells arose very early in the history of planet Earth, almost as soon as conditions on our planet would permit. Over the last several decades, most origin of life biologists and geochemists have placed the origin of the first life at about 3.8 billion years ago, Meyer, just after the cessation of the meteorite bombardment of the Earth called the Late Heavy Bombardment, 4.1 to 3.8 Meyer. The latest evidence from biogenic carbon in zircon crystals suggests that life was already present 4.1 Meyer in the Hadean era, even before the Late Heavy Bombardment, when life could survive only in subterranean niches. Either way, life seems to have arisen abruptly about as soon as it possibly could, given conditions on the early Earth. The origin of photosynthesis. The origin of photosynthesis was a key event that made later plant and animal life on Earth possible. Photosynthesis involves two intricate and integrated sets of complex biochemical processes known as photosystems 1 and 2, which are in turn made of many equally complex proteins. The earliest existence of cyanobacteria, the first photosynthetic cells, is documented by stromatolites from 3.7 billion-year-old rocks from the Ishwa supercrustal belt in Greenland. Nevertheless, indirect evidence suggests an even earlier origin of photosynthesis, about 3.8 billion years ago. Because the late heavy meteorite bombardment, 4.1 to 3.8 Meyer, repeatedly boiled away the existing oceans into steam atmospheres and left only subterranean environmental niches, photosynthesis was possible in the Earth's oceans only after the bombardment ceased. That implies that photosynthesis, with all its integrated biochemical complexity, originated abruptly as soon as the Earth first offered a stable and suitable environment for the process to occur. Archean genetic expansion. This event is not so much documented by real fossils as by the identification of fossil genes through genomic studies. Lawrence David and Eric Alm found that the genomic fossil record indicates that the collective genome of life expanded between 3.3 and 2.8 billion years ago. During this period, 27% of all presently existing gene families came into being by rapid evolutionary innovation. Arguably, the generation of this amount of new genetic information vastly exceeds the creative power of the neo-Darwinian mechanism of mutation and natural selection, given the extreme rarity of functional genes and proteins within the space of possible DNA and amino acid sequences. As Meyer argued in Chapter 2 of this volume, a randomly driven mutational search is overwhelmingly more likely to fail than to succeed in finding even one functional gene, let alone all the many genes that arose during this Archean expansion, in available evolutionary time. Avalon explosion. During the Ediacaran, the latest period of the Precambrian era, an enigmatic group of organisms appear abruptly in the fossil record. Radiometric dating studies fix the date for the first appearance of these Ediacaran fauna at about 575 to 565 million years ago, Meyer. These strange marine organisms, Garden of Ediacara, include microbial mats covering the sea bottom and enigmatic large sessile organisms that lack any visible feeding apparatus, and mostly have a quilted body with glide symmetry and fractal growth. Late Precambrian era sediments around the world have yielded three main types of Ediacaran fossils. The first group consists of the Precambrian sponges. The second is the distinctive group of fossils from the Ediacaran hills in Australia. The creatures fossilized there include such well-known forms as the flat, air mattress-like body of Dickinsonia, the enigmatic Sprigina, with its elongated and segmented body and alleged head shield, and the frond-like Charnia. The third group of fossils, named Kimberella, discovered in the cliffs along the White Sea in northwestern Russia, have been claimed to be primitive mollusks, but this identification is highly controversial. Nevertheless, apart from sponges and a few controversial fossils that have been attributed to algae, cnidarians, and primitive mollusks, the Ediacaran fauna have no obvious relationship to later life forms, and their systematic status is highly disputed, ranging from identifications as giant protists, to representatives of an independent multicellular kingdom, to metazoan animals, or even lichens. Whatever their classification, all groups originate abruptly without any known putative ancestors during what is now known as the Avalon explosion, 575 to 565 Meyer. Indeed, the Ediacaran fossils provide evidence of a puzzling leap in biological complexity. Before the Ediacaran organisms appeared, the only living forms documented in the fossil record for over 3 billion years were single-celled organisms, colonial algae, and possible sponges. Although the humble Ediacaran biota look simple beside most of the later Cambrian animals, they exhibit a much higher degree of complex organization than the single-celled organisms and colonial algae that preceded them. Cambrian explosion. The Cambrian explosion refers to a dramatic period in the history of life when many new and anatomically sophisticated animals appeared suddenly in the sedimentary layers of the geologic column without any discernible evidence of simpler ancestral forms in the earlier layers below. Fossil discoveries during this period attest to the first appearance of animals representing more than 20 phyla, the largest division of animal classification, as well as many more subphyla and classes, each manifesting distinctive body plans, where a body plan represents a unique arrangement of body parts and tissues. Indeed, animals representing most of the body plans that have ever existed on Earth first appeared during this explosive event. One especially dramatic fact of the Cambrian explosion is the first appearance of many novel marine invertebrate animals, representatives of separate invertebrate phyla, subphyla, and classes in the traditional classification scheme. Some of these animals have mineralized exoskeletons, including those representing phyla such as echinoderms, brachiopods, and arthropods, each with their clearly distinct and novel body plans. Several unexpected features of the Cambrian explosion from a Darwinian point of view are, 1. The sudden appearance of novel animal forms, 2. An absence of transitional intermediate fossils connecting the Cambrian animals to simpler Precambrian forms, 3. A startling array of completely novel animal forms with novel body plans, and 4. A pattern in which radical differences in form in the fossil record arise before more minor, small-scale diversification and variations. This latter pattern turns on its head the Darwinian expectation of small incremental changes only gradually resulting in larger and larger differences in form. The abruptness of the explosion is also dramatic from both a geological and an evolutionary standpoint. Most experts estimate the duration of the Cambrian explosion at about 10 to 25 million years, and date the explosion around 540 to 515 Meyer. Others emphasize that the main pulse of this event occurred within only 530 to 520 Meyer. Other studies even imply that between 13 and 16 new animal phyla arose within a narrow 5 to 6 million year window of the larger explosive radiation. In any case, most Cambrian experts agree that the majority of Cambrian animal phyla lack any putative fossil ancestors within the preceding Ediacaran biota. Thus, the Cambrian explosion has been variously called evolution's Big Bang and Darwin's dilemma. Great Ordovician Biodiversification Event, GOBE. While general animal body plans representing distinct phyla, subphyla, and classes first appeared in the Cambrian explosion, these marine invertebrate groups greatly diversified on lower taxonomic levels, example, about 300 new families, during a relatively short period of time in an event known as the Great Ordovician Biodiversification, about 485 to 460 Meyer. This explosive diversification of marine life has been called Life's Second Big Bang by James O'Donoghue, who mentions that the Ordovician explosion was every bit as momentous for animal evolution as the Cambrian one. Devonian Nectarine Revolution. Christian Klug et al. Described a radical change in the composition of the marine fauna of the early Devonian. While previously the marine ecosystems were dominated by planktonic, drifting, and demersal, near-sea bottom, taxa, between 410 and 400 Meyer a very sudden and enormous expansion of marine nectaric, actively swimming, animals occurred in which groups such as ammonoid cephalopods and jawed fish make their first appearance. Within just 10 million years such activity active swimmers increased from only 5% to about 75% of the marine fauna. Odontode explosion. The term odontode explosion was coined by Fraser et al. For the sudden appearance of vertebrate dentition. Within 10 million years, 425 to 415 Meyer, between the late Silurian and early Devonian, all major groups of jawed fish with teeth and tooth-like structures, odontodes, appear abruptly in the fossil record. These include stem gnathostomes like the Arthrodeiron Entylognathus, 423 Meyer, spiny sharks or Acanthodii, Neropizocanthus, 423 to 419 Meyer, the oldest known cartilaginous fishes or chondrichthyes, sharks like Stigmatus and Plectrodus, 423 to 419 Meyer, and the oldest known bony fishes or Osteichthyes, the latter already with the modern subgroups of lobe-finned Sarcopterygii, Guayu, 423.5 Meyer, and ray-finned Actinopterygii, Mimania, 415 Meyer. Siluro-Devonian radiation of terrestrial biotas. The sudden origin and diversification of vascular land plants, Tracheophyta, in the late Silurian and early Devonian is one of the great mysteries in the history of life. One of the two oldest known vascular land plants, Barraguinacea, already belongs to the modern subgroup of club mosses. Richard Bateman et al. Conclude that the Siluro-Devonian primary radiation of land biotas is the terrestrial equivalent of the much-debated Cambrian explosion of marine faunas. Carboniferous insect explosion. In the Pennsylvanian, Upper Carboniferous era, between 318 and 300 Meyer, when the world was dominated by vast swamp forests, a large diversity of different winged insect groups appeared suddenly without any known transitional forms in the older Mississippian, Lower Carboniferous, or Devonian strata. These include not only giant paleopterous insects like the extinct Paleodictyopterans, mayflies, and dragonflies, or primitive neopterous insect orders like stoneflies, roaches, and orthopterans, but also thrips, bugs, and even advanced holometabolans like wasps, beetles, and scorpion flies. Triassic explosion. This event was also called the early Triassic Metazoan radiation or post-Permian radiation. No new phyla and classes, but many new orders and families originate abruptly after the end Permian mass extinction, about 252 Meyer, among marine invertebrates, example, bivalves and ceratites, insects, example, Coleoptera and Diptera, and tetrapods, see below. Peter Ward explains that the diversity of Triassic animal plans is analogous to the diversity of marine body plans that resulted from the Cambrian explosion. It also occurred for nearly the same reasons and, as will be shown, was as important for animal life on land as the Cambrian explosion was for marine animal life. Early Triassic terrestrial tetrapod radiation. Directly after the Great Permo-Triassic mass extinction, the first representatives of modern tetrapod taxa appear suddenly, within a short window of time, between 251 and 240 Meyer. These include the first dinosaurs, Niosaurus, the first turtles, Popocheles, the first lizard relatives, Lepidosauramorpha, Paliguana, the first crack relatives, Crurotarsi, Tenodiscosaurus, and the first mammal-like animals, Mammalia forms, Haramyida. Except for the latter two groups, they all appear virtually out of thin air, without discernible connections to any known ancestors. Early Triassic marine reptile radiation. After the Great Permian mass extinction, 15 different families of marine reptiles appear abruptly between 248 and 240 Meyer in the early Triassic. They include, for example, ichthyosaurs, plesiosaur-like pistosaurids, hooposuchians, nothosaurs, thalatosaurs, pachypleurosaurs, tanistrophids, placodontians, and the enigmatic Aptodentatus. A vertebrate paleontologist who is an agnostic and a renowned scientist specializing in ichthyosaurs, and who must remain anonymous to protect his career, told us that the sudden appearance of viviparous, fully formed fish-like ichthyosaurs within 4 million years after the Permo-Triassic mass extinction made him doubt the neo-Darwinian story. Mid-Triassic gliding reptile radiation. Within only 2 million years of the mid-Triassic, 230 to 228 Meyer, there is a sudden appearance of gliding and flying reptiles, like Charovipteryx, with wings on the legs, Macistotrachelos, and the unrelated Kuaniosauridae, with gliding membrane across lateral rib-like projections, Longisquama, with long feather-like scales on the back, and the earliest pterosaurs like Priondactylus. Mosasaur radiation. Sudden discontinuous origins are found not only in the history of higher taxa but also within subordinate groups. A good example is the abrupt origin and diversification of mosasaurs in the last 25 million years of the Upper Cretaceous, when they are said to have evolved from 1-meter-long shore-dwelling lizards, Agialosauridae, into fully marine snake-like giants of up to 17 meters length, Mosasauridae. They quickly diversified into numerous species around the world, filling different ecological niches. Putative ancestors of mosasauroids prior to the late Cretaceous are not known. Moreover, even its proposed sister taxon Coniosaurus is of late Cretaceous age and thus not a plausible ancestral precursor. Any evolutionary relationship to recent monitor lizards and/or snakes is also contested and a matter of considerable debate among specialists. Radiation of flowering plants. Charles Darwin called the abrupt origin of flowering plants during the Cretaceous period an abominable mystery. Indeed, nearly all early fossils of modern angiosperms first appeared abruptly in the Cretaceous and then rapidly diversified between 130 and 115 Meyer. Darwin was deeply bothered by the pattern of their origin because the seemingly sudden appearance of so many angiosperm species in the upper chalk conflicted strongly with his gradualist perspective on evolutionary change. Though paleontologists in China have recently found a few angiosperms from the mid-Jurassic period, such as Euanthus, Dura herba, and Euhania, the classification of these fossilized plants as modern angiosperms remains in some dispute. There is also no evidence that these plants were ancestral to the later Cretaceous groups, and the paleontologists who have classified them have not proposed them as such. Indeed, none of these mid-Jurassic period plants can be unambiguously attributed to any subgroup of modern angiosperms, all of which did first appear in the early Cretaceous. Thus, the enigmatic rise of angiosperms still represents an inextricable knot, an unresolved puzzle for those who assume the common ancestry of all forms of life. Radiation of modern placental mammals. The first orders of placental mammals also appear abruptly in the fossil record, during the Paleocene epoch between 62 and 49 Meyer, without known precursors. Paleontologists call this series of events the mammalian radiation. According to J. David Archibald, within approximately 15 million years of dinosaur extinction, most of the 20 extant orders of placentals had appeared along with some 16 other orders that are now extinct. This was a truly explosive radiation and diversification. Not only do many, probably about 15 of the extant, mammalian orders appear suddenly, but when they appear they are already separated into their distinctive forms. For example, the orders Carnivora, which includes bears, Chiroptera, which includes bats, and Perissodactyla, which includes horses, all first appear and are clearly differentiated from each other by their distinctive forms and features. The first fossil bat, for instance, is unquestionably a bat, capable of true flight. Yet we find nothing resembling a bat in the earlier Mesozoic fossil record. Radiation of modern birds. The lineages of 95% of modern bird species also originated abruptly during the Paleocene epoch or the Tertiary, or Paleogene, period, as did most of the mammalian orders. Just like the placental mammalian radiation, the abrupt appearance of modern birds has been dated to a similarly narrow window of time from 65 and 55 Meyer. The recent genomic analysis by Richard Prum presented a comprehensive time-calibrated phylogeny of modern birds. This work suggests that only 4 bird lineages, ancestral species of ratites, Galloanseres, strisaurs, and the common ancestor of all remaining neoaves, predated and survived the mass extinction event marking the Cretaceous-Tertiary, or Cretaceous-Paleogene, boundary. The most species-rich group, neoaves, originated abruptly and diversified rapidly after this event. This avian radiation has been appropriately called the explosive evolution of avian orders, avian explosion, and even Big Bang for tertiary birds. Moreover, no undisputed fossils of crown group neoaves have been found in sediments from the Cretaceous or older, rendering dubious molecular studies placing the origin and diversification of modern avian orders prior to the Cretaceous-Tertiary boundary. Origin of genus Homo. John Hawkes et al. Suggested that our own genus, Homo, originated abruptly 2 million years ago with sudden interrelated anatomical changes. This inspired a press release with the headline, "New Study Suggests Big Bang Theory of Human Evolution." Hawkes et al. Also emphasize that no gradual series of changes in earlier Australopithecine populations clearly leads to the new species, and no Australopithecine species is obviously transitional. This may seem unexpected because for 3 decades Habeline species have been interpreted as being just such transitional taxa, linking Australopithecus through the Habelines to later Homo species. But with a few exceptions, the known Habeline specimens are now recognized to be less than 2 MYR old and therefore are too recent to be transitional forms leading to H. sapiens. See Chapter 11, by Casey Luskin, for a more detailed discussion of the hominid fossil record. The top-down pattern of appearance. This pervasive pattern of fossil appearance raises an additional difficulty for the theory of universal common descent and the Darwinian picture of the history of life. Darwinian theory, both classical and modern, implies that as new animal forms first began to emerge from a common ancestor, they would be quite similar to each other, and that larger differences in the forms of life, what paleontologists call disparity, would emerge only much later as the result of the accumulation of many small incremental changes. In its technical sense, disparity refers to the major differences in form that separate the higher-level taxonomic categories such as phyla, classes, and orders. In contrast, the term diversity refers to minor differences among organisms classified as different genera or species. Put another way, disparity refers to life's basic themes, diversity refers to the variations on those themes. According to the theory of universal common descent and the current understanding of how the mutation, natural selection mechanism works, the differences in form, or morphological distance, between evolving organisms should increase gradually over time as small-scale mutations accumulate by natural selection to produce increasingly complex forms and structures, including, eventually, new body plans. In other words, one would expect small-scale differences or diversity among species to precede large-scale morphological disparity among phyla. As the former Oxford University neo-Darwinian biologist Richard Dawkins puts it, what had been distinct species within one genus become, in the fullness of time, distinct genera within one family. Later, families will be found to have diverged to the point where taxonomists, specialists in classification, prefer to call them orders, then classes, then phyla. Darwin himself made this point in The Origin of Species. In explaining his famous branching tree diagram, he noted how higher taxa should emerge from lower taxa by the accumulation of numerous slight variations. The actual pattern in the fossil record, however, contradicts this expectation. Instead of more and more species eventually leading to more genera, leading to more families, orders, classes, and phyla, the fossil record shows representatives of separate phyla appearing first, followed by lower-level diversification on those basic themes. For example, during the Cambrian explosion representatives of many higher taxa such as phyla and classes, each representing distinctive body plans, first appear abruptly in the fossil record. Only later do different order and family and genus-level representatives of those distinctive body plans originate, in events such as the Great Ordovician biodiversification event or the mammalian radiation, for example. As paleontologists Douglas Irwin, James Valentine, and Jack Sepkoski note, the fossil record suggests that the major pulse of diversification of phyla occurs before that of classes, classes before that of orders, orders before that of families. The higher taxa do not seem to have diverged through an accumulation of lower taxa. Yet, the theory of universal common descent depicts, and predicts, just the opposite, the proliferation of species and other representatives of lower-level taxa occurring first and then building to the disparity of the highest taxonomic differences such as those between different phyla or different classes. Thus, the top-down pattern of appearance on display in the fossil record provides another evidential challenge to UCD. The polyphyletic interpretation of the fossil record. The pattern of appearance of major groups of organisms in the fossil record, both in the abruptness and discontinuity of those appearances and in the unexpected way in which disparity precedes diversity, seems to contradict the monophyletic picture of the history of life entailed by the theory of universal common descent. That suggests the possibility, at the very least, that the monophyletic picture may not be the one that best fits the fossil evidence. True, universal common descent does anticipate the progression from less complex to more complex forms of life that is generally evident in the fossil record. But the pattern of discontinuity and abrupt appearance seems on its face to contradict the way in which the theory portrays the history of life. Similarly, the top-down pattern of appearance contradicts its picture of the gradual accumulation of small incremental change over time. Indeed, UCD portrays the history of life as a great branching tree in which each new lineage emerges through just such a process of gradual and continuous, rather than abrupt and discontinuous, change. Of course, proponents of a monophyletic view have sought to offer auxiliary ad hoc hypotheses, such as the artifact hypothesis, in order to explain away the absence of expected evidence of genuine transitional fossils, i.e., temporal intermediates, connecting alleged ancestors and descendant forms in the sedimentary record. Nevertheless, conjoining UCD with such an auxiliary hypothesis is by no means the only way to explain the pattern of discontinuity evident in the fossil record, if it provides an adequate explanation of that discontinuity at all, see below. Consider, instead, the polyphyletic view of biological history. It depicts the history of life as an orchard of separate, disconnected trees in which major new groups of plants and animals are introduced into the fossil record progressively and discontinuously. This view explains fossil succession equally well, but, arguably, describes the discontinuous pattern of appearance more accurately, or, at least, more naturally and simply, without auxiliary hypotheses, than a monophyletic view does. Indeed, pervasive discontinuity is precisely what one should expect to find in the fossil record based on a polyphyletic view of the history of life. The polyphyletic view and morphological intermediates. But what about the intermediate forms of life discussed above that proponents of common descent cite in support of their theory? How does their existence square with the claim that the fossil record shows a pervasive discontinuity? And how would a polyphyletic view explain such intermediates? Recall, first, the distinction we made above between morphological and temporal intermediates. The vast majority of all intermediates in the fossil record exemplify the morphological rather than the temporal kind. For a fossil to be demonstrably part of a temporal sequence, that intermediate fossil must lie between a plausible ancestor and its possible descendants in the sedimentary strata. Consequently, when proponents of universal common descent assert that fossil intermediates are very common, see above, and doubters of descent claim that transitional fossil are mostly absent, neither side is strictly incorrect. Instead, both sides often talk past each other because they have different types of intermediates in mind. When proponents of universal common descent talk about transitional fossils, they usually refer to fossils such as Archaeopteryx that exhibit a mosaic of characters wherein some but not all of the characters from a proposed putative ancestor group, such as reptiles, are present in the intermediate form, while at the same time some, but not all, of the characters of the putative descendant group, such as birds, are also present in the intermediate. As noted above, the theory of universal common descent would expect that many such morphologically intermediate forms would be present in the fossil record, and, indeed, they are. Thus, UCD can offer a ready explanation for the presence of many such intermediates in the record, especially the many forms that lie morphologically in between different higher taxa, i.e., orders and families. It must be noted, though, that most of such transitional fossils also exhibit specialized anatomical features that exclude them from the direct ancestral lineage and place them on side branches of the tree close to the alleged descendants. They are not putative ancestors. Nevertheless, a polyphyletic view of the history of life can account for such morphologically intermediate forms as well. Given the otherwise pervasive discontinuity and absence of genuine transitional sequences, i.e., temporal intermediates, between major groups of organisms discussed above, proponents of a polyphyletic view do not regard fossils such as Archaeopteryx as representatives of a temporally transitional sequence leading from ancestor to descendant, nor need they do so, but instead as precisely a mosaic of traits produced by an intelligent designer who, like human engineers or artists of our acquaintance, may choose to combine different traits into unique combinations exemplifying a distinctive form with elements or parts in common with two or more other products of his creation, see also note 60. The polyphyletic view, discontinuity, and alleged transitional intermediates. For this reason, to decide whether a polyphyletic or monophyletic view of the history of life best fits the data, we think the most important class of evidence to consider remains the pervasive pattern of discontinuity and the abrupt appearance of major groups of organisms. As we noted earlier, the attempt to explain that pattern away as an artifact of incomplete sampling of, or incomplete preservation within, the fossil record has largely failed within paleontology. Meyer explains more about why paleontologists increasingly reject the artifact hypothesis in Chapter 3 of his book Darwin's Doubt. And yet, evidence of true transitional sequences, i.e., temporal intermediates, as we have called them, is extremely rare within the fossil record, especially between higher taxonomic groups. Even the most dramatic of the alleged transitional sequences, such as the mammal-like reptile sequence and the land mammal-to-whale sequences, both touted as proof positive of universal common descent, are 1. At best, extremely rare exceptions to an otherwise pervasive pattern of discontinuity, or 2. At worst, not at all the evidence of a continuous transformation that proponents of universal common descent claim. Consider the alleged sequence to the fully aquatic whales. Though often cited as an example of a smooth evolutionary transition, this alleged transitional sequence itself displays dramatic evidence of abrupt appearance. In the walking whales, from land to water in 8 million years, leading cetacean paleontologist J. G. M. The Wisson admits that in a dramatic transition, whales were undergoing fast evolutionary change, with features that change abruptly. The Wisson likens the evolution of whales from land mammals to converting a bullet train into a nuclear submarine. Whales, he notes, started out with a perfected body adapted to life on land. They changed it, in about 8 million years, to a body perfectly tuned to the ocean. More recent fossil evidence shows that the first fully aquatic whales, the basilosaurids, appeared even more abruptly than previously thought. Indeed, basilosaurids first appeared 49 million years ago, perhaps within only 4.5 million years after the earliest Pachycetidae, a family of terrestrial mammals that are supposedly ancestral to whales. The basilosaurids even predate some of their supposed protocetid ancestors, such as the 47.5 million-year-old protowhale Myocetus, a mammal that gave birth on land, had well-developed hind limbs, and lacked even rudimentary tail flukes. Indeed, the phylogenetic tree based on cladistic analysis of fossils does not reveal a gradual origin of aquatic adaptations. Instead, the defining features of true whales appear abruptly in the clade Pelagiceti. Similar, though arguably more acute, problems afflict the putative reptile-like mammal transitional sequence. Thus, though common descent and its fully connected monophyletic picture of biological history can explain some of the fossil evidence, such as the progression of increasingly complex forms and the increasing similarity of fossil form to modern forms of life as observers ascend the stratigraphic column, the polyphyletic view can explain those same classes of evidence and can, in our judgment, explain other aspects of the fossil evidences, such as fossil discontinuity, a top-down pattern of appearance, and the pervasive absence of genuine temporal transitional intermediates, more adequately and more simply than can a monophyletic view. Indeed, a polyphyletic view would seem to explain and describe more adequately the overall pattern of increasing complexity and abrupt appearance than would a monophyletic view. Other classes of evidence and the theory of universal common descent. We have found that this same logical relationship obtains between other classes of biological evidences and these competing views of biological history. If only some facets of the evidence in question are considered, then a monophyletic view of the history of life explains the evidence as well as a polyphyletic view. But if other facets of that evidence are considered, then a polyphyletic view ends up providing a more adequate explanation of all the facets of that relevant class of evidence. Consider, for example, molecular homology, the class of evidence that many evolutionary biologists think supports UCD most decisively. When biologists compare the amino acid sequences of proteins and genes in different species, they often find that they are quite similar in the letter-by-letter arrangement of their information-bearing subunits. Comparisons of the chimp and human genomes have indicated that the two are between 95% and 99% similar in sequence. Proponents of UCD explain this similarity as the result of chimpanzees and humans having a common ancestor, one that possessed an ancestral genome that later evolved in two slightly different ways. But that's only one possible explanation. The similarity between different genes and proteins in different organisms also might have arisen separately as the result of an intelligent designer choosing to provide similar molecular-level functional capabilities in different organisms. For example, on this view, hemoglobin proteins in chimps and humans should have similar amino acid sequences or structures, as they do, since they perform the same function in each animal, namely, carrying oxygen in the bloodstream. Thus, as with fossil progression, the evidence of sequence similarity admits more than one explanation. Moreover, as was the case with the fossil evidence, an alternative picture to the monophyletic view of biological history better accounts for other aspects of the molecular evidence. Consider, if Darwin's tree of life picture is accurate, then we should expect different types of biological evidence to point to the same phylogenetic tree. Since life had only one history, then a family history of organisms based on comparative anatomy should match one based on comparisons of DNA, RNA, and proteins. Many studies have shown, however, that trees derived from analyses of anatomy often conflict with trees based on biomacromolecules. Some recent examples for molecular and morphological data producing wildly different phylogenetic trees are grasses, metazoan animals, reptiles, i.e., the position of turtles, and lizards, all of which contradict the result one would expect based on the theory of universal common descent. Worse, various molecular analyses often generate widely different evolutionary trees. As biologist Michael Lynch observes, analyses based on different genes, and even different analyses based on the same genes, can yield a diversity of phylogenetic trees. More recently, genomics experts have found thousands of genes in different organisms with no known similarity to any other known gene. The pervasiveness of these non-homologous orphan genes is completely unexpected, given UCD. Yet, if different forms of life originated discontinuously and separately, then there would be no reason to expect that trees derived from different analyses would generate a single convergent tree. Moreover, if different forms of life were intelligently designed, with a mosaic of characteristics, some of which they share in common with some organisms and others of which they share in common with different organisms, then we would expect phylogenetic analyses to generate conflicting trees depending on which character was chosen. Indeed, phylogenetic analyses of different characters present in several different human-designed technological objects have been shown to generate precisely such conflicting trees. Finally, there are instances where the evidence for UCD has simply crumbled. In The Origin, Darwin claimed that embryos of different classes of vertebrates progress through similar phases of development as they grow from embryos to adults. He thought this indicated that different vertebrate classes shared a common ancestor in which that common pattern of development first originated. It turns out, however, that different classes of vertebrates do not progress through similar phases of embryological development. Yet, Darwin regarded alleged similarities in vertebrate development as the strongest single class of facts in favor of common descent. 6. Conclusion. In summary, the case for UCD rests in part upon 1. Factual claims that have evaporated, 2. Circumstantial evidence that admits alternative explanation, and 3. Evidence, such as fossil discontinuity and conflicting phylogenetic trees, that is better explained by a polyphyletic view of biological history. For that reason, we have both become skeptical about the theory of universal common descent and versions of theistic evolution that affirm this second meaning of evolution, as described in the scientific and philosophical introduction to this volume. 11. Universal common descent, a comprehensive critique. Casey Luskin. Summary. Some theistic evolutionists will occasionally acknowledge problems with the mechanism of mutation and natural selection, but almost all theistic evolutionists claim that the historical part of Darwinian theory, universal common descent, is beyond dispute. Since Darwin's time, the theory of universal common descent has rested upon a number of independent lines of evidence and argument: biogeography, fossils, anatomical homology, and embryological similarity. In recent decades, molecular homology has been added to that list. This chapter will show that each of these separate lines of evidence is equivocal at best and that, instead, many new lines of evidence cast serious doubt upon the supposed congruence of these lines of evidence, challenging the case for universal common descent. Some theistic evolutionists will occasionally acknowledge problems with the mechanism of mutation and natural selection, or at least will decline to defend its creative power. Nevertheless, almost all theistic evolutionists claim that the historical part of Darwinian theory, universal common descent, is beyond dispute. Prominent theistic evolutionists treat universal common descent as almost an axiom of all biological science, and ridicule skeptics through comparisons to geocentrists or flat earthers. Since Darwin's time, the theory of universal common descent has rested upon a number of independent lines of evidence and argument: biogeography, fossils, anatomical homology, and embryological similarity. In recent decades, molecular homology has been added to that list. This chapter will show that each of these separate lines of evidence is equivocal at best and that, instead, many new lines of evidence cast serious doubt upon the supposed congruence of these lines of evidence, challenging the case for universal common descent. As other chapters have discussed, theistic evolution essentially takes a fully materialistic evolutionary view of biological history and says, by the way, this is how God did it. But the term evolution can have different meanings, some of which are controversial and some of which are not. For many, evolution simply means change over time. Both theistic evolutionists and Darwin skeptics affirm that this definition of evolution is uncontroversial and correct. A second definition is universal common ancestry, the hypothesis that all living organisms are genetically related through descent with modification. Under this view, not only are all living humans related to one another, but we also share a common ancestor with apes, and going back farther, we're related to everything from horses to tuna fish to broccoli to foot fungus and bacteria. This definition of evolution is controversial among many, though not all, Darwin skeptics, and is increasingly controversial among evolutionary biologists. The third definition of evolution claims that natural selection acting upon random mutations was the driving mechanism behind the history of life. This definition is the most controversial among scientists both inside and outside of the evolutionary community, and it holds that the mechanisms producing change over time, definition 1, and common ancestry, definition 2, were apparently blind and undirected. Other chapters in Section 1, Part 1 of this book have amply addressed the inadequacy of evolution's mechanism, the third definition. The purpose of this chapter is to examine only the second definition, universal common ancestry. At first glance, common ancestry may not seem crucial to addressing theistic evolution. After all, the third definition is the one that addresses whether the history of life was unguided, a central question in the debate over theistic evolution. Moreover, common ancestry is compatible with intelligent design. For example, one possible way to view intelligent design is that God actively guided the history of life, but did so in a manner such that organisms share a common ancestry. Such a view supports both intelligent design and common ancestry yet avoids many of the logical, philosophical, and scientific difficulties that theistic evolution encounters when claiming that the entire history of life appears unguided, even though it really wasn't. Nonetheless, for a variety of reasons, common ancestry is an important part of this conversation. First, the pursuit of truth is of the utmost importance. If universal common ancestry is true, we should want to know about that. If it isn't, we should modify our views accordingly. Second, theistic evolutionists devote much energy to arguing for common ancestry. They often mistakenly cite evidence for common ancestry as evidence for the full-blown Darwinian story and an apparently naturalistic history of life, conflating the second and third definitions of evolution. While this rhetorical strategy is logically flawed, evidence for common ancestry is not necessarily evidence for blind natural selection. If the evidence for universal common ancestry is weak, then their argument faces not just a logical problem but also a factual one. Moreover, in practice common ancestry sometimes, though not always, serves as a gateway belief, taking people away from an intelligent design-based view to a full-throated theistic evolutionary view, and for some, then onto an atheistic view. The importance of common ancestry to this conversation is seen in that theistic evolutionists commonly argue for their view not by citing evidence for natural selection but rather by focusing on the evidence for common ancestry, and they often do so using the strongest of language. Finally, once the mechanism of evolution, definition 3, comes under scrutiny, and life's history no longer appears unguided, then evolutionary scientists lose an important rationale for claiming that all life is genetically related. If life evolved through an apparently unguided process like natural selection, then it follows that all life must be related. But if all life is not related, this challenges standard neo-Darwinian accounts of biological history. Thus, another important reason to discuss common ancestry is that if life is not universally related, this undercuts a core problematic tenet of theistic evolution. For these reasons, it is appropriate to devote some space to scientifically examining common ancestry. Most of the other chapters in this section of the book, Section 1, Part 2, will focus on specific claims of human-ape common ancestry. This chapter, however, will focus more broadly on universal common ancestry, the idea that all living organisms are related. Theistic evolutionists strongly endorse common ancestry. The importance of common ancestry to theistic evolution is witnessed in the extremely strong language that theistic evolutionists use when defending the concept. Francis Collins argues in the language of God that the conclusion of a common ancestor for humans and mice is virtually inescapable. In Coming to Peace with Science, biologist and former BioLogos president Darrell Falk writes regarding mammals that virtually all geneticists are convinced they share common ancestors. Another frequent BioLogos author, Dennis Venema, a biologist at Trinity Western University, argues that numerous independent lines of genomics evidence strongly support the hypothesis that our species shares a common ancestor with other primates. Robert Asher, a paleontologist at Cambridge University, writes in Evolution and Belief, Confessions of a Religious Paleontologist, that the idea that the natural world around us does not teem with evidence in support of Darwin's theory of evolution, that humanity does not share common ancestry with other forms of life on Earth via the mechanism of descent with modification, is profoundly mistaken. He further charges that creationists are incapable of a fair, honest evaluation of the data regarding common ancestry. In a 2011 InterVarsity Press book, Francis Collins and physicist Carl Giberson compare those who doubt common ancestry to geocentrists, writing, virtually all geneticists consider that the evidence proves common ancestry with a level of certainty comparable to the evidence that the Earth goes around the Sun. Elsewhere Giberson employed similar rhetoric, stating, biologists today consider the common ancestry of all life a fact on par with the sphericity of the Earth, unsubtly implying that those who doubt common ancestry are no better than flat earthers. Proponents of universal common ancestry may distastefully aim to use ridicule to bully skeptics into submission, but that in itself doesn't mean common descent is therefore wrong. Despite the outlandish rhetoric, the evidence is worth carefully considering. Before we investigate the evidence, it's important to note that it is theoretically possible that common ancestry might be true, or false, at multiple levels of the taxonomic hierarchy. For example, universal common ancestry hypothesizes that all living organisms are related. Thus, universal common ancestry might be false, but common ancestry could still be true at lower taxonomic levels, such as among all animals, or all vertebrates, all mammals, or all primates, etc. Indeed, even if we question common ancestry among those various groups, everyone would probably agree that all humans share a common ancestor. Ultimately, common ancestry must be evaluated on a case-by-case basis depending on the evidence within the particular group being studied. Evaluating the case for common ancestry among every single high and low taxonomic grouping is far beyond the scope of this chapter, and probably would entail an impossibly lengthy inquiry. This chapter will thus evaluate the case for universal common ancestry as it is commonly advocated in textbooks and in popular books by theistic evolutionists and evolution advocates. This approach will afford an analysis of the typical arguments for common ancestry, and allow the reader to critically evaluate whether those arguments hold up. The case for universal common ancestry is often said to be cumulative, based on multiple lines of evidence including biogeography, fossils, DNA and anatomical similarities, and embryology. Because common descent is said to be demonstrated by multiple, independent lines of congruent evidence, those categories of evidence should be evaluated independently. This chapter will examine whether the evidence supports common ancestry in those different areas, starting with biogeography. 2. Does the evidence support common ancestry? A. Biogeography. Biogeography is the study of the distribution of organisms in both time and space over the history of the Earth. Defenders of neo-Darwinism commonly contend that biogeography strongly supports their viewpoint. For example, the National Center for Science Education, NCSE, a pro-Darwin advocacy group, cites a consistency between biogeographic and evolutionary patterns and argues, his continuity is what would be expected of a pattern of common descent. Much biogeographical data, however, has little to do with Darwinian evolution and does not provide special evidence for common ancestry. It can be easily explained as the result of migration and continental drift, too. Conventional ideas accepted by virtually everyone in this debate. However, the NCSE's arguments ignore the many biogeographical puzzles that have vexed evolutionary biologists because they show a marked discontinuity between biogeography and common ancestry. Evolutionary explanations of biogeography fail when terrestrial or freshwater organisms appear in a location, such as an isolated island or continent, at which no standard migratory mechanism can explain how those species arrived from their proposed evolutionary ancestors. In other words, take any two populations of organisms, and theistic evolution claims that if we go back far enough, they must be linked in space and time by common descent. But sometimes it is virtually impossible to explain how two particular populations arrived at their current geographical locations from some common ancestral population. For example, a severe biogeographical puzzle for common ancestry is the origin of South American monkeys, called platyrrhines. Based on molecular and morphological evidence, New World platyrrhine monkeys are thought to be descended from African Old World or Catarrhine monkeys. The fossil record shows that monkeys have lived in South America for about 30 million years. But plate tectonics shows that Africa and South America separated around 100 to 120 million years ago. Meyer, and South America was an isolated island continent from about 80 to 3.5 Meyer. If South American monkeys split from African monkeys around 30 Meyer, neo-Darwinism must somehow explain how monkeys crossed hundreds, if not thousands, of kilometers of open ocean to end up in South America. This poses a major problem for common ancestry, one recognized by multiple experts. A HarperCollins textbook on human evolution states, the origin of platyrrhine monkeys puzzled paleontologists for decades when. And how did the monkeys get to South America? Primatologists John Flegel and Christopher Gilbert explain. The most biogeographically challenging aspect of platyrrhine evolution concerns the origin of the entire clade. South America was an island continent throughout most of the Tertiary, 66 to 2.5 million years ago. And paleontologists have debated for much of this century how and where primates reached South America. For those unfamiliar with the explanations of evolutionary scientists, their responses to such puzzles can be almost too incredible to believe. They propose not that common descent might be wrong, but that monkeys must have rafted across the Atlantic Ocean, from Africa to South America, to colonize the New World. The HarperCollins textbook explains, the rafting hypothesis argues that monkeys evolved from prosimians once and only once in Africa, and made the waterlogged trip to South America. Of course, there can't be just one seafaring monkey, or it will die, leaving no offspring. Thus, at least two monkeys, or perhaps a single pregnant monkey, must have made the rafting voyage. Flegel and Gilbert admit the rafting hypothesis raises a difficult biogeographical issue because South America is separated from Africa by a distance of at least 2,600 kilometers, 600 miles, making a phylogenetic and biogeographic link between the primate faunas of the two continents seem very unlikely. But they are wedded to common ancestry, and are obligated to find such a link, whether likely or not. Unwilling to consider non-evolutionary options, they conclude, the rafting hypothesis is the most likely scenario for the biogeographic origin of platyrrhines. In other words, the unlikely monkey rafting hypothesis is made likely only because they assume common descent must be true. Needless to say, the rafting hypothesis itself faces serious difficulties. Mammals like monkeys have high metabolisms and require large amounts of food and water. Flegel and Gilbert thus concede that overwater dispersal during primate evolution seems truly amazing for a mammalian order, and conclude, he reasons for the prevalence of rafting during the course of primate evolution remain to be explained. Or, as another expert puts it, the mechanical aspect of platyrrhine dispersal is virtually irresolvable because evolutionary models must invoke a transoceanic crossing mechanism that is implausible, rafting, or suspect at best. Flegel and Gilbert compare the monkey's voyage to winning the lottery. By a stroke of good luck, anthropoids were able to win the sweepstakes. This is by no means the only case where evolutionary biologists are forced to invoke rafting or other speculative mechanisms of oceanic dispersal to explain away difficult problems. Other biogeographical conundra include the presence of lizards and large caviomorph rodents in South America, the arrival of bees, lemurs, and other mammals in Madagascar, the appearance of elephant fossils on various islands, the appearance of freshwater frogs across isolated oceanic island chains, and numerous other examples. This problem exists for extinct species as well. A 2007 paper in Annals of Geophysics notes the still unresolved problem of disjointed distribution of fossils on the opposite coasts of the Pacific. However, this paper doesn't invoke rafting, instead it proposes something even more unlikely. Populations became separated due to an expanding Earth, a long-discarded geological hypothesis, different from well-accepted modern theories of plate tectonics, that could be taken seriously only when trying to save common descent from falsification. A 2005 review in Trends in Ecology and Evolution explains the essence of the problem. A classic problem in biogeography is to explain why particular terrestrial and freshwater taxa have geographical distributions that are broken up by oceans. Why are southern beaches, not Hophagus spp, found in Australia, New Zealand, New Guinea and southern South America? Why are there iguanas on the Fiji Islands, whereas all their close relatives are in the New World? After considering several unexpected biogeographical examples, the review concludes, these cases reinforce a general message of the great evolutionist, Darwin: given enough time, many things that seem unlikely can happen. Indeed, unlikely does appear to be the message here. If you're going to retain common ancestry, you must accept some extraordinary biogeographical claims. When evolutionary scientists are forced to resort to fantastical, expanding Earth hypotheses, or unlikely accounts of species rafting across oceans, common ancestry clearly faces a challenge. B. The fossil record. A popular college-level biology textbook explains, fossils are the only direct record of the history of life. This seems generally correct, making the fossil record an ideal place for testing universal common ancestry. The textbook's author, geologist Donald Prothero, has elsewhere written that the fossil record is an amazing testimony to the power of evolution, with documentation of transitions that Darwin could only have dreamed about. If you feel otherwise, Prothero continues, then you're a creationist, who shares much in common with the neo-Nazi Jew-hating Holocaust deniers. But what do fossils say about evolution? If all living organisms are related, as universal common ancestry predicts, then the fossil record should seemingly contain transitional forms that show the intermediate stages between life's various groups. But the history of life bears a repeated pattern of explosions, where new fossil types appear abruptly, without clear evolutionary precursors. Perhaps the most famous is the Cambrian explosion, where many of the major living animal groups, called phyla, appear in the fossil record in a sudden geological eye blink, lasting 5 to 10 million years, and possibly less. Before the Cambrian, very few fossils having anything to do with modern animal phyla are found in the record. As one invertebrate zoology textbook states, most of the animal phyla that are represented in the fossil record first appear, fully formed and identifiable as to their phylum, in the Cambrian some 550 million years ago. The fossil record is therefore of no help with respect to understanding the origin and early diversification of the various animal phyla. The diversity of complex animals that appear in the Cambrian explosion is impressive, ranging from worms to arthropods to mollusks to even vertebrate fish. But some familiar animals, like dinosaurs, parrots, or camels, don't appear until much later. Some evolutionists claim this progression is sufficient to demonstrate common ancestry. It isn't. While the fact that life has changed over time doesn't bother intelligent design, the fact that reptiles, birds, and mammals don't appear until after the Cambrian period could be a major problem for neo-Darwinian evolution if, whenever these groups do appear, they do so in an abrupt fashion that betrays the predictions of neo-Darwinian common ancestry. For many of these subgroups of animals, again, they appear abruptly, in patterns of explosions. While during the Cambrian explosion numerous phyla and classes representing basic body plans originated, writes paleontologist Walter Etter, the post-Cambrian Ordovician radiation was manifested by an unprecedented burst of diversification at lower taxonomic levels. He continues, the almost exponential increase in diversity was much more rapid during this great Ordovician biodiversification event, GOBE, than at any other time, from the Cambrian to the present, noting the increase was, for the most part, abrupt. Regarding the origin of major fish groups, Columbia University geoscientist Arthur Strawler wrote that this is one count in the creationists' charge that can only evoke in unison from paleontologists a plea of nolo contendere, no contest. We also see an explosive and rapid appearance of other marine organisms such as ammonites, other hard-shelled marine invertebrates, and mosasaurs. As for plants, a paper in Annual Review of Ecology and Systematics explains that the origin of land plants is the terrestrial equivalent of the much-debated Cambrian explosion of marine faunas. Regarding angiosperms, flowering plants, scientists refer to a big bloom or explosion event. As one paper states, angiosperms appear rather suddenly in the fossil record, with no obvious ancestors for a period of 80 to 90 million years before their appearance. Land animals show similar patterns. The fossil record shows an explosion of tetrapods when terrestrial vertebrates appear. A 2011 article in Science admitted that tracing the evolutionary origin of major dinosaur groups has been a major challenge for paleontologists. A prominent ornithology textbook observes the explosive evolution of major living bird groups. Similarly, many authorities cite an explosion or explosive diversification of major mammal groups in the Tertiary. Paleontologist Niles Eldridge notes that there are all sorts of gaps, absence of gradationally intermediate, transitional forms between species, but also between larger groups, between, say, families of carnivores, or the orders of mammals. Eldridge and some others attempt to explain many of these abrupt appearances of major fossil groups through punctuated equilibrium. This model accepts that major groups of organisms appear abruptly, but attempts to offer an evolutionary explanation where new species arise in small, short-lived populations that are unlikely to leave fossil remnants of transitions. This model has many problems, and a literal reading of the fossil record consistently shows a pattern of abrupt explosions of new types of organisms, which contradicts common descent, the opposite of what we would predict from a Darwinian process of small changes adding up to larger ones. As biologist Jeffrey Schwartz at the University of Pittsburgh explains, we are still in the dark about the origin of most major groups of organisms. They appear in the fossil record as Athena did from the head of Zeus, full-blown and raring to go, in contradiction to Darwin's depiction of evolution as resulting from the gradual accumulation of countless infinitesimally minute variations. Comparing those who recognize this non-Darwinian pattern to Holocaust deniers won't make it go away. See molecular and morphological phylogenetic trees. Perhaps the most common argument for universal common ancestry encountered by students in college-level biology textbooks is the universality of the genetic code, the claim that all life uses the same nucleotide triplets to encode the same amino acids. However, the genetic code is not universal. Many variants in the genetic code are known among various organisms. If the alleged universality of the genetic code provides evidence for universal common ancestry, should its non-universality count as evidence against it? Whatever the answer, despite the variance, it is true that the vast majority of organisms use the same standard code, and all life forms employ similar types of biomolecules, such as DNA, RNA, nucleotides, and proteins. Are such widespread biomolecular similarities evidence for common ancestry? A 2010 paper in Nature, a formal test of the theory of universal common ancestry, argued yes. Universal common ancestry is primarily supported by two further lines of evidence, various key commonalities at the molecular level, including fundamental biological polymers, nucleic acid genetic material, L-amino acids, and core metabolism, and the near universality of the genetic code. The article's author, evolutionary biochemist Douglas Theobald, concluded that universal common ancestry is the best explanation for these widespread biomolecular similarities. But best compared to what? Theobald tested universal common ancestry against the exceedingly unlikely hypothesis that living organisms independently evolved the same biomolecules and sequences by sheer chance. Universal common ancestry appeared compelling only because it was being compared to a preposterous null hypothesis. As critics writing in Biology Direct observed, cognoscenti cringed when they saw the Theobald paper, knowing that it is trivial. It is trivial because the straw man that Theobald attacks in a text largely formulated in convoluted legalese is that significant sequence similarity might arise by chance as opposed to descent with modification. True, universal common ancestry is one possible explanation for many genetic similarities we observe between organisms, and probably better than chance, but are there other viable explanations? Indeed there are. Intelligent agents frequently reuse the same parts in different designs to meet functional requirements, such as reusing wheels on cars and airplanes, or reusing key computer codes in different versions of Microsoft Windows. As Paul Nelson and Jonathan Wells observe, an intelligent cause may reuse or redeploy the same module in different systems, without there necessarily being any material or physical connection between those systems. Even more simply, intelligent causes can generate identical patterns independently. If we suppose that an intelligent designer constructed organisms using a common set of polyfunctional genetic modules, just as human designers, for instance, may employ the same transistor or capacitor in a car radio or a computer, devices that are not homologous as artifacts, then we can explain why we find the same genes expressed in the development of what are very different organisms. Thus, common design, the intentional reuse of a common blueprint or components, is a viable explanation for the widespread functional similarities among the biomolecules found in different types of organisms. Universal common ancestry is not the only possible explanation. Indeed, contra Theobald's arguments for universal common ancestry, not all fundamental biomolecules are universal among organisms. As the authors of one paper found, several core components of the bacterial, DNA, replication machinery are unrelated or only distantly related to the functionally equivalent components of the archaeal eukaryotic replication apparatus, leading them to suggest DNA replication likely evolved independently in the bacterial and archaeal eukaryotic lineages. Even more striking, another paper compared the genomes of 1,000 different prokaryotic organisms and found that of the 1,000 genomes available, not a single protein is conserved across all genomes. But it isn't mere similarity among biomolecules that evolutionary biologists claim demonstrates universal common ancestry. They often claim that patterns of similar nucleotide and amino acid sequences of genes and proteins allow organisms to be organized into a phylogenetic tree of life, figure 11.1, showing the evolutionary relationships between all living organisms. This tree of life was Darwin's only illustration in The Origin of Species, and it has become the most famous icon representing his theory. But does the tree of life exist? In the 1960s, soon after the genetic code was uncovered, pioneering scientists Linus Pauling and Emil Zuckerkandl predicted that phylogenetic trees based on biomolecules would confirm expectations of common descent already held by evolutionary biologists who studied morphology, i.e., the physical traits of organisms. They declared that if the two phylogenetic trees are mostly in agreement with respect to the topology of branching, the best available single proof of the reality of macroevolution would be furnished. Presumably, then, if this prediction failed, and if there were sharp conflicts between trees built using different data sources, a compelling disproof of macroevolution would also be furnished. Hoping to validate Pauling and Zuckerkandl's prediction, biologists began sequencing genes from all manner of living organisms. In the 1990s, this led to a discovery that confounded evolutionary biologists. Life falls into three basic domains which cannot be resolved into a neat, tree-like pattern. Thus, the prominent biochemist W. Ford Doolittle lamented. Molecular phylogenists will have failed to find the true tree, not because their methods are inadequate or because they have chosen the wrong genes, but because the history of life cannot properly be represented as a tree. He explained that for many biologists, it is as if we have failed at the task that Darwin set for us, delineating the unique structure of the tree of life. The basic problem is that one gene leads to one version of the tree of life, but another gene leads to an entirely different tree. What seems to imply a closer evolutionary relationship in one case, i.e., two similar genes, doesn't in another. To put it another way, biological similarity is constantly appearing in places where it wasn't predicted by common descent, leading to conflicts between phylogenetic trees. When two trees conflict, at least one must be wrong. And if one tree must be wrong, i.e., in one case genetic similarity was not a good indicator of an evolutionary relationship, how do we know that both aren't wrong, i.e., in both cases genetic similarity is not indicating an evolutionary relationship? Numerous technical papers have noted the prevalence of contradictory phylogenetic trees among various taxonomic groups. In 1998, a study in genome research plainly observed that different proteins generate different phylogenetic trees. A 2009 article in Trends in Ecology and Evolution acknowledged, evolutionary trees from different genes often have conflicting branching patterns. A 2013 paper in Trends in Genetics reported that the more we learn about genomes the less tree-like we find their evolutionary history. Perhaps the most candid admissions came in a 2009 article in New Scientist titled, Why Darwin Was Wrong About the Tree of Life. It quoted researcher Eric Baptiste admitting that the Holy Grail was to build a tree of life, but today that project lies in tatters, torn to pieces by an onslaught of negative evidence. According to the article, many biologists now argue that the tree concept is obsolete and needs to be discarded. The paper recounted the results of a study by Michael Sivonen, which compared 2,000 genes across 6 animal phyla. In theory, he should have been able to use the gene sequences to construct an evolutionary tree showing the relationships between the 6 animals. He failed. The problem was that different genes told contradictory evolutionary stories. Sivonen succinctly explained the problem: we've just annihilated the tree of life. It's not a tree anymore, it's a different topology entirely. What would Darwin have made of that? Clearly, molecule-based trees often conflict with one another. But what about Pauling and Zuckerkandl's prediction that molecule-based phylogenetic trees should match those constructed by morphology? A review article in Nature titled Bones, Molecules, or Both? Explained that evolutionary trees constructed by studying biological molecules often don't resemble those drawn up from morphology, admitting that battles between molecules and morphology are being fought across the entire tree of life. A classic example involves attempts to construct a phylogenetic tree of the animal phyla. Traditionally, many phyla were grouped according to whether they have a central body cavity, called a coelom. But molecular data contradicted that grouping, and instead placed organisms that are morphologically very different, such as nematodes and arthropods, very close. A Nature paper reported how unexpected this grouping was, considering the greatly differing morphologies, embryological features, and life histories of the molting animals. It was initially surprising that the ribosomal RNA tree should group them together. Other fundamental animal characteristics, such as symmetry and early developmental processes, also yield a pattern of conflicting trees. Higher up the tree of life, conflicts persist. In 2014, the sequencing of various bird genomes led to the unexpected result that many types of birds that were previously thought to be closely related, water birds, birds of prey, and songbirds, evolved their groups defining traits convergently. As Nature put it, the tree of life for birds has been redrawn. The problem was, once again, after genomic data was sequenced and understood, many basic habits and lifestyles of birds no longer fit into a nested hierarchy. As a final example, a 2013 paper acknowledged problems encountered when trying to reconcile conflicting versions of the mammalian tree. Untangling the root of the evolutionary tree of placental mammals has been nearly an impossible task. The good news is that only 3 possibilities are seriously considered. The bad news is that all 3 possibilities are seriously considered. Paleontologists favor a root anchored by Xenarthra, example, sloths and anteater, whereas molecular evolutionists have favored the 2 other possible roots, Afrotheria, example, elephants, hyraxes, and tenrex, and Atlantogeneta, Afrotheria plus Xenarthra. Now, 2 groups of researchers have scrutinized the largest available genomic data sets bearing on the question and have come to opposite conclusions, needless to say. More research is needed. But is more research always solving these problems? A 2012 paper noted that phylogenetic conflict is common, and frequently the norm rather than the exception, since incongruence between phylogenies derived from morphological versus molecular analyses, and between trees based on different subsets of molecular sequences has become pervasive as data sets have expanded rapidly in both characters and species. In any case, these frequent discrepancies between molecular and morphology-based trees, and between various molecule-based trees, have led some scientists to conclude that the prediction of Zuckerkandl and Pauling was fundamentally wrong. A paper in the journal Biological Theory explained. Molecular systematics is, largely, based on the assumption, first clearly articulated by Zuckerkandl and Pauling, that degree of overall similarity reflects degree of relatedness. This assumption derives from interpreting molecular similarity, or dissimilarity, between taxa in the context of a Darwinian model of continual and gradual change. Review of the history of molecular systematics and its claims in the context of molecular biology reveals that there is no basis for the molecular assumption. To put it another way, conflicts between morphological and molecular trees seriously challenge common ancestry, and, as we will soon see, undermine the methods used to infer it. But what about those happy cases where molecule-based trees show some congruence with morphology-based trees? Does this provide some kind of special evidence for common descent? Not at all. Recall that morphological trees are based on comparing similarities in anatomical traits between different organisms. But if anatomical traits are generated by gene sequences in DNA, then it follows that organisms with more similar anatomy will typically have more similar DNA. Because of this gene morphology linkage, one need not even consider common ancestry to predict that DNA-based trees, groupings of organisms with more similar DNA, might show some resemblance to morphology-based trees, groupings of organisms with more similar anatomy. And both types of similarities, anatomical and DNA, can be explained by common design just as well as by common descent. Assumptions, epicycles, and ad hoc hypotheses. Centuries ago, when astronomers held to a geocentric model of the solar system, they would often encounter data that ran directly counter to that model. Sometimes planets would appear to temporarily move backwards, in retrograde motion. Early scientists explained away the contrary data by invoking the epicycle. Epicycles didn't actually explain anything. Such auxiliary hypotheses were adopted after the fact for the sole purpose of saving the flawed geocentric model from falsification. Eventually, scientists accepted that planets were not moving backwards and agreed that the geocentric model of the solar system was wrong. Planets orbited the Sun, not the Earth. Today, evolutionary biology faces a similar dilemma. Biologists are constantly uncovering similarities between organisms that appear in patterns not predicted by universal common descent. Proponents of neo-Darwinian evolution thus adopt modern-day epicycles, ad hoc hypotheses invoked to explain why data run counter to the tree of life hypothesis. To appreciate how this works, we must examine some of the core assumptions that underlie evolutionary tree building. First and foremost, phylogenetic trees are based on the assumption that common ancestry is true. That assumption, and it is merely an assumption, is so deeply embedded in evolutionary thinking that theorists often forget it's there. In a rare example, Elliot Sober and Michael Steele acknowledge, it is a central tenet of modern evolutionary theory that all living things now on Earth trace back to a single common ancestor, and this proposition is central because it is presupposed so widely in evolutionary research. They acknowledge that phylogenetic methods assume that a tree exists and that common ancestry is correct. Whether one uses cladistic parsimony, distance measures, or maximum likelihood methods, the typical question is which tree is the best one, not whether there is a tree in the first place. A bioinformatics textbook concurs. The key assumption made when constructing a phylogenetic tree from a set of sequences is that they are all derived from a single ancestral sequence, i.e., they are homologous. These authorities affirm a crucial point: phylogenetic methods do not demonstrate common ancestry, they assume it. In other words, evolutionary biology doesn't test whether all organisms fit into a nested hierarchy, i.e., a phylogenetic tree, but rather assumes that common ancestry is true. If the data does not fit with preconceptions about common ancestry, then various methods are used to force-fit the data into a tree. Thus, Michael Sivonen notes the pro-tree biases of tree-building algorithms. Because tree analysis tools are used so widely, they tend to introduce a bias into the interpretation of results. Hence, one needs to be continually reminded that submitting multiple sequences, DNA, protein, or other character states, to phylogenetic analysis produces trees because that is the nature of the algorithms used. A more explicitly recognized assumption is that shared biological similarity between two species indicates inheritance from a common ancestor. This assumption is reflected in the statement quoted earlier from the journal Biological Theory: degree of overall similarity reflects degree of relatedness. We'll call this the main assumption. The main assumption sounds nice in theory, but in practice, it fails constantly. For instance, under the main assumption, the reason you have 2 eyes, and your dog has 2 eyes, is that you shared a common ancestor with 2 eyes. That is a possibility. But cephalopods, arctopi and squid, also have 2 eyes, and according to standard evolutionary wisdom, there's no reason to think your most recent common ancestor with cephalopods had 2 eyes. We're not even sure if it had eyes. Perhaps, the defender of common ancestry replies, both organisms independently evolved 2 eyes, just by chance. Perhaps. But human cephalopod similarities go much deeper. Cephalopods have a camera eye with a basic design that is almost identical to human eyes. Surely such similarities are taken to indicate common ancestry, right? Wrong. According to evolutionary biologists, the extreme similarities between human and squid eyes result from convergent evolution, where two different organisms independently stumbled upon almost the same complex biological designs. Convergence can be found at some of the highest conceivable levels of complexity. For example, comb jellies have advanced nervous systems, but molecular studies suggest they branched off very close to the base of the animal tree before such complexity evolved. However, sponges, which branch off later, according to molecular data, lack such structures. This means that either animal brains evolved convergently, i.e., they arose multiple times, independently, or important and complex nervous cells were lost in sponges. Such extreme convergence is disconcerting to many evolutionary biologists. As Richard Dawkins acknowledges, it is vanishingly improbable that exactly the same evolutionary pathway should ever be traveled twice. Yet he admits there are numerous examples in which independent lines of evolution appear to have converged, from very different starting points, on what looks very like the same endpoint. Not to worry, Dawkins tells us. Rather than facing the challenge of convergent evolution, he simply declares, it is all the more striking a testimony to the power of natural selection. Beyond the fact that it is highly unlikely, extreme convergent evolution presents an even more serious problem for neo-Darwinian theory. It shows that the main assumption has failed, that biological similarity does not necessarily indicate inheritance from a common ancestor. This challenges the heart of the methodology used to infer common descent. But why should that trouble us? After all, what assumption always holds true? It should trouble us because the main assumption, that biological similarity indicates a common ancestor, fails not occasionally, but frequently. Indeed, so often does the main assumption fail, and so quick are evolutionary biologists to tolerate its failure, that we cannot help but question the many instances where biologists believe the main assumption held true. The frequent failure of the main assumption calls into doubt the core assumptions and rational bases used to construct phylogenetic trees, the lifeblood of common ancestry. One evolutionary systematist, Nicholas Matsky, maintains that these failures of methodology pose no problems for common descent, as he notes that we can statistically analyze the congruency of trees to assess whether the assumptions of tree building are holding true. In response to Darwin skeptics, he posted two phylogenetic trees, in this case, cladograms, and claimed that they demonstrated the common ancestry of arthropods. In reality, however, they demonstrated how the methods of phylogenetic reconstruction often fail to establish the assumptions that underlie common ancestry. A main goal of tree building is to construct a phylogenetic tree that minimizes the number of evolutionary events necessary to explain the observed distribution of traits. An evolutionary event is the gain or loss of a trait during the course of evolution. The fewer evolutionary events required, the better the data fits a tree. In the parlance of evolutionary biology, this is called maximizing parsimony. One statistical method of determining the extent to which a data set fits a tree-like pattern is to calculate a tree's consistency index, CI. This is found by taking the minimum number of evolutionary events required by the overall data set, divided by the number of events required by the tree. A high CI, closer to 1, indicates the data fits a tree-like pattern. A lower CI, closer to 0, usually indicates the data is inconsistent with a tree. The CI also measures the degree to which convergent evolution is required by a tree. In other words, a CI tells you how frequently the main assumption of tree building has failed. But what happens when the best tree you can find, i.e., the tree with the highest CI, still has a low CI? Consider the CIS for the arthropod trees posted by Matsky. One had CI of 0.565. This means that 43.5% of the time, a given trait or character analyzed in the data set was not distributed in a tree-like pattern, meaning the main assumption, that biological similarity results from common ancestry, failed. If an assumption fails 43.5% of the time, can one justify making it in the first place? Numerous similar examples could be given. A tree published in Nature purported to show the evolutionary relationships of many living mammals, but its CI was 0.43, meaning the main assumption failed in 57% of cases. Another Nature paper yielded a mammal tree with a CI of 0.34, a 66% failure rate. A paper using DNA to study bird relationships produced a tree with a CI of 0.36, a 64% failure rate. A 2007 paper in Proceedings of the Royal Society B produced a tree of early tertiary mammals with a CI of 0.35, the main assumption failed 65% of the time. Likewise, consider the CI of the other cladogram Matsky posted, figure 11.2, which purports to show the relationships of various Cambrian arthropods. It has a CI of 0.384, which even the original authors admit was rather low. This means that in the cladogram Matsky referenced as a demonstration of common descent, the main assumption of tree building failed 61.6% of the time. In such cases, the main assumption failed more often than it held true. Perhaps the main assumption should be rewritten as, biological similarities indicate inheritance from a common ancestor, except for when they don't. Is common descent testable? To be sure, plenty of trees enjoy much higher CIS. But even in those happy cases, how can we know whether the main assumption is valid, given how often it fails elsewhere? And if the main assumption hadn't held, would that have even mattered? Indeed, why do evolutionary biologists tolerate the frequent failure of their field's core assumptions? They tolerate it because to do otherwise would be to abandon common ancestry. Thus, they build trees even when they aren't sure if the main assumption is true, or even worse, when they know the data doesn't fit very well into a tree-like pattern. The open secret of evolutionary phylogenetic reconstruction, therefore, is that, using the statistical methods of tree construction, you can force virtually any data set to fit a tree, even if it strongly contradicts a tree-like pattern. Sometimes, even data sets that do fit a tree and are well supported by statistical analyses can sharply conflict. As scientists commenting on a Nature paper stated, trees produced by a number of well-supported studies have come to contradictory conclusions. Good statistical support for a tree, apparently, doesn't always demonstrate common descent. This demands the question, what possible pattern would refute common ancestry? Strongly supported tree-like patterns don't necessarily demonstrate common ancestry. And even when over half of the data is inconsistent with a tree, evolutionary biologists go ahead and impose a tree-like pattern. A 2010 paper reported trees with CIS under 0.1, meaning more than 90% of the data didn't fit a tree. Predictably, the authors did not question common ancestry and had their own epicycles to try to explain the non-tree-like patterns. At what point can we falsify common descent? For evolutionary biologists, it seems we cannot, because common ancestry is not on the table for falsification. Apparently no pattern is necessarily inconsistent with common ancestry because you can always invoke as much convergent evolution, or loss of traits, as needed to force the data into a tree. And when those explanations are unlikely, other epicycles, like horizontal gene transfer, incomplete lineage sorting or coalescence, and rapid evolution, to list a few, can be invoked to explain away data that doesn't fit a tree. For example, one paper found that 23% of the human genome contradicts the standard human ape tree, but unworriedly claimed the discrepancies simply result from incomplete lineage sorting. Another paper found that 30% of the gorilla genome contradicts the conventional human-ape phylogeny, and explained away the data in the same manner. Large percentages of whole genomes can contradict standard evolutionary trees, and that is never even seen as a possible challenge to common ancestry. Indeed, modern genome sequencing has discovered thousands of orphan genes, unique genes that exhibit no homology, sequence similarity, to any other known gene. These genes ought to refute common ancestry because they cannot be compared to genes from other species, and thus do not fit into any phylogenetic tree. The problem is usually ignored. Conversely, genome sequencing regularly uncovers genes that do bear homology to other known genes, but that homology should not be there because, according to common ancestry, the closest homologue is known only from very distantly related species. These cases, too, are not taken as evidence against common descent, but instead are said to indicate horizontal gene transfer, HGT, also called lateral gene transfer or LGT, where organisms obtain genes from neighboring organisms rather than from a parent. Evolutionists sometimes reply that such phylogenetic oddities arise only when studying microorganisms like bacteria at the base of the tree of life, organisms that are known to readily swap genetic material through HGT. But this objection fails, since the tree of life faces incongruities among higher organisms where such gene swapping is not prevalent. As Carl Woese, a pioneer of evolutionary molecular systematics, explains, Phylogenetic incongruities can be seen everywhere in the universal tree, from its root to the major branchings within and among the various taxa to the makeup of the primary groupings themselves. To explain away phylogenetic conflicts in higher branches, evolutionary biologists predictably argue that HGT must also be occurring in higher organisms, even if we don't directly observe it. One study found hundreds of genes in animals that were unexpected under common ancestry, but simply explained them away by invoking HGT. Another study found that the angiosperm amborella is rich in foreign genes that don't fit the standard phylogeny. HGT came to the rescue and explained why they were there. But HGT is not necessarily directly observed in these higher organisms, rather, it is inferred to occur based on the presence of so-called foreign genes. Under this mindset, genes that severely conflict with the standard phylogeny are not taken as evidence that the phylogeny might be wrong. Rather, they are taken as evidence for HGT. One paper admits this reasoning. In the phylogenetic approach, each instance of topological discordance between a gene tree and a trusted reference tree is taken as a prima facie instance of LGT. When data that contradicts a paradigm becomes evidence for the epicycles rather than against the paradigm, it's clear that the paradigm is in crisis. This is exactly what we see taking place with regard to universal common ancestry. D. Embryology. In a letter to the American botanist Asa Gray, Charles Darwin urged that embryology was by far the strongest single class of facts in favor of his theory. Much has changed in the 150-plus years since Darwin penned those words, but embryology remains a favorite line of evidence cited by evolutionists to support common descent, particularly among the vertebrates. Most modern biology textbooks will print some diagram depicting the early embryos of different vertebrate species as highly similar, then will claim that these similarities reflect common ancestry. Life Science, published by Holt, provides typical language. Early in development, the human embryos and the embryos of all other vertebrates are similar. These early similarities are evidence that all vertebrates share a common ancestor. For decades, students were also taught that ontogeny recapitulates phylogeny. Called recapitulation theory, this idea was promoted by the German biologist Ernst Haeckel, who believed that the development of an organism, ontogeny, recapitulates its evolutionary history, phylogeny. Since the standard evolutionary view holds that humans evolved from fish, recapitulation theory taught that at one point between conception and birth, we all went through a fish stage. Biologists now know that vertebrate embryos do not replay their supposed earlier evolutionary stages, and firmly hold that recapitulation theory is false. The concept has been removed from textbooks, but many textbooks still use inaccurate diagrams which overstate the degree of similarity between vertebrate embryos. Indeed, the journal Science observed that generations of biology students may have been misled by Haeckel's embryo drawings in textbooks. These drawings, commonly reprinted or adapted in textbooks, overstate the degree of similarity between vertebrate embryos in their earliest stages. According to Stephen J. Gould, Haeckel's methods can only be called fraudulent because he simply copied the same figure over and over again when depicting the embryos of different species. This led embryologist Michael Richardson to call them one of the most famous fakes in biology. In 2000, biologist Jonathan Wells published Icons of Evolution, which raised the public's consciousness about Haeckel's fraud, ultimately forcing many publishers to remove Haeckel's inaccurate drawings from most textbooks. Many textbooks, however, still claim that the early stages of vertebrate development are highly similar. But are those claims accurate? No, they are not. Embryologists have found considerable differences among vertebrate embryos from their earliest stages onward, contradicting what we are told to expect from common ancestry. Two of the earliest stages of vertebrate development are cleavage and gastrulation. During cleavage, a newly fertilized zygote undergoes rapid cell division until the embryo becomes a tiny ball of cells, laying out the basic axes that will define the body plan. Next, during gastrulation, the embryo increases in size while forming distinct germ layers which will later develop into individual organs. Yet a paper in Systematic Biology states, such early stages as initial cleavages and gastrula, tyon, can vary quite extensively across vertebrates. Likewise, a 2010 paper in Nature stated, counter to the expectations of early embryonic conservation, i.e., similarity, many studies have shown that there is often remarkable divergence between related species both early and late in development. Or, as another article in Trends in Ecology and Evolution stated, despite repeated assertions of the uniformity of early embryos within members of a phylum, development in those early stages is very varied. Rather than looking highly similar in their early stages, vertebrate embryos look more like what we see in Figure 11.4. To their credit, some evolutionary biologists acknowledge that vertebrate embryos begin development differently, but then they claim that embryos pass through a highly similar midpoint stage, called the pharyngular, or phylotypic, or tailbud stage. They propose an hourglass model, where this converging midpoint stage of development reveals common ancestry, Figure 11.5. Prominent atheist and developmental biologist P. Z. Myers named his popular blog Pharyngular, where he has argued that vertebrate embryos at the phylotypic or pharyngular stage do show substantial similarities to one another that are evidence of common descent. That's simply a fact. But does this pharyngular stage exist? In a groundbreaking study published in Anatomy and Embryology, a team of embryologists investigated this question and noted, it is almost as though the phylotypic stage is regarded as a biological concept for which no proof is needed. After photographing vertebrate embryos during this purportedly similar stage, they found differences in major traits, including body size, body plan, growth patterns, and timing of development. They conclude the evidence is, contrary to the evolutionary hourglass model, because vertebrate embryos show considerable variability during the purported phylotypic stage. In their view, this wide variation in morphology among vertebrate embryos is difficult to reconcile with the idea of a phylogenetically conserved tail bud stage. Likewise, a study in Proceedings of the Royal Society of London B found that embryological data is counter to the predictions of the phylotypic stage, since phenotypic variation between species was highest in the middle of the developmental sequence. It noted that a surprising degree of developmental character independence argues against the existence of a phylotypic stage in vertebrates. Even P. Z. Myers has conceded that early vertebrate embryos can vary greatly and that there is wide variation in the status of the embryo. But, he tried to explain why these facts pose no challenge to common ancestry. I wish I could get that one thought into these guys' heads, he wrote. Evolutionary theory predicts differences as well as similarities. That's intriguing. Earlier, Myers cited the substantial similarities between vertebrate embryos as evidence of common descent. But later, when forced to admit the wide variation among embryos, he argued that evolutionary theory predicts differences too. Perhaps so, but then how can he cite the similarities among embryos in the pharyngular stage as evidence for common ancestry? In reality, Myers's comments reflect the fact that, in practice, evolutionary theory predicts whatever it finds. In other words, common ancestry predicts nothing. Myers's logic might help common descent avoid falsification, but it doesn't construct a robust model that makes testable predictions. It seems we're back to common ancestry predicts similarities except for when it doesn't. As the old adage says, the theory which explains everything really explains nothing. 3. What's left of the congruence argument for universal common ancestry? At the beginning of this chapter, we noted that the case for common descent is often said to be cumulative, based on multiple lines of evidence including biogeography, fossils, DNA and anatomy, and embryology. How is the theory faring? In biogeography, evolutionists resort to unlikely and speculative explanations where species must raft across vast oceans in order for common descent to account for their unexpected locations. Paleontology fails to reveal the continuous branching pattern predicted by common ancestry, and the fossil record is dominated by abrupt explosions of new life forms. Regarding molecular and morphology-based trees, conflicting phylogenies have left the tree of life in tatters. Phylogenetic methods are reduced to predicting that shared similarity indicates common inheritance, except for when it doesn't. Similar problems confound embryology, where evolutionary biologists predict similarities will exist between vertebrate embryos, except for when we find differences, and then it predicts those too. Much data contradicts the sometimes made predictions of common descent, but what, if anything, does evolutionary biology actually predict? As P. Z. Myers has shown us, common descent seems to predict whatever is expedient. If there's any clear pattern here, it's this: the data often fails to fit the predictions of universal common descent, but when that happens, proponents of common descent simply change their predictions. This raises the question of the scientific status of common descent. At best, it's a scientific theory that is contradicted by much evidence. At worst, it's not even a scientific theory that makes concrete, testable predictions. For these and many other reasons, even some mainstream evolutionary scientists are becoming increasingly skeptical of universal common ancestry and the monopoly of life. A 2009 paper in Trends in Genetics noted that breakdowns in core neo-Darwinian tenets like the traditional concept of the tree of life, or that natural selection is the main driving force of evolution, indicate the modern synthesis has crumbled, apparently, beyond repair. A 2012 paper in Annual Review of Genetics explicitly doubted universal common ancestry, suggesting life might indeed have multiple origins. Another paper in Biology Direct noted that the sudden emergence of new complex life forms contradicts a tree pattern. Major transitions in biological evolution show the same pattern of sudden emergence of diverse forms at a new level of complexity. The relationships between major groups within an emergent new class of biological entities are hard to decipher and do not seem to fit the tree pattern that, following Darwin's original proposal, remains the dominant description of biological evolution. To be sure, these authors support some form of unguided materialistic evolution. But the precise reason that they are critiquing the classical evolutionary model is that much data contradicts universal common ancestry. 21st century biology seems to be following the evidence beyond universal common ancestry and the neo-Darwinian tree of life. Our friends in the theistic evolution community would be wise to follow suit, or at least to tone down their rhetoric against reasonable skeptics of universal common ancestry. 12. 5 questions everyone should ask about common descent. Paul A. Nelson. Summary. According to the theory of universal common descent, UCD, all organisms on Earth have descended by modification from a common ancestor, dubbed the last universal common ancestor, Luca. Within the past 20 years, however, a growing number of evolutionary biologists have expressed doubts that Luca ever existed. Their skepticism of Luca and hence of UCD rests on an important rule of biological inference known as the principle of continuity. The principle of continuity holds that every step in any evolutionary pathway must be biologically possible. This principle actually challenges UCD, or the tree of life, not only at its base but throughout its branches as well. 5 key questions should be asked of any hypothesis of common descent, to make sure that the hypothesis answers the demands of the principle of continuity, and also to examine the larger context within which UCD lives as a biological and historical theory. UCD should not be maintained as an axiom, but should be vulnerable to evidential challenges, like any other scientific theory. Introduction. Is common descent really a theory that no biologist doubts? Biology, a familiar aphorism tells us, is a science of exceptions. The protein hemoglobin binds oxygen molecules, O2, with the iron atoms at its center, carrying O2 in the bloodstream of animals, except when it doesn't. Hemocyanin carries O2 in animals such as the octopus and horseshoe crab, binding the molecule with copper, not iron, making their blood appear blue, not red. Draw any generalization about life on Earth, and, from the back of the room, the exceptions will stand and take a bow. A handful of biological propositions, however, seem truly exceptionless or universal in scope. All organisms are cells, or are made up of cells. All organisms have other organisms as parents. All organisms have descended with modification from a single common ancestor, itself an organism, designated the last universal common ancestor, abbreviated LUCA, at the root of the tree of life. These propositions about life on Earth are seen by most biologists to hold so generally that each is thought to rise to the status of a theory, or even a law. Thus, we have, respectively, the cell theory, the law of biogenesis, the theory of common descent. Here many biologists, including theistic evolutionists, would add, with some forcefulness, and no well-respected biologist doubts any of these theories or laws. Yet they do. Not the cell theory, or the law of biogenesis, however, today, it is the theory of common descent that is in trouble, possibly very serious trouble, from which it may never escape. Thereupon hangs a tale, which I tell in this chapter. When the late National Academy of Sciences molecular evolutionist Carl Woese wrote, "The time has come for biology to go beyond the doctrine of common descent," he was using doctrine to mean a scientific concept held dogmatically, standing in the way of biological understanding. Geneticist Craig Venter, whose company Celera Genomics independently sequenced the human genome in 2001, said that common descent was counterintuitive to him. "I don't necessarily buy that there is a single ancestor. It's counterintuitive to me. I think we may have thousands of recent common ancestors and they are not necessarily so common." Neither Woese nor Venter were motivated by creationism or intelligent design, ID, which they oppose, as do the growing number of evolutionary biologists who openly express their doubts about common descent. Rather, these biologists see the tree of life fracturing at its base, near where the thickest parts of the trunk, i.e., the major domains of bacteria, archaea, and eukarya, emerge from the prebiotic soil. LUCA, they contend, never existed as a discrete cell that was the unique ancestor to these three domains. No single organism ever lived as the your-parent to all life on this planet. Nonetheless, while these biologists see common descent in this universal sense as strictly false, they think that most groups of single-celled organisms, as well as fungi, plants, and animals, do share common ancestors higher up in the tree of life. Yet the same reasoning that leads skeptics of universal common descent to doubt the existence of LUCA can readily be extended to the supposed common ancestors of more recent branches in the tree of life. Doubts about LUCA arise from its apparent biological impossibility, under an important rule of biological inference known as the principle of continuity. As we shall see, the principle of continuity actually challenges the tree of life everywhere, not simply at its base. But common descent and continuity are themselves embedded in a wider scientific and philosophical context. How one evaluates the theory depends on the decisions one makes about that wider context, on questions such as the probability of the naturalistic origin of life, or abiogenesis, how many times abiogenesis occurred, and whether intelligent design is a live possibility. To paraphrase John Donne, no theory is an island, and common descent is no exception. The relevance of common descent to the subject of this book hardly needs elaborating. Biologos, the leading organization in the United States promoting theistic evolution, states that evolution means, among other things, that all the life forms on Earth share a common ancestor, i.e., common descent. To stress that no legitimate doubt exists about the theory, Biologos adds, "There is very little debate in the scientific community about this broad characterization of evolution. Anyone who claims otherwise is either uninformed or deliberately trying to mislead. The observational evidence explained by common ancestry is overwhelming. But serious questions about common descent have existed since Darwin's time, and are even more significant today. Let's abbreviate common descent as UCD. i.e., universal common descent. This will stand for Darwin's idea that all organisms on Earth stemmed from a single common ancestor. We'll use UCD for hypotheses about the shared ancestry of particular groups of organisms, where UCD may or may not be presupposed. The scope of UCD includes all life on this planet, and even particular CD theories, such as the common ancestry of the animals, encompass enormous swaths of organismal diversity. Given this vast breadth, it is impossible in a single chapter, or even an entire book, to address every dimension of the problem, although, in Chapter 11, Casey Luskin introduces some of the main controversies. Instead, this chapter analyzes how common descent hypotheses are constructed and how they are tested, setting these matters in the wider context of the origin of life and the possibility of intelligent design. 5 key questions capture the most important issues arising from UCD and CD. Think of these questions as a toolkit for evaluating any hypothesis of common ancestry. Before we consider the 5 questions, however, we should clear away some potential misunderstandings about the relationship between the concepts of evolution, common descent, materialism, or naturalism, and intelligent design. The terms evolution and common descent do not name the same idea. When Darwin built his case for UCD in The Origin of Species, 1859, evolution, meaning theories about the origin by natural processes of new groups of organisms, had already been in the air for many decades. Darwin's own grandfather Erasmus, 1731 to 1802, speculated about the idea, and during the Beagle Voyage, 1831 to 1836, Darwin had absorbed Charles Lyell's critique of French zoologist Jean-Baptiste de Lamarck's, 1744 to 1829, theory of evolution. Thus, refuting UCD does not necessarily refute evolution, because the latter idea allows for much more than common descent or shared ancestry. The philosophies of materialism and naturalism do not entail UCD. For that reason, falsifying UCD would not necessarily threaten those worldviews. Lamarck, for example, was a materialist with respect to biology, but he described evolution as occurring in multiple independent lineages, each spontaneously generated. Such a process yields separate trees of life, not a single tree, see Figure 12.1. The German materialist and embryologist Ernst Haeckel, 1834 to 1919, also thought spontaneous generation occurred throughout the history of life. Hence, the single-celled organisms alive today did not descend from ancient cells, but originated recently, arising directly from non-living materials. Again, such an evolutionary scenario will generate multiple independent trees. Lastly, nearly all evolutionists who currently doubt UCD accept naturalism or materialism as their philosophy of science. Thus, falsifying UCD would not by itself threaten those philosophies. Common descent may be false at one level of taxonomic inclusiveness, yet true at another level. If that statement makes you scratch your head, look at figures 12.2a and b. Notice that, in the hypothetical branching patterns of Figure 12.2a, the animals share common ancestry, CD, but UCD is false, multiple independent trees of life exist. In Figure 12.2b, by contrast, the animals don't share a common animal ancestor, but UCD is true, all organisms stem from Luca. 4. This and other reasons, it is critical not to use the terms common descent or common ancestry without specifying which groups are included in the hypotheses of descent or ancestry. Every hypothesis of common descent needs to be weighed on its own merits, with respect to the evidence at hand. Common descent and intelligent design are not mutually exclusive. An ID colleague of mine, biochemist Michael Behe, argues vigorously for design while also accepting UCD. So did the Harvard botanist Asa Gray, 1810 to 1887, a Christian who proposed God purposefully caused variations to occur, a design hypothesis which Darwin rejected despite Gray's urging. Neither UCD nor CD restricts what one may infer about the causes of evolutionary change, meaning that, without additional philosophical assumptions, intelligent design remains a live possibility even if UCD or CD turned out to be the case. Table 12.1 shows the historical diversity of opinion on UCD in relation to intelligent design. Notice 4 positions are possible. Every quadrant in the matrix is occupied by prominent biologists of the past or present. Given these points, and the range of positions on offer, the reader may be wondering, well, if UCD is endorsed by some intelligent design theorists, and if UCD and materialism are independent of each other, why bother challenging UCD, or CD, at all? Wouldn't the wiser course be to withhold judgment about the theory, at least provisionally, since most biologists still accept UCD? Perhaps, but we think the wisest course is to pursue the truth. Here is the only question about UCD ultimately worth asking: is the theory true? If UCD is true, then Christians, indeed, everyone, will have to make their peace with it. If UCD isn't true, however, then no one, Christians included, should hang on to the theory. To begin, we should examine how evolutionary biologists normally infer that 2 or more species share a common ancestor. This will provide the terms and concepts we shall use throughout the remainder of the chapter. The anatomy of inferences to common ancestry. Richard Dawkins says many things that are beautifully clear, wrong, maybe, but nonetheless clear and easy to follow. In The Blind Watchmaker, Dawkins explains why he thinks all organisms share a common ancestor. It is a fact of great significance that every living thing, no matter how different from others in external appearance it may be, speaks almost exactly the same language at the level of the genes. The genetic code is universal. I regard this as near-conclusive proof that all organisms are descended from a single common ancestor. The odds of the same dictionary of arbitrary meanings arising twice are almost unimaginably small. 0.7. We can use this excerpt from Dawkins as a model for showing how common descent hypotheses are constructed. Every necessary element of any CD hypothesis is present and working within his argument. Dawkins is making a case for UCD. Indeed, the universal genetic code argument for UCD is one of the most widespread in biology, occurring not only in popular books like The Blind Watchmaker but in textbooks and the technical literature as well. Chances are, you have seen this argument before. The terms highlighted with bold italics below will be the concepts we shall employ elsewhere. Figure 12.3 depicts Dawkins's argument. Boxes P and Q could be any two species or taxa, singular taxon, meaning biological group. Let's say taxon P is the domesticated dog, Canis familiaris, and taxon Q is portobello mushrooms, Agaricus bisporus. P and Q both exhibit biological characters, in this instance, the genetic code. The genetic code comprises the 64 trinucleotide translation specifications by which information passes from nucleic acids, such as DNA and RNA, to amino acids, as proteins are assembled. Biological characters include any observable features of organisms, ranging from their molecular details, such as the genetic code, up to complex behaviors, such as nest building or speech. Dawkins asks us to decide which scenario is more likely. Did the character, genetic code, arise once, in the unknown common ancestor for P and Q, figure 12.3a, or twice independently, as depicted in figure 12.3b? Now the single arrow in figure 12.3a, leading to box CA, the unknown common ancestor where the code first appears, and the two separate arrows leading to P and Q in 12.3b, become important. In fact, these arrows, which we can call the origination pathways and probabilities, carry the weight of Dawkins's argument. Origination probabilities are estimated from the evolutionary processes and mechanisms that cause new biological characters to appear. The odds of the same dictionary of arbitrary meanings arising twice, argues Dawkins, are almost unimaginably small. Here is why. Let us suppose the origination probability for the first appearance of the genetic code occurring even once is far less than 1.0, and Dawkins estimates the probability, p, to be vanishingly small, much closer to 0.0 than to 1.0, because the code appears to him to be a chemically arbitrary, frozen accident. If so, then invoking two such frozen accidents, occurring independently of each other, as in 12.3b, the separate origin scenario, yet with those accidents somehow landing on the same apparently arbitrary molecular meanings, will be the product, p2, because the independent probabilities in 12.3b multiply by each other. Originating the genetic code even once was hard enough. But twice? The same code? Thus, the separate origin scenario, 12.3b, will be much more improbable than the common descent scenario, 12.3a, that is, it will be exactly the square, p2, of the origination probability, p, in 12.3a, if we consider only the character of the genetic code, and use Dawkins's assumption of a vanishingly small origination probability. Given the genetic code they share as an essential molecular character, therefore, dogs and portobello mushrooms must have stemmed from a common ancestor. The mushrooms won't mind, but don't tell your dog. At this point, Dawkins's argument should make sense to the reader. We catch plagiarists by using the same probabilistic reasoning. If boxes P and Q in figure 12.3b, for example, represented two texts supposedly written independently of each other, yet they contained whole paragraphs that were word for word identical, a scenario of separate origins would strike us as utterly unlikely. Common descent is a theory of transformation, not similarity. Dawkins's argument for UCD may seem conclusive. But how could we test it? After all, UCD isn't supposed to be a deduction, or an axiom, but an empirical hypothesis, potentially vulnerable to contrary evidence. The answer starts by thinking carefully about what any common descent hypothesis asserts, when compared to its logical opposite, separate origins. Notice the arrows leading from the common ancestor Ca to taxa P and Q. These are the evolutionary transformation pathways and probabilities, and we need to know them, too, before we can decide if common descent or separate origins is more likely. We need to know these pathways and the underlying causal processes operating during the branching from Ca, because if their associated probabilities turn out to be too small, or actually zero, common ancestry loses to separate origins. And that is because common descent is not mainly a theory of similarity. This is not the usual view, which takes the similarities that organisms exhibit to be both the principal evidence for UCD and what the theory explains best. Notice, however, that similarity, i.e., pattern matching, exists in both the common descent and separate origin scenarios. How we explain similarity under separate origins, we consider below. Rather, common descent is mainly a theory of transformation. The theory claims it is biologically possible to transform species, starting with a common ancestral form and ending with very different descendant taxa. Taxa P and Q have different names because they exhibit different characters. If CD is true, those characters must arise as novel transformations on the branches from the common ancestor. CA, common descent must explain the origin of differences, novel characters, and not simply similarities, along natural pathways or branching lineages starting with an ancestral form which did not possess those characters. Logically, CA cannot exhibit the same characters that define its descendants, because if the same novel features distinguishing taxon P, for instance, also exist in CA, then the ancestral and descendant species would necessarily belong to the same taxon. This claim, namely, that species can be, and were, viably transformed over time fundamentally distinguishes both UCD and CD from separate origins. Similarity occurs in both scenarios. Transformation, however, does not. Therefore, finding and testing mechanisms and processes of species transformation represents the main explanatory challenge facing UCD and CD, as Darwin understood in 1859. Darwin also understood that the challenge of explaining species transformations provided a way to test UCD and CD, which brings us to our first critical question. Question 1. If species were not connected by common descent, how would we know it? Many of the answers to this question in the biological literature may strike one as facetious. The author appears not to take the question seriously. After all, common descent is a fact, and who worries about testing facts, and therefore offers what seems to be a flippant reply. Consider, for instance, evolutionary biologist John Maynard Smith's proposed test for neo-Darwinian evolution. If someone discovers a deep-sea fish with varying numbers of luminous dots on its tail, the number at any one time having the property of a prime number, I should regard this as rather strong evidence against neo-Darwinism. And if the dots took up in turn the exact configuration of the heavenly constellations, I should regard it as an adequate disproof. The apparent flippancy of this answer, however, conceals a deeper truth. If we ask why the hypothetical character of constellations on deep-sea fish would disprove common descent, we uncover a bona fide test first proposed by Darwin, and widely used by evolutionary biologists today. UCD and CD claim species can be dramatically transformed over time, and, in the process, can give rise to the entirely novel characters which define new taxa. As Darwin himself realized, however, if those transformations cannot occur, UCD and CD would absolutely break down. If it could be demonstrated that any complex organ existed, which could not possibly have been formed by numerous, successive, slight modifications, my theory would absolutely break down. This test, proposed by Darwin in The Origin of Species, has come to be known as the principle of continuity. The principle of continuity, hereafter, continuity, asserts that every point in any hypothesized pathway of evolutionary transformation must be biologically possible. This may seem to be a truism. Since Darwin, however, continuity has been widely employed by biologists to test hypothesized evolutionary pathways. In 1968, for example, Francis Crick used continuity to rule out certain hypotheses about the evolution of the genetic code. Those hypotheses proposed that the size of codons, triplets of DNA nucleotides specifying amino acids in protein assembly, had changed from 1 to 2 to 3 nucleotides in length. This seems highly unlikely, argued Crick, since it violates the principle of continuity. A change in codon size necessarily makes nonsense of all previous messages and would almost certainly be lethal. Figure 12.4 illustrates Crick's argument. The hypothesized pathway from R with 1 or 2 nucleotide length codons to S with 3 nucleotide codons would necessarily pass through an inviable transitional phase, Crick argued, because the amino acid specifying information carried by the shorter codons would be disrupted if their size increased, a state incompatible with protein assembly and hence cell function. Here is a natural language analogy, here is an natural language analogy. Continuity demands reproductive capability because being able to reproduce is an absolutely necessary, i.e., essential, condition for the organisms in any evolutionary lineage. Just as there is no crying in baseball, to quote Tom Hanks's character, manager Jimmy Duggan, from the movie A League of Their Own, there is, quite literally, no failing to leave offspring in common descent. If a hypothesized transformation pathway is biologically impossible, whether because of the lethality it would entail, or because of any lesser functional obstacle preventing reproduction, it could not and therefore did not happen. Moreover, continuity also demands no jumps and no foresight. In addition to no crying in baseball, there is also no hitting the ball and running straight to third base. A living system cannot leap to a distant novel functional or structural state in a single try, or in any series of increments larger than what undirected evolutionary processes will allow. Evolutionary processes cannot jump to distant destinations, i.e., to novel complex characters, because undirected processes, by definition, do not know where they are going. Moreover, the multiple coordinated changes required to specify novel functional characters map to very small probabilistic targets, lying within vastly larger non-functional spaces, where undirected processes are far likelier to land and wander irreversibly. For any long journey from A to B to C, and so on, it helps to know where you are supposed to be going. Darwin understood the no jumps demand of continuity, because he put it front and center in the opening of the origin. Someone might say, he noted, that after a certain unknown number of generations, some bird had given birth to a woodpecker, but this assumption seems to me to be no explanation, for it leaves the case of the co-adaptations of organic beings to each other and to their physical conditions of life, untouched and unexplained. It is, therefore, of the highest importance to gain a clear insight into the means of modification and co-adaptation. We need to know the details of the pathways of transformation, or what Darwin calls the means of modification, in order to decide if an evolutionary pathway is possible or not. Continuity therefore obtains wherever organisms exist in lineages, and, accordingly, is deployed as an indispensable tool of critical analysis in every corner of evolutionary theory. As explained by Yuri Wolf and Eugene Koonin, continuity applies when the origin of any novel complex system is hypothesized to have occurred. When an evolutionary biologist strives to explain the origin of a truly novel system that is seen only in its elaborately complex state and, at face value, appears to be irreducibly complex, the task is much harder. Because evolution has no foresight, no system can evolve in anticipation of becoming useful once the requisite level of complexity is attained. Darwin did not use a specific term for this crucial tenet of evolutionary biology. We will call it the continuity principle. Evolution of complex systems still needs to be deconstructed into successive steps and explained in a Darwinian way. Anyone who studies evolutionary theory closely will see the sharp blade of continuity, whether explicitly by name or implicitly, slicing through origination and transformation hypotheses everywhere. Evolutionary biologist David Penny, for instance, uses continuity to dismiss origination hypotheses for the ribosome invoking its current function, because those hypotheses violate continuity's no foresight rule. The ribosome is a huge macromolecular complex and there are many steps leading up to the synthesis of the peptide bond that joins amino acids. There is no way that a ribosome could have evolved de novo in a single step and meet our guidelines of continuity. It is impossible on an incremental model that a very complex structure could evolve for something that does not yet exist. Here is an easy way to remember the significance of continuity for testing UCD and CD. Continuity and common descent always travel together, everywhere. If a transformation hypothesis holds that taxa P and Q stemmed from a common ancestor by an evolutionary pathway, see Figure 12.5, then, because continuity is logically entailed whenever any such pathway is proposed, the rule must be satisfied over the whole length of that pathway. That brings us to the second key question. Question 2. What were the actual transformation pathways, satisfying the continuity rule, which connect all organisms to Luca? For many leading evolutionary biologists, answering this question leads to a negative outcome for UCD. For them, continuity renders Luca itself impossible, and thus, the associated transformation pathways to the three major domains were never traversed by species. In other words, UCD is false. In 2002, in an article titled On the Evolution of Cells, Carl Woese explained, Why he rejected the historical existence of Luca, the last universal common ancestor, as an actual, identifiably unique cell, the progenitor of all organisms alive today. Woese argued that the characters making up the cellular architectures found in the three major domains of life, the bacteria, archaea, and eukarya, architectures which Woese himself did much to discover, are too different to have evolved by descent from a single common ancestor. The transformation pathways from Luca are therefore too improbable, or, to put the same point another way, the dramatic transformations required make Luca biologically impossible. Take the essential processes of DNA replication, for instance, characters so basic they belong to the core set of vital functions within any cell. Virtually no homology, similarity due to material descent, Woese wrote, exists between the bacterial genome replication mechanism and that basically common to the archaea and the eukaryotes. Modern genome replication mechanisms seem to have evolved twice. In other words, if Luca existed, the similarity in these essential cellular characters which one would have expected to see, given the inescapable reproductive requirements of continuity, is not observed. The same absence of fundamental similarity, Woese observes, exists in other basic cellular characters, to such a degree that what needs explaining is not why the major cell designs are so similar, but why they are so different. This apparent contradiction can be resolved by assuming that the highly diverse cell designs that exist today are the result of a common evolution in which each of them began under, significantly, different starting conditions. Continuity breaks apart the primary evolutionary lineages putatively stemming from Luca, which, as a logical consequence, must itself vanish as a real organism. Because Luca necessarily associates physiologies that have not been observed together in any modern lineage and asks that all of this come about through vertical inheritance, Woese contends, we are left with no consistent and satisfactory picture of the universal ancestor. It is time to question underlying assumptions. And what is the result of that questioning? The universal ancestor, says Woese, is not an entity, not a thing. When Luca disappears as a real cell, therefore, we no longer expect to find a single tree of life, but a forest or a network, containing at least three separate, i.e., independent, cellular starting points. Extant life on Earth, Woese concludes, is descended not from one, but from three distinctly different cell types. Molecular evolutionist W. Ford Doolittle of Dalhousie University rejects Luca on logically identical grounds. Continuity cannot be satisfied. As in Woese's case, he focuses on the biological impossibilities raised by the transformation pathways needed to derive the enormous genetic diversity of life from a single ancestral cell. Calling Luca a profoundly problematic entity, Doolittle argues there could have been no single common ancestral cell whose genome harbored a direct ancestor, either the last common ancestor or their lineal predecessors, of all the genes present in all genomes today. As he explains, the rapidly increasing number of different genes and proteins, i.e., characters, that, if Luca existed, must trace their ancestry via transformation pathways from that single organism, entail that Luca instantly becomes a biologically unrealistic supercell. In the end, UCD requires a last universal common ancestor with an enormous range of metabolic capabilities, essentially any gene that is now seen in at least one contemporary archaean, and one contemporary bacterium must find a home in that ancient cell. Such a genome of Eden hypothesis seems, to us, unappealing. If Luca did not exist, concludes Doolittle, then Darwin arguably was wrong. For most of life and most of its history, descent with modification is not the simple branching process he envisioned. Woese and Doolittle are not alone in rejecting the existence of Luca and finding UCD false. University of California geneticist Michael Sivanen argues that there is no reason to even postulate that a Luca ever existed, because the modern cell could have evolved in multiple parallel lineages. Earliest life could have been truly polyphyletic, i.e., arising in several independent lineages. French microbiologist Didier Raoult states flatly that there is no such thing as a tree of life. The idea of a tree of life, which stringently follows Darwin's theory, is not pertinent in the genomic age. National Center for Biotechnology Information Lab Director Eugene Koonin likewise waves Luca and the tree of life aside. A single, uninterrupted TOL does not exist, he contends. For this growing cadre of evolutionary biologists, Luca has disappeared under the scrutiny of continuity, and the base of the tree of life has splintered into pieces. Continuity applies as a rule, of course, not just at the base of the tree of life, but upwards into its branches, such as at the origin of the animals. Evolutionary biologist and embryologist T.J. Horder explains how continuity applies to animal evolution, where the transformations in question necessitate the modification of developmental sequences. When considering such pathways, he writes, there exists an essential requirement which should be met by any hypothetical evolutionary sequence. A continuous sequence of morphogenetic events in an embryo is a repetition of a continuous sequence of morphological steps built up through the preceding evolving series of embryos, each stage of which must have been functionally advantageous in the transitional organism. This will be referred to as the continuity principle. Now, however, we run into a major complication, and the inescapable larger context mentioned in my introduction to this chapter. The law of biogenesis, the origin of life, and UCD as axiom. Thus far, we have been arguing that UCD and CD are tested by continuity, using modus tollens. If common descent is proposed for some taxa, via a transformation pathway, then continuity must be satisfied along the entire length of that pathway, but continuity has not been satisfied, therefore, that pathway did not occur, and common descent, in this instance at least, is false. But common descent and continuity are not the only relevant considerations before us. As soon as we climb up into the tree of life, away from its earliest, simplest beginnings in a prebiotic milieu, we immediately collide with another of the genuinely universal propositions in biology, namely, the law of biogenesis, all life comes from life. P. B. Medawar and J. S. Medawar call this proposition arguably the most fundamental in biology. In its affirmative form, the law of biogenesis states that all living organisms are the progeny of living organisms that went before them. The familiar Latin tag is omne vivum ex vivo, all that is alive came from something living. In other words, every organism has an unbroken genealogical pedigree extending back to the first living things. In its negative form, the law can be taken to deny the occurrence, or even the possibility, of spontaneous generation. The odd thing about the law of biogenesis is that no evolutionary biologist knows exactly when during Earth's history the law began to hold universally, as it surely does today, but before which the law could not have been the case. Under any naturalistic hypothesis for the origin of life, the law of biogenesis must be violated, spectacularly so, in fact, to get single-celled organisms up and running from non-living starting materials. Once upon a time, there was no life on this planet, today there is. At some point in the past, therefore, the law of biogenesis could not have been true. Nonetheless, evolutionary biologists today, unlike their 19th-century predecessors such as Lamarck and Haeckel, would say that after life began in its simplest possible form, the possibility of abiogenesis ceased, irreversibly, and from that ill-defined interval to the present, any organism we observe must have descended from at least one parent that was itself an organism. See Figure 12.6. It requires little imagination to see how the law of biogenesis, when coupled with assumptions about the uniqueness of the origin of life on Earth, could yield UCD as a deduction, moreover, that renders continuity powerless to test the theory. It simply wouldn't matter if we knew the transformation pathways from Luca or not, UCD must be true. This explains, incidentally, why pervasive ignorance about the transformation pathways throughout the tree of life rarely counts against UCD. With a single occurrence of abiogenesis, there is literally nowhere else for organismal lineages to go, except back to Luca. This may also explain why, paradoxically, given their philosophical materialism, so many neo-Darwinian theorists are eager to assign a vanishingly small probability to the origin of life on Earth, so small in fact that the event could have occurred only once within the lifetime of the solar system. Indeed, if we buy the premise of a single origin of life, UCD achieves the status of a historical fact. As evolutionary biologist Keith Thompson explains, UCD can be derived from the twin premises that life arose only once on Earth and that all life proceeds from existing life. Given these premises, and if the pattern of diversity among organisms has changed over time, then it must follow that any new forms have appeared as the result of a process of descent from pre-existing forms. Descent from common ancestors is based upon such unassailable logic that we act as though it is a fact. Deductive or axiomatic formulations of UCD are surprisingly widespread in the biological literature. Evolutionary biologist Kenneth Weiss, for instance, notes that the assumption of a point source, that is, an abiogenesis singularity, from which all life derives, is important to the entire theory, i.e., UCD, so we insist upon spontaneous generation in the beginning, but must forbid it at any other time. Postulate what one needs, namely, a first cell, climb up from the prebiotic melia to that cell, once, and only once, and then kick away the ladder. Which brings us to our third key question. Question 3. Have we genuinely tested UCD, or merely assumed its truth? For evolutionary biologists, assuming a single origin of life is a classic good news bad news move. The good news is that UCD follows as a deductive consequence, all organisms must be related in one great tree of life. The status of UCD as historical fact is assured. The bad news turns out to be exactly the same. By postulating the singularity of a unique, one-off, event of abiogenesis in Earth history, UCD follows as a deductive consequence, but now UCD becomes untestable. No biological evidence, no matter how compelling, could possibly challenge the theory. Assume a single origin of life event, and because of the law of biogenesis, Luca must have existed, with countless pathways of transformation connecting that primordial cell to all other life, whatever we actually observe. Every organism, every species on this planet will necessarily lie within what might be called the Luca horizon. Axiomatic formulations of UCD deductively channel all evolutionary inferences into a theoretical funnel running back to Luca, outside of which no organism ever existed. Suppose one thinks, for example, that evidence indicates some animal group, say, the arthropods, did not share a common ancestor. Evolutionary biologist Jeffrey Fryer cites continuity as his reason for doubting the common ancestry of that group. Those interested in tracing the course of evolution, he writes, are concerned with mechanisms and how one mechanism or complex organism might have been transformed into another, maintaining functional continuity throughout the transformation. Under the scrutiny of continuity, he argues, it is difficult to envisage a common ancestor even of crustaceans and tracheids. No worries, these lineages will still find their branching points and common ancestors somewhere within the tree of life. Under axiomatic UCD, all discontinuities or disparities in form and function among species can only be apparent, not true or aboriginal differences. The late Harvard paleontologist Stephen Jay Gould, who contended vigorously for the sudden origin of the remarkable disparities of animal form in events such as the Cambrian explosion, allayed the anxieties of his evolutionary critics by affirming the all-enveloping embrace of axiomatic UCD. Did any proponent of increased disparity ever doubt that a cladogram, branching diagram of common descent, would root, if not in the arthropoda at least at a more inclusive level? We are not, after all, creationists, and we do accept a monophyletic origin for life. Axiomatic UCD robs biological evidence of its power to test the theory. Now, if one wants UCD to be the case, come what may, testing against evidence will only be a nuisance, but few biologists in my experience are willing consciously to go there. They know that, throughout the history of science, a widely accepted but erroneous theory may become so deeply embedded within a community of thought that its influence on scientific perception becomes all but invisible. When that occurs, writes philosopher of biology Elliot Sober, the theory guides scientific inquiry without being vulnerable to the testimony of nature. So, should UCD be held axiomatically? The reader can see for herself how that happens, and can decide if she wants to place UCD beyond ordinary testing, or to keep the theory vulnerable to the testimony of nature. Gould touched on another aspect of the larger context surrounding UCD. We are not, after all, creationists, he wrote. But why should the issue of creationism have arisen in a technical discussion of arthropod evolution? Time to turn to our fourth key question. Question 4. When explaining the history of life, have we assumed methodological naturalism only, or have we allowed for the possibility of intelligent design? Gould brought up creationism because, in addition to his paleontological and evolutionary theory labors, he worked as a historian of biology. Gould knew well the reasoning of the biologists and natural historians in the yes-to-design and no to UCD quadrant, which occupies the lower right corner of Table 12.1, above. Louis Agassiz, for instance, whose name graces Harvard's Agassiz Museum of Comparative Zoology, where Gould kept his own office, resisted UCD for evidential reasons, but also because Agassiz's scientific worldview included the possibility of intelligent design. Gould understood from his study of Agassiz's work, not to mention the writings of other leading pre-Darwinian biologists, that for them, aboriginal or primary discontinuity, i.e., separate origins, not UCD, was the main signal emerging from the fossil record, from comparative anatomy, and from the functional complexities of organisms. Once the possibility of intelligent design has entered the causal mix, of course, the range of explanations possible for any phenomenon grows accordingly. Nowhere is this illustrated more vividly than when we consider the cause of similarities, overwhelmingly the principal line of evidence for UCD. All living things have much in common, wrote Darwin in the Summation of the Origin. Therefore I should infer from analogy that probably all the organic beings which have ever lived on this earth have descended from some one primordial form, i.e., Luca, the root of UCD. When living things are similar, they must be related by common descent, right? Propinquity of descent, argued Darwin elsewhere in the Origin, is the only known cause of the similarity of organic beings. Now, not only is this claim false, indeed, Darwin himself knew it was false, but the whole record of comparative and historical biology since 1859 testifies to the incompetence of similarities to sort misleading resemblances from genuine family relationships. Similarity does not entail common descent, which is only one of its many possible causes. Parentage, or begetting, i.e., genuine descent, is not a similarity relation. The reader does not need to know the history of evolutionary theory, or comparative biology, or to be a biologist, to see that similarity may have multiple causes other than material descent. Consider a suggestive thought experiment. Let's go back to the plagiarism example, sketched above. Figure 12.7a shows two texts, written separately by two college students enrolled in a philosophy course, but each containing an entire paragraph, say, of 100 words length, word for word identical when compared across the two documents. Plagiarism? Maybe. But suppose we have 30 such texts, written by 30 different students, and each contains the same 100-word paragraph in question, see Figure 12.7b. In fact, the philosophy class has 30 students enrolled, and they all submitted their essays on the same day. Figure 12.7. Two essays, supposedly written independently of each other. Interestingly, the likelihood of plagiarism goes down, dramatically, as the number of texts including the same paragraph goes up, because as our sample grows, and we learn more, the probability increases that the cause of the paragraph matching was the professor's essay assignment. Cite this paragraph from Descartes' Discourse on Method in your paper and analyze its argument. Raw similarity, in other words, underdetermines its true cause. To identify the true cause, we need to know the full range of possible causes, and ruling out any of them a priori can only hinder our search. Consider an even simpler thought experiment, which requires your participation. This chapter was formatted to leave some white space, and the 5 blank lines below. 1, 2, 3, 4, 5. Sign your name as you normally would on a legal form or bank check, 5 times. If you experience moral qualms about writing in books, as I do, use a pencil so you can erase it later, or use a separate sheet of paper. Do you observe any similarity in the 5 signatures? These patterns share no material descent. What they do share is you, a unique intelligent agent, as their true cause. If we approach the phenomena of similarity in biology with the possibility of intelligent design in our explanatory toolkit, the gravitational pull of common descent as the only reasonable explanation is greatly weakened, as it should be. Hence, our final key question. Question 5. In the light of intelligent design as a causal possibility, what histories for life on Earth might be the case? Answering that question opens before us a beautiful vista of fascinating research possibilities. The next step requires you and your intellectual freedom. Get started. 13. The Battle Over Human Origins, Introduction to Chapters 14 to 16. And K. Gauger. Summary. The origin of humanity, where we came from, is an issue with many ramifications. It impacts our self-understanding in multiple ways. Did we evolve from a common ancestor shared with chimps, or might we have a unique origin? Mainstream science says that it is incontrovertible that we are the product of evolution. Is it therefore necessary to adapt our understanding of Scripture, or might the science be overstated? This short chapter, along with chapters 14 to 16, will argue that the question of our origin is far from settled, and that there are scientific arguments to be made in favor of a unique origin for humanity. There is no need to change traditional scriptural interpretations based on inconclusive science. The intersection of evolutionary theory with Christian faith, in particular on the topic of human origins, leads immediately to one question: are we descended from two first parents or not? This question raises a multitude of difficulties, both theologically and scientifically. Not surprisingly, the Christian Church is divided on this issue, with some emphasizing the scientific arguments and some the biblical. The standard scientific worldview accepts neo-Darwinism as the explanation for our origin. More specifically concerning human origins, the neo-Darwinian view is that we have a purely natural origin, fully explicable by unguided natural selection acting on random variation. According to this view, the fossil record shows that we evolved from an ape-like ancestor, we share common ancestry with chimps, and we evolved from a population of several thousand, at a minimum. Theistic evolutionists operate from the perspective that this received view is accurate and trustworthy, in fact, more likely to be understood correctly than thousands of years of theological tradition. In their view, the scientific evidence against the possibility of a historical Adam and Eve is overwhelming. Our common descent from ape-like ancestors is obvious. Thus the first chapters of Genesis are viewed as non-historical. In this section of the book we are focusing on the scientific evidence relating to human origins. Nevertheless, we acknowledge that the debate over human origins is so consequential because it has implications that go beyond science. The difficulty for many Christians, as is pointed out in Wayne Grudem's Biblical and Theological Introduction, is that discarding traditional readings of Genesis seems to vitiate the basis for essential doctrines of the Christian faith. Many of the theistic evolutionists who advocate for acceptance of the neo-Darwinian story themselves claim that the scientific evidence necessitates the loss of a historical first human couple, which in turn removes the idea of original sin and the fall, among other things. It is important to stress that the Darwinian story in its unadulterated form is purely materialistic. It says that we are supposed to have evolved through an unguided process into the modern humanity we see today. The appearance of intelligence, language, and even morality and spirituality are attributed to undirected and incremental natural selection, either that or some highly improbable coincidence, of which we are the lucky recipients. There is no story of the infusion of a soul at any point, because in the materialistic worldview there are no souls. Intelligence, language, morality, and spirituality are all the product of happenstance and the gradual selection of more fit individuals. They derive not from any higher power but from the evolutionary process, which leads to increasingly complex social development. Darwin wrote quite clearly on this point: the idea of a universal and beneficent creator does not seem to arise in the mind of man until he has been elevated by long-continued culture. In short, man is not made in God's image, God is made in man's image. It follows that our moral sense, as Darwin called it, is the product of our social instincts. Even consciousness disappears in the naturalistic mindset, which is ironic. Indeed, to paste the idea of a benevolent creator onto this Darwinian story is like slapping a band-aid on a gaping wound. How is one to stanch the bleeding? If the new Darwinian understanding of human origins has consequences this drastic, it is imperative that it be carefully examined for its rigor and veracity. Is it possible that, in the effort to accept the prevailing neo-Darwinian view, theistic evolutionists have lost sight of the truth? Might the story actually be wrong? Scientific consensus has been wrong many times: phlogiston, humoral pathology, luminiferous ether, astrology, globalism, spontaneous generation, eugenics. The list could go on, and it would be a shame, surely, to discard our Christian heritage for a mess of pottage. Given our society's tendency to put science in the place of authority, we may be tempted to accept what we are told as proven established science. But we should be quite cautious about accepting any scientific theory with effects as far-reaching as neo-Darwinism has. And there is good reason for doubt about the theory. Neo-Darwinism fails to explain many significant things in biology, as should be apparent from the previous chapters. It cannot account for the origin of information, the appearance of complex body plans in the Cambrian, or how an embryo develops into an adult. Even the idea of common descent, an idea that is essential for the neo-Darwinian story, is subject to challenge. Alternate theories such as those offered by the extended evolutionary synthesis also have problems. So just at a time when neo-Darwinism seems to be collapsing, why should Christians accept the story of our evolutionary origin from apes? Should we not investigate the merits of the case? Most especially, should we not examine the evidence that is offered in support of the theory of human evolution? The argument. Because of the settled science of today, theistic evolutionists, or evolutionary creationists, accept the idea of our common ancestry with chimps. To review, 3 dubious claims have been made concerning this. First, the fossil evidence reveals intermediates between us and our ape-like ancestors, apparently bridging the gap between us and them. Second, the fact that our DNA is nearly identical to chimp DNA is taken as evidence of our common ancestry. Third, population genetics says that the first steps on the way to humanity arose in a population of about 10,000, and that no fewer than several thousand ancestors could have ever existed in the human lineage. Now, are these claims true? How solid is the science behind them? That is what we examine in the next 3 chapters, 14 to 16. These chapters will make the following key points. The actual evidence in the fossil record is starkly at odds with the narrative of human evolution. Realistic candidates for species transitional to humans are conspicuous by their absence. The alleged similarity between humans and chimpanzees at the genetic level is exaggerated. As our understanding of the complexity of the genome increases, so does the realization of the differences between humans and chimps. In particular, arguments relating to junk DNA have been vitiated by recent discoveries concerning the important, and often species-specific, functions of these elements of the genome. The time needed for humans to have evolved from an ancestor in common with chimps is orders of magnitude greater than the time available in conventional evolutionary models. Conventional population genetics models assume common descent of humans, that chimps and humans share a common ancestor. We propose an alternative model, one based on a unique origin of an original pair of humans with initial created diversity, giving 4 versions of each initial non-sex chromosome. This is consistent with the evidence of a block structure for a large percentage of human DNA, and is open to verification by means of computer simulation. For all these reasons, the hypothesis of our shared ancestry with chimps, a hypothesis most contentious among Christians and vital to the theistic evolutionary position, is deeply flawed, and is a very unsound basis for revising the very foundations of Christian doctrine. 14. Missing transitions, human origins and the fossil record. Casey Luskin. Summary. The standard evolutionary view of human origins, generally accepted by theistic evolutionists, holds that our species, Homo sapiens, evolved from ape-like species through apparently unguided evolutionary processes like natural selection and random mutation. Theistic evolutionists and other evolutionary scientists often claim the fossil evidence for this Darwinian evolution of humans from ape-like creatures is incontrovertible. But their viewpoint is not supported by the fossil evidence. Hominin fossils generally fall into one of 2 groups, ape-like species and human-like species, with a large, unbridged gap between them. Virtually the entire hominin fossil record is marked by fragmented fossils, especially the early hominins, which do not document precursors to humans. Around 3 to 4 million years ago, the Australopithecines appear, but they were generally ape-like and also appear in an abrupt manner. When our genus Homo appears, it also does so in an abrupt fashion, without clear evidence of a transition from previous ape-like hominins. Major members of Homo are very similar to modern humans, and their differences amount to small-scale microevolutionary changes. The archaeological record shows an explosion of human creativity about 30 to 40,000 years ago. Despite the claims of evolutionary paleoanthropologists and the media hype surrounding many hominin fossils, the fragmented hominin fossil record does not document the evolution of humans from ape-like precursors, and the appearance of humans in the fossil record is anything but a gradual Darwinian evolutionary process. Theistic evolutionists should appreciate that. Christians who doubt standard evolutionary accounts of human origins hold legitimate views that are backed by scientific evidence. Evolutionists commonly tell the public that the fossil evidence for the Darwinian evolution of our species, Homo sapiens, from ape-like creatures is incontrovertible. Theistic evolutionists are no exception. In 2009, Southern Methodist University anthropology professor Ronald Wetherington testified before the Texas State Board of Education that human evolution has arguably the most complete sequence of fossil succession of any mammal in the world. No gaps. No lack of transitional fossils. So when people talk about the lack of transitional fossils or gaps in the fossil record, it absolutely is not true. And it is not true specifically for our own species. According to Wetherington, human origins shows a nice clean example of what Darwin thought was a gradualistic evolutionary change. Theistic evolutionists like Wetherington who teach at Christian universities are often adamant about the evidence for human evolution, sometimes more so than their counterparts at secular schools. But does the fossil record support their claims? Digging into the technical literature reveals a story starkly different from the one presented by Wetherington and other evolutionists. Far from supplying a nice clean example of gradualistic evolutionary change that has no gaps or no lack of transitional fossils, the record shows a dramatic discontinuity between ape-like and human-like forms. Human-like fossils appear abruptly in the record, without clear evolutionary precursors, contradicting Darwinian expectations. The fossil record does not show that humans evolved from ape-like precursors. The fragmented field of paleoanthropology. The discipline of paleoanthropology studies the fossil remains of ancient hominins and hominids. Hominins and hominids may be defined differently, depending on which expert you consult, but this chapter will define hominins as humans and all of our extinct, supposed, ancestors and relatives tracing back to our, again, supposed, most recent common ancestor with chimpanzees. The term hominid is often used in roughly the same manner, although officially it means any member of family Hominidae, including humans, great apes, chimpanzees, gorillas, and orangutans, and any extinct species descended from their proposed most recent common ancestor. Thus, a hominin is a hominid, but a hominid is not necessarily a hominin. Paleoanthropologists face many daunting challenges in their quest to explain human evolution from this hypothetical human-ape common ancestor. Their field is fragmented in multiple senses, making it difficult to bolster evolutionary accounts of human origins. First, the fossil record is fragmented, and long periods of time exist for which there are few hominin fossils. National Geographic acknowledges, fossils attributed to Homo in the period 2 to 3 million years ago are exceedingly rare, and you could put them all into a small shoe box and still have room for a good pair of shoes. So, fragmentary and disconnected is the data, according to Harvard zoologist Richard Lewontin, that despite the excited and optimistic claims that have been made by some paleontologists, no fossil hominid species can be established as our direct ancestor. A second challenge is the fragmented nature of the fossil specimens themselves. Typical hominid fossils consist of mere bone scraps, making it difficult to form definitive conclusions about their morphology, behavior, and relationships. Primatologist Frans de Waal observes that the skeleton of the common chimpanzee is nearly identical to the bonobo, but they have great differences in behavior. On the sole basis of a few bones and skulls, writes de Waal, no one would have dared to propose the dramatic behavioral differences recognized today between the bonobo and the chimpanzee. He issues a warning for paleontologists who are reconstructing social life from fossilized remnants of long-extinct species, problems that intensify when fossil bones are missing. Indeed, this is often the case, as Stephen Jay Gould commented, Ost hominid fossils, even though they serve as a basis for endless speculation and elaborate storytelling, are fragments of jaws and scraps of skulls. Flesh reconstructions of extinct hominins are likewise subjective. They often attempt to diminish the intellectual abilities of humans and overstate those of apes. One high school textbook caricatures Neanderthals as intellectually primitive even though they exhibited intelligence and culture, and casts Homo erectus as a bungling, stooped form, even though its skeleton is extremely similar to modern humans. Conversely, the same textbook portrays an Australopithecine, which in reality had a chimp-sized brain, with gleams of human-like intelligence and emotion, a common tactic in illustrated books on human origins. University of North Carolina, Charlotte anthropologist Jonathan Marks warns against such fallacies of humanizing apes and ape-ifying humans. The words of the famed physical anthropologist Ernest Houghton from Harvard University remain valid. Alleged restorations of ancient types of man have very little, if any, scientific value and are likely only to mislead the public. Third, the field itself is fragmented. The sparse nature of the data, combined with the desire to make confident assertions about human evolution, often betrays objectivity and leads to sharp disagreements. An article in Science titled "The Politics of Paleoanthropology" compared the task of reconstructing human evolution to that of reconstructing the plot of War and Peace with 13 randomly selected pages, leading to conflicts that make it difficult to separate the personal from the scientific disputes raging in the field. Similarly, Donald Johanson and Blake Edgar divulge that ambition and lifelong quests for recognition, funding, and fame can make it difficult for paleoanthropologists to admit when they are wrong. Human evolutionary studies have been plagued by debate and controversy. The reasons for argument over a fragment of a jaw, a partial skull, a handful of teeth, or an uncertain geological date are not that difficult to understand. Often a scrappy fossil generates diverse interpretations. The stakes are high. Hominid fossils are glamorous and bring scientific and popular success to those who find them. The fossils themselves become revered and coveted. The appearance of discordant evidence is sometimes met with a sturdy reiteration of our original views. It takes time for us to give up pet theories and assimilate the new information. In the meantime, scientific credibility and funding for more fieldwork hang in the balance. The quest for recognition can inspire outright contempt toward other researchers. After interviewing paleoanthropologists for a documentary, PBS Nova producer Mark Davis recounted that a Neanderthal expert thought the last one I talked to was an idiot, if not an actual Neanderthal. Paleoanthropology is rife with dissent and has few universally accepted theories. Even the most established and confidently promoted evolutionary models of human origins are based on limited evidence. As Nature editor Henry G. conceded, fossil evidence of human evolutionary history is fragmentary and open to various interpretations. 2. The standard story of human evolution. Despite the disagreements, there is a standard story of human evolution which is retold in countless textbooks, news media articles, and coffee table books. Indeed, virtually all of the scientists cited in this chapter accept some evolutionary account of human origins. A representation of the most commonly believed hominin phylogeny is portrayed in Figure 14.1. Starting with the early hominins, and moving through the Australopithecines, and then into the genus Homo, this chapter will review the fossil evidence and assess whether it supports this standard account of human evolution. As we shall see, the evidence, or lack thereof, often contradicts this evolutionary story. 3. Early hominins. In 2015, 2 leading paleoanthropologists reviewed the fossil evidence regarding human evolution in a prestigious scientific volume titled Macroevolution. They acknowledged the dearth of unambiguous evidence for ancestor-descendant lineages, and admitted the evolutionary sequence for the majority of hominin lineages is unknown. Most hominin taxa, particularly early hominins, have no obvious ancestors, and in most cases ancestor-descendant sequences, fossil time series, cannot be reliably constructed. Nevertheless, numerous theories have been promoted about early hominins and their ancestral relationships to humans. Sahelanthropus judensis, the Tumai skull. Although Sahelanthropus judensis, also called the Tumai skull, is known only from one skull and some jaw fragments, it has been called the oldest known hominin on the human line. But not everyone agrees. Brigitte Sennett, of the Natural History Museum in Paris, called Tumai the skull of a female gorilla, and Cow wrote in Nature that Sahelanthropus was an ape, not bipedal, and that many features link the specimen with chimpanzees, gorillas or both, to the exclusion of hominids. This debate has continued, but leading authorities caution that teeth and skull fragments alone are probably not reliable for reconstructing the phylogenetic relationships of higher primate species and genera, including those among the hominins. Unfortunately, for hominins like Sahelanthropus, craniodental fragments are all we have. In his testimony at the 2009 Texas evolution hearings, Ronald Wetherington stated that every fossil we find reinforces the sequence that we had previously supposed to exist rather than suggesting something different. But Sahelanthropus provides a striking counterexample to that assertion. Commenting on Tumai, Bernard Wood of George Washington University observed, "A single fossil can fundamentally change the way we reconstruct the tree of life," and explained that if this advanced-looking skull belonged to our ancestor, then it plays havoc with the tidy model of human origins. In other words, if Sahelanthropus was a human ancestor, then many later supposed human ancestors, including the Australopithecines, are forced out of our family line. Precious little Aurantugenensis. Auran, which means original man in a Kenyan language, was a chimpanzee-sized primate known only from an assortment of bone fragments, including pieces of the arm, thigh, lower jaw, and some teeth. When initially discovered, the New York Times declared, fossils may be earliest human link, and reported that Auran may be the earliest known ancestor of the human family. Nature responded to such hype by warning that the excitement needs to be tempered with caution in assessing the claim of a 6-million-year-old direct ancestor of modern humans. That seems like wise advice. Paleoanthropologists initially claimed Auran's femur indicated bipedal locomotion, appropriate for a population standing at the dawn of the human lineage, but a later Yale University press commentary admitted, all in all, there is currently precious little evidence bearing on how Auran moved. If Auran did prove an upright walking hominin from 6 million years ago, Meyer, would that qualify it as our ancestor? Not necessarily. Evolutionary paleoanthropologists often use bipedality as a litmus test for membership in the human lineage, but the fossil record has produced bipedal apes which even evolutionists recognize were far removed from human origins. According to a Nature commentary, one such ape, Aureopithecus, acquired bipedalism convergently with humans, and provides an object lesson that human-like features such as bipedalism don't necessarily indicate human ancestry. Much as the Tumai skull threatened to rewrite the hominin tree, if Auran were our ancestor it too would displace most Australopithecines from our lineage. Simply too little is known to determine its lifestyle, locomotion, or evolutionary relationships. As two critics argued in Nature, Auran is not a hominin, and we are a long way from a consensus on its role in human evolution. Ardipithecus ramidus, Irish stew or breakthrough of the year. In 2009, Science announced the long-awaited publication of Ardipithecus ramidus, a would-be hominin fossil that lived about 4.4 Meyer. Expectations mounted after its discoverer, UC Berkeley paleoanthropologist Tim White, promised a phenomenal individual that would be the Rosetta Stone for understanding bipedalism. The media eagerly employed the hominin they affectionately dubbed Ardi to evangelize the public for Darwin. Discovery Channel ran the headline Ardi, oldest human ancestor, unveiled, and quoted White calling Ardi as close as we have ever come to finding the last common ancestor of chimpanzees and humans. The Associated Press declared, world's oldest human-linked skeleton found, and stated, the new find provides evidence that chimps and humans evolved from some long-ago common ancestor. Science named Ardi the Breakthrough of the Year for 2009, and introduced her with the headline, A New Kind of Ancestor, Ardipithecus Unveiled. Calling Ardi new may have been a poor word choice, since she was discovered in the early 1990s. Why did it take some 15 years to publish the analyses? A 2002 article in Science explains the bones were so soft, crushed, squished, and chalky, that White reported, when I clean an edge it erodes, so I have to mold every one of the broken pieces to reconstruct it. Later reports similarly acknowledged that portions of Ardi's skeleton were found crushed nearly to smithereens and needed extensive digital reconstruction, including the pelvis, which looked like an Irish stew. National Geographic similarly noted that Ardi's trampled remains were badly crushed and distorted, and so fragile they would turn to dust at a touch. Claims about bipedal locomotion require accurate measurements of the precise shapes of key bones, like the pelvis. Can one trust declarations of a Rosetta Stone for understanding bipedalism when Ardi was crushed to smithereens? Science quoted various paleoanthropologists who were skeptical that the crushed pelvis really shows the anatomical details needed to demonstrate bipedality. Even some who accepted Ardi's reconstructions weren't satisfied that she was a bipedal human ancestor. Primatologist Esteban Sarmiento concluded in Science that all of the R. ramidus bipedal characters cited also serve the mechanical requisites of quadrupedality, and in the case of R. ramidus foot segment proportions, find their closest functional analog to those of gorillas, a terrestrial or semi-terrestrial quadruped and not a facultative or habitual biped. Bernard Wood questioned whether Ardy's postcranial skeleton qualified it as hominin. And Cow wrote in Nature that, if Ardipithecus is assumed to be a hominin, then it had remarkably high levels of homoplasy, similarity, among extant great apes. In other words, Ardy had ape-like characteristics which, if we set aside the preferences of Ardy's promoters, should imply a closer relationship to apes than to humans. An article in Nature stated that Ardy's being a human ancestor is by no means the simplest, or most parsimonious explanation. Stanford anthropologist Richard Klein agreed, "I frankly don't think Ardy was a hominid, or bipedal." Sarmiento even observed that Ardy had characteristics different from both humans and African apes, such as its unfused jaw joint, which ought to remove her far from human ancestry. Whatever Ardy was, everyone agrees she was initially badly crushed and needed extensive reconstruction. No doubt this debate will continue, but are we obligated to accept the human ancestor talking points promoted by Ardy's discoverers in the media? Sarmiento doesn't think so. According to Time magazine, he regards the hype around Ardy to have been overblown. 4. Later hominins, the Australopithecines. Many paleoanthropologists believe the Australopithecines were upright walking hominins and ancestral to our genus Homo. Dig into the details, however, and ask basic questions like who, where, and when, and there is much controversy. As we'll discuss in parts 5 and 6 of this chapter, one paper noted, there is little consensus on which species of Australopithecus is the closest to Homo, if any. Even the origin of genus Australopithecus itself is unclear. A. Retroactive confessions of ignorance. In 2006, National Geographic ran a story titled "Fossil Find is Missing Link in Human Evolution, Scientists Say," reporting the discovery of what the Associated Press called the most complete chain of human evolution so far. The fossils, belonging to species Australopithecus anamensis, were said to link Ardipithecus to its supposed Australopithecine descendants. What exactly was found? According to the technical paper, the claims were based upon canine teeth of intermediate masticatory robusticity. If a few teeth of intermediate size and shape make the most complete chain of human evolution so far, then the evidence for human evolution must be indeed quite modest. Besides learning to distrust media hype, there is another lesson here. Accompanying the praise of this missing link were retroactive confessions of ignorance, where evolutionists acknowledge a severe gap in their model only after thinking they have found evidence to plug that gap. Thus, the technical paper reporting these teeth admitted that, until recently, the origins of Australopithecus were obscured by a sparse fossil record. It continued. The origin of Australopithecus, the genus widely interpreted as ancestral to Homo, is a central problem in human evolutionary studies. Australopithecus species differ markedly from extant African apes and candidate ancestral hominids such as Ardipithecus, Auran, and Sahelanthropus. Evolutionists who retroactively confess ignorance about some previously unfilled gap risk falling into a situation where the new evidence which supposedly filled the gap may not prove very convincing. This seems to be the case here, where a couple of teeth were all that stood between an unsolved central problem in human evolutionary studies, the origin of Australopithecines, and the most complete chain of human evolution so far. Moreover, we're left with uncontested admissions that the Australopithecines differ markedly from their supposed early hominin ancestors. Australopithecine origins, apparently, remain obscure. B. Australopithecines are like apes. While Ardipithecus, Auran, and Sahelanthropus are controversial due to their fragmentary remains, there are sufficient known Australopithecine specimens to generally understand their morphology. Australopithecus, which literally means southern ape, is a genus of extinct hominins that lived in Africa from about 5 to 1.2 Meyer. Splitters, paleoanthropologists who infer many different species, and lumpers, those who see fewer, have created a variety of taxonomic schemes for the Australopithecines. The four most commonly accepted species are Afarensis, Africanus, Robustus, and Boisei. Robustus and Boisei are larger, boned and more robust, and are sometimes classified within genus Paranthropus. They are thought to represent a later living offshoot that went extinct without leaving any living descendants. The smaller, gracile forms, Afarensis and Africanus, probably lived earlier, and are classified within the genus Australopithecus. The most well-known Australopithecine fossil is Lucy, which belonged to Afarensis, one of the most complete known fossils among pre-Homo hominins. She is often described as a bipedal ape-like creature that is an ideal precursor to humans. In 2009, I visited a traveling exhibit of Lucy's bones. In addition to the fact that Lucy did not look human-like, see Figure 14.2, I was also struck by the incompleteness of her skeleton. Only 40% was found, and a large percentage is mere rib fragments. Very little useful material from Lucy's skull was recovered, and yet she is one of the most significant specimens ever found. There are some possible reasons for skepticism over whether Lucy represents a single individual, or even a single species. In an exhibit video, Lucy's discoverer, Donald Johansen, admitted that her bones were found scattered across a hillside. His written account quotes Lucy researcher Tim White further explaining how the bones were not found together, since the fossil wasn't found in situ, it could have come from anywhere above. There's no matrix on any of the bones we've found either. All you can do is make probability statements, that Lucy was a single individual. Johansen admits that one more rainstorm might have washed Lucy's bones away, never to be found. What might have happened during prior storms to mix her up with who knows what? Could Lucy represent bones from multiple individuals or even multiple species? The classic rejoinder notes that none of Lucy's bones appear duplicated, suggesting they represent a single individual. This is certainly possible, but given the fragmented and scattered nature of the skeleton, the rebuttal is far from conclusive. In particular, it's difficult to definitively say that key bones said to indicate her upright posture, the half pelvis and half femur, belong to one individual. Indeed, in 2015 it was decided that one of Lucy's vertebrae probably came from a baboon. The scientist who discovered the apparent anomaly explained it was washed or was otherwise transported in the mix of Lucy's remains. Could the same have happened with any of her other bones? Regardless, the Lucy exhibit confidently asserted, Lucy's species walked bipedally, in much the same way as we do, and her skeleton, approximate, a chimpanzee-like head perched atop a human-like body. Lucy did have a small, chimp-like head, as paleoanthropologist Lee Berger observes. Lucy's face would have been prognathic, jutting out almost to the same degree as a modern chimpanzee. But Bernard would refute the misapprehension that she resembled some ape-human hybrid. Australopithecines are often wrongly thought to have had a mosaic of modern human and modern ape features, or, worse, are regarded as a group of failed humans. Australopithecines were neither of these. Others have questioned whether Lucy walked like humans or was significantly bipedal. Mark Collard and Leslie Aiello observe in Nature that much of her body was quite ape-like, especially with respect to the relatively long and curved fingers, relatively long arms, and funnel-shaped chest. They report good evidence from Lucy's hand bones that her species, knuckle, walked, as chimps and gorillas do. A New Scientist article adds that Lucy appears well adapted for climbing, since everything about her skeleton, from fingertips to toes, suggests that Lucy and her sisters retained several traits that would be very suitable for climbing in trees. Richard Leakey and Roger Lewin argue that Afarensis and other Australopithecines almost certainly were not adapted to a striding gait and running, as humans are. They recount paleontologist Peter Schmid's striking surprise upon realizing Lucy's non-human qualities. Everyone had talked about Lucy as being very modern, very human, so I was surprised by what I saw. What you see in Australopithecus is not what you'd want in an efficient bipedal running animal. The shoulders were high, and, combined with the funnel-shaped chest, would have made arm swinging very improbable in the human sense. It wouldn't have been able to lift its thorax for the kind of deep breathing that we do when we run. The abdomen was pot-bellied, and there was no waist, so that would have restricted the flexibility that's essential to human running. Other studies confirm Australopithecine differences from humans, and similarities with apes. Their inner ear canals, responsible for balance and related to locomotion, are different from Homo but similar to great apes. Traits like their ape-like developmental patterns and ape-like ability for prehensile grasping by their toes led a nature reviewer to say that ecologically they, Australopithecines, may still be considered as apes. Another analysis in Nature found the Australopithecine skeleton shows a mosaic of features unique to themselves and features bearing some resemblances to those of the orangutan, and concluded the possibility that any of the Australopithecines is a direct part of human ancestry recedes. A 2007 paper reported, gorilla-like anatomy on Australopithecus afarensis mandibles, which was unexpected, and cast doubt on the role of afarensis as a modern human ancestor. As for Lucy's pelvis, many claim it indicates bipedal locomotion, but Johansson and his team reported it was badly crushed, with distortion and cracking, when first discovered. These problems led one commentator in the Journal of Human Evolution to propose that Lucy's pelvis is so different from other Australopithecines and so close to the human condition, due to error in the reconstruction, creating a very human-like sacral plane. Another paper concluded that a lack of clear fossil data prevents paleoanthropologists from making firm conclusions about Lucy's mode of locomotion. Prevailing views of Lucy's posture are almost impossible to reconcile. To resolve such differences, more anatomical, fossil evidence is needed. The available data at present are open to widely different interpretations. Paleoanthropologist Leslie Aiello states that when it comes to locomotion, Australopithecines are like apes, and the Homo group are like humans. Something major occurred when Homo evolved, and it wasn't just in the brain. The something major was the abrupt appearance of the human-like body plan, without direct evolutionary precursors in the fossil record. Lacking intermediates. If humans evolved from ape-like creatures, what were the transitional species between the ape-like hominins just discussed and the truly human-like members of the Homo genus found in the fossil record? There aren't any good candidates. The demise of Homo habilis. Many have cited Homo habilis, literally handy man, as a tool-using species that was a transitional link between the Australopithecines and Homo. But its association with tools is doubtful and appears driven mainly by evolutionary considerations. Many questions remain about exactly which specimens belong to habilis. Ian Tattersall, an anthropologist at the American Museum of Natural History, calls it a wastebasket taxon, little more than a convenient recipient for a motley assortment of hominin fossils, and believes more than one hominid species is represented. Penn State paleoanthropologist Alan Walker explains the severity of these disagreements. His is not a matter of some fragmentary fossils that are difficult to agree on. Whole crania are placed by different people in different species or even genera. Even ignoring these difficulties and assuming habilis was a real species, chronology precludes it from being ancestral to Homo. Habiline remains postdate the earliest fossil evidence of the genus Homo. Morphological analyses further confirm that habilis makes an unlikely intermediate between Australopithecus and Homo, and show Homo habilis doesn't even belong in Homo. An authoritative review in Science, by Bernard Wood and Mark Collard, found that habilis differs from Homo in terms of body size, shape, mode of locomotion, jaws and teeth, developmental patterns, and brain size, and should be reclassified within Australopithecus. Another article in Science similarly notes that habilis matured and moved less like a human and more like an Australopithecine, and had a diet more like Lucy's than that of H. erectus. Another paper found postcranial remains of H. habilis appear to reflect an Australopith-like body plan. Like the Australopithecines, many features of habilis indicate they were more similar to modern apes than to humans. According to Wood, habilines grew their teeth rapidly, like an African ape, in contrast to the slow dental development of modern humans. An analysis in Nature of habilis ear canals found its skull is most similar to baboons and suggested habilis relied less on bipedal behavior than the Australopithecines. The article concluded that the unique labyrinth of the habilis skull represents an unlikely intermediate between the morphologies seen in the Australopithecines and H. erectus. A study by Sigrid Hartwig Scherer and Robert D. Martin in the Journal of Human Evolution found the skeleton of habilis was more similar to living apes than were other Australopithecines like Lucy. They conclude, it is difficult to accept an evolutionary sequence in which Homo habilis, with less human-like locomotor adaptations, is intermediate between Australopithecus afarensis and fully bipedal Homo erectus. Alan Walker and Pat Shipman similarly called habilis more ape-like than Lucy and remarked, rather than representing an intermediate between Lucy and humans, habilis looked very much like an intermediate between the ancestral chimp-like condition and Lucy. Hartwig Scherer explains that Habili displays much stronger similarities to African ape limb proportions than Lucy, results she calls unexpected in view of previous accounts of Homo habilis as a link between Australopithecines and humans. Homo naledi versus Australopithecus sediba, the link resurrected? The news media might be heavily biased toward evolution, but at least it is predictable. Whenever a new hominin fossil is discovered, reporters seize the opportunity to push human evolution. Thus it was no surprise when news outlets buzzed about the latest human ancestor after a new species, Homo naledi, was unveiled in 2015. CNN declared, Homo naledi, new species of human ancestor discovered in South Africa. The Daily Mail reported, scientists discover skull of new human ancestor Homo naledi. PBS pronounced, trove of fossils from a long-lost human ancestor. And so on. To be sure, the find is striking because it represents probably the largest cache of hominin bones, many hundreds, ever found. In a field where a single scrap of jaw ignites the community, this is a big deal. But do we know that Homo naledi is a human ancestor, as so many news outlets declared? As usual, dig into the details and the answer is no. The primary claim about Homo naledi is that it was a transitional form or mosaic, a small-brained, upright-walking hominin with a trunk similar to the Australopithecines, but with human-like hands and feet. But the technical material shows that even some of those supposedly human-like traits have unique features. The hands showed a unique combination of anatomy, including unique first metacarpal morphology, and long, curved fingers which suggest naledi was, unlike humans, well-suited for climbing and suspension. Its foot differs from modern humans in having more curved proximal pedal phalanges, and features suggestive of a reduced medial longitudinal arch, giving it an overall unique locomotor repertoire. The foot shows, again, that unlike humans, it was likely comfortable climbing trees. The technical papers also reveal unique features in the femur and tibia, making a hindlimb which differs from those of all other known hominins. As for the head, cranial morphology of H. naledi is unique. Sound familiar? Whatever it was, overall naledi appears highly unique. Indeed, the discoverers of naledi called it a unique mosaic previously unknown in the human fossil record. Such terminology should raise a red flag. In the parlance of evolutionary biology, mosaic usually means a fossil has a suite of traits that are difficult to fit into the standard evolutionary tree. This is the case here. In 2010, some of the same scientists who discovered and promoted naledi, a team led by Lee Berger of the University of Witwatersrand, were promoting a different hominin species, Australopithecus sediba, as the intermediate du jour between the Australopithecines and Homo. However, sediba and naledi differ in important ways that make them unlikely partners in an evolutionary lineage. Specifically, sediba, classified within Australopithecus, had an advanced Homo-like pelvis, surprisingly human teeth, and a human-like lower trunk, whereas naledi, placed within Homo, bears an Australopith-like and primitive pelvis, primitive teeth, and a primitive or Australopith-like trunk. An Australopithecine with apparently advanced Homo-like features seems a poor candidate to evolve into a member of Homo with primitive Australopith-like versions of those same features. If the goal is to elucidate a lineage ending with modern humans, key traits are evolving in the wrong direction. Thus, although both sediba and naledi have been called a human ancestor by some of the same people, no less, evolutionarily speaking, both claims should not be true unless you're willing to tolerate a very messy tree. As one news outlet put it, each, sediba and naledi, has different sets of Australopith-like and human-like traits that can't be easily reconciled on the same family tree. Paleoanthropologists have also criticized claims that sediba was our ancestor. One called it way too primitive to be the ancestor of the human genus Homo and warned the fossil is surrounded by hype and overinterpretation. Another thought it merely represents Australopithecus africanus. Others have observed that sediba postdates Homo and has the wrong traits. Commenting on sediba, Harvard's Daniel Lieberman said, "The origins of the genus Homo remain as murky as ever," and Donald Johansson remarked, "The transition to Homo continues to be almost totally confusing." Even Lee Berger acknowledged when publishing on sediba that the ancestry of Homo and its relation to earlier Australopithecines remain unresolved. Another dubious claim about Naledi is that it intentionally buried its dead, a testimony to its supposedly human-like intellect. Burying dead in the cave where it was found would require shimmying through a steep, narrow crevice while dragging a body a long distance in the dark, a physically challenging task for any hominin of any level of intelligence. For many reasons, multiple scientists, including two of Berger's colleagues at the University of Witwatersrand, dispute the intentional burial hypothesis. Alison Brooks of George Washington University observed that claims of intentional burial are so far out there that they really need a higher standard of proof. There are also controversies about whether Naledi belongs within Homo. Biological classification is a subjective enterprise, but given the species' small brain and Australopith-like pelvis and trunk, its placement within Homo has proven controversial. Even Berger predicted, there may be debate over the Homo designation, as the species is quite different from anything else we have seen. Ian Tattersall writes that Naledi possesses an unusual combination of morphological attributes, which seem to align more with the Australopiths than with any Homo. A 2016 study found that Naledi's place within Homo is ambiguous, and concluded Naledi doesn't appear intermediate between Australopithecus and Homo. A final death blow to claims for Homo naledi as an ancestral or transitional fossil is its age. When first published, Naledi's promoters suggested, on the basis of evolutionary considerations rather than geological evidence, that it lived 2 to 3 Meyer. But at that time the fossils hadn't been dated geologically, leading Carol Ward of the University of Missouri to warn, without dates, the fossils reveal almost nothing about hominin evolution. This didn't stop paleoanthropologists from speculating about its evolutionary importance. They claim Naledi probably lived 2 to 3 Meyer and stated, if these fossils are late Pliocene, 2.5 Meyer, or early Pleistocene, 2.2 Meyer, it is possible that this new species of small-brained, early Homo represents an intermediate between Australopithecus and Homo erectus. In 2017 Homo naledi's remains were surprisingly dated to the startlingly young age of between 236,000 and 335,000 years, an order of magnitude younger than the age predicted by evolutionary considerations, and far too young to be related to the evolution of humans or ancestral to our species. Writing for BioLogos, anthropologist James Kidder candidly admitted, nearly everyone in the scientific community thought that the date of the Homo naledi fossils, when calculated, would fall within the same general time period as other primitive early Homo remains. We were wrong. 5 years before the unveiling of Naledi, the media touted Sediba as the newest human ancestor. But cooler heads prevailed, and it was shown that Sediba was not our ancestor. What will become of Homo naledi? Many have protested the hype over Naledi, and its trajectory resembles other hominins for which hyped claims of transitional or ancestral status eventually failed. When evaluating media claims of the newest human ancestor, a dose of healthy skepticism is warranted. 6. A Big Bang origin of Homo. After realizing that Habili could not serve as a link between Homo and Australopithecus, two paleoanthropologists lamented, this muddle leaves Homo erectus without a clear ancestor, without a past. Indeed, with species like Habili, Sediba, or Naledi unable to serve as intermediates, it is difficult to find fossil hominins to serve as direct transitional forms between the ape-like Australopithecines and the first human-like members of Homo. The fossil record shows abrupt changes which correspond to the appearance of our genus Homo about 2 Meyer. From its first appearance, Homo erectus was very human-like, and differed markedly from prior hominins which were not human-like. Yet Homo erectus appears abruptly, without apparent evolutionary precursors. An article in Nature explains the origins of the widespread, polymorphic, early Pleistocene H. erectus lineage remain elusive. The marked contrast between any potential ancestor, Homo habili or other, and the earliest known H. erectus might signal an abrupt evolutionary emergence sometime before its first known appearance in Africa at 1.78 MYR million years ago. Uncertainties surrounding the taxon's appearance in Eurasia and Southeast Asia make it impossible to establish accurately the time or place of origin for H. erectus. Whatever its time and place of origin, and direction of spread, this species dispersed widely, and possibly abruptly, before 1.5 MYR. That article was written in 2002, but the problem remains. A 2016 paper admits, although the transition from Australopithecus to Homo is usually thought of as a momentous transformation, the fossil record bearing on the origin and earliest evolution of Homo is virtually undocumented. While that paper argues that the evolutionary distance between Australopithecus and Homo is small, it nonetheless concedes that, by almost all accounts, the earliest populations of the Homo lineage emerged from a still unknown ancestral species in Africa at some point between approximately 3 and approximately 2 million years ago. Early members of Homo, namely Homo erectus, show unique and previously unseen features that contributed to this abrupt appearance. The technical literature observes an explosion, rapid increase, and approximate doubling in brain size associated with the appearance of Homo. Wood and Collard's major science review found that only one single trait of one hominin species qualified as intermediate between Australopithecus and Homo, the brain size of Homo erectus. However, this one trait does not necessarily indicate that humans evolved from less intelligent hominids. Intelligence is determined largely by internal brain organization, and is much more complex than the singular dimension of brain size. Indeed, brain size is not always a good indicator of intelligence or evolutionary relationships. As Wood and Collard observe, relative brain size does not group the fossil hominins in the same way as the other variables. This pattern suggests that the link between relative brain size and adaptive zone is a complex one. Case in point, Neanderthals had an average skull size larger than modern humans. Given the range of modern human skull size variation, see Table 14.1, a progression of relatively small to very large skulls could easily be concocted using bones from living humans alone. Such a lineup might give the false impression of some evolutionary lineage when it is no such thing. Moreover, as seen in Table 14.1, Erectus had an average brain size within the range of modern human variation. Finding a few skulls of intermediate size does little to demonstrate that humans evolved from primitive ancestors. Much like the explosive increase in skull size, a study of the pelvis bones of Australopithecines and Homo found a period of very rapid evolution corresponding to the emergence of the genus Homo. One Nature paper noted that early Homo erectus shows such a radical departure from previous forms of Homo, such as H. habilis, in its height, reduced sexual dimorphism, long limbs and modern body proportions that it is hard at present to identify its immediate ancestry in East Africa, or anywhere else, for that matter. A paper in the Journal of Molecular Biology and Evolution found that Homo and Australopithecus differ significantly in brain size, dental function, increased cranial buttressing, expanded body height, vision, and respiration, and stated, "We, like many others, interpret the anatomical evidence to show that early H. sapiens was significantly and dramatically different from Australopithecines in virtually every element of its skeleton and every remnant of its behavior." Noting these many differences, the study called The Origin of Humans a real acceleration of evolutionary change from the more slowly changing pace of Australopithecine evolution, and stated that such a transformation would have required radical changes. The anatomy of the earliest H. sapiens sample indicates significant modifications of the ancestral genome and is not simply an extension of evolutionary trends in an earlier Australopithecine lineage throughout the Pliocene. In fact, its combination of features never appears earlier. These rapid and unique changes are termed a genetic revolution, where no Australopithecine species is obviously transitional. For those not constrained by an evolutionary paradigm, it is not obvious that this transition took place at all. The stark lack of fossil evidence for this hypothesized transition is confirmed by 3 Harvard paleoanthropologists. Of the various transitions that occurred during human evolution, the transition from Australopithecus to Homo was undoubtedly one of the most critical in its magnitude and consequences. As with many key evolutionary events, there is both good and bad news. First, the bad news is that many details of this transition are obscure because of the paucity of the fossil and archaeological records. As for the good news, they admit, thus we lack many details about exactly how, when, and where the transition occurred from Australopithecus to Homo. We have sufficient data from before and after the transition to make some inferences about the overall nature of key changes that did occur. In other words, the fossil record shows ape-like Australopithecines, before, and human-like Homo, after, but not fossils documenting a transition between them. In the absence of intermediates, we're left with inferences of a transition based strictly on the assumption of evolution, that an undocumented transition must have occurred somehow, sometime, and someplace. They assume this transition happened, even though we do not have fossils documenting it. Even science admits, our genus Homo is thought to have evolved a little more than 2 million years ago from the earlier hominid Australopithecus. But there are few fossils that provide detailed information on this transition. Likewise, William Kimbell of Arizona State University stated in Nature, the evolutionary events that led to the origin of the Homo lineage are an enduring puzzle in paleoanthropology. Another review similarly notes the seemingly abrupt appearance of H. erectus. Similarly, the great evolutionary biologist Ernst Mayr recognized the abrupt appearance of our genus. The earliest fossils of Homo, Homo rudolfensis and Homo erectus, are separated from Australopithecus by a large, unbridged gap. How can we explain this seeming saltation? Not having any fossils that can serve as missing links, we have to fall back on the time-honored method of historical science, the construction of a historical narrative. Another commentator proposed that the evidence implies a Big Bang theory of the appearance of Homo. This large, unbridged gap between the ape-like Australopithecines and the abruptly appearing human-like members of genus Homo challenges evolutionary accounts of human origins. 7. All in the family. In contrast to the Australopithecines, the major members of Homo erectus and the Neanderthals, Homo neanderthalensis, are very similar to us. Some paleoanthropologists have even classified erectus and neanderthalensis as members of our own species, Homo sapiens. Homo erectus appears in the fossil record a little over 2 Meyer. Its name means upright man, and unsurprisingly, below the neck they were extremely similar to us. An Oxford University Press volume notes erectus was human-like in its stature, body mass, and body proportions. An analysis of 1.5 million-year-old Homo erectus footprints indicates a modern human style of walking and human-like social behaviors. Unlike the Australopithecines and habilines, erectus is the earliest species to demonstrate the modern human semicircular, ear-canal morphology. Another study found that total energy expenditure, TEE, a complex character related to body size, diet, and food-gathering activity, increased substantially in Homo erectus relative to the earlier Australopithecines, approaching the high TEE value of modern humans. Researchers believe the earliest representatives of Homo erectus were more similar to modern humans than to the earlier and contemporaneous Australopithecines, due to their larger relative brain sizes, larger bodies, slower rates of growth and maturation, dedicated bipedal locomotion, and smaller teeth and jaws, and major changes in diet and foraging behavior. These features reflect a lifestyle that was more similar to that of modern humans than that inferred for earlier and contemporaneous hominins. While the average brain size of Homo erectus is less than the modern human average, erectus cranial capacities are within the range of normal human variation. Table 14.1. Donald Johansson suggests that, were erectus alive today, it could mate with modern humans to produce fertile offspring. In other words, were it not for our separation by time, we might be considered interbreeding members of the same species. The Cambridge Encyclopedia of Human Evolution observes, it is very difficult judging whether a particular kind of early human had developed a language, society or art. Nonetheless, erectus remains have been found on islands, where the most reasonable explanation is that they arrived by boat. Carl Wegman of North Carolina State University said, "We all have this idea that early man was not terribly smart. The findings show otherwise. Our ancestors were smart enough to build boats and adventurous enough to want to use them." As for Neanderthals, though they have been stereotyped as bungling and primitive, if a Neanderthal walked down the street, you probably wouldn't notice. Wooden-collared note, skeletons of H. neanderthalensis indicate that their body shape was within the range of variation seen in modern humans. Washington University paleoanthropologist Eric Trinkhaus maintains that Neanderthals were no less intelligent than contemporary humans, and argues, they may have had heavier brows or broader noses or stockier builds, but behaviorally, socially and reproductively they were all just people. University of Bordeaux archaeologist Francesco Di Erico agrees, Neanderthals were using technology as advanced as that of contemporary anatomically modern humans and were using symbolism in much the same way. Though controversial, hard evidence backs these claims. Anthropologist Stephen Molnar explains that the estimated mean size of Neanderthal cranial capacity, 1,450 cubic centimeters, is actually higher than the mean for modern humans, 1,345 cubic centimeters. One paper in Nature suggested the morphological basis for human speech capability appears to have been fully developed in Neanderthals. Indeed, Neanderthal remains have been found associated with signs of culture including art, burial of their dead, and complex tools, including musical instruments like the flute. While dated, a 1908 report in Nature reports a Neanderthal-type skeleton wearing chain mail armor. Archaeologist Martin Aern said, regarding toolmaking, in many ways, Neanderthals were just as smart or just as good as us. Morphological mosaics, skeletons showing a mix of modern human and Neanderthal traits, suggest Neanderthals and modern humans are members of the same species who interbred freely. Indeed, scientists now report Neanderthal DNA markers in living humans, supporting proposals that Neanderthals were a subrace of our own species. As Trinkaus says regarding ancient Europeans and Neanderthals, he would understand both to be human. There's good reason to think that they did as well. Darwin skeptics continue to debate whether we are related to Neanderthals and Homo erectus, and evidence can be mounted both ways. The present point, however, is this: even if we do share common ancestry with Neanderthals or erectus, this does not show that we share ancestry with any non-human-like hominins. As noted, Leslie Aiello observes, Australopithecines are like apes, and the Homo group are like humans. According to Sigrid Hartwig Scheurer, the differences between human-like members of Homo such as erectus, Neanderthals, and us reflect mere microevolutionary effects of size variation, climatic stress, genetic drift and differential expression of common genes. Whether we are related to them or not, these small-scale differences do not show the evolution of humans from non-human-like or ape-like creatures. 9. Conclusion. Despite the claims of evolutionary paleoanthropologists and unceasing media hype, the fragmented hominin fossil record does not document the evolution of humans from ape-like precursors. While the hominin fossil record is marked by incomplete and fragmented fossils, known hominins fall into two separate groups, ape-like and human-like, with a distinct gap between them. The genus Homo appears in an abrupt, non-Darwinian fashion, without evidence of an evolutionary transition from ape-like hominins. Other members of Homo appear very similar to modern humans, and their differences amount to small-scale microevolutionary change, providing no evidence that we are related to non-human-like species. But there's more evidence that contradicts an evolutionary model. Many researchers have recognized an explosion of modern human-like culture in the archaeological record about 35 to 40,000 years ago, showing the abrupt appearance of human creativity, technology, art, and even paintings, showing the rapid emergence of self-awareness, group identity, and symbolic thought. One review dubbed this the creative explosion. Indeed, a 2014 paper co-authored by leading paleoanthropologists admits we have essentially no explanation of how and why our linguistic computations and representations evolved, since non-human animals provide virtually no relevant parallels to human linguistic communication. This abrupt appearance of modern human-like morphology, intellect, and culture contradicts evolutionary models, and may indicate design events in human history. Of course, there is room for civil disagreement among Christians on these questions. But theistic evolutionists who believe that humans evolved from ape-like species should, at the very least, temper their rhetoric in light of the sparse fossil evidence. They certainly should not demand that the Church accept the evolutionary consensus on human origins. The fossil evidence simply isn't that clear. If anything, the fossil record contradicts the central evolutionary claim that humans evolved from ape-like creatures. After all, as noted earlier, two top paleoanthropologists have admitted that the evolutionary sequence for the majority of hominin lineages is unknown. With the fossil evidence for human evolution so weak, why should our theistic evolutionist brothers and sisters insist that the Church must adopt their viewpoint? 15. Evidence for human uniqueness. Anne K. Gauger, Ola Hostja, and Colin R. Reeves. In terms of what makes us human, it's not the things that are the same, it is the things that are different that matter. Jonathan Marks. Summary. scientists claim that our extreme genetic similarity with chimps, on the order of 98.7% identity, indicates we share common ancestry. This statement neglects several facts. First, our genetic differences are larger than that number represents. Common estimates of similarity are based on comparisons of the single nucleotide changes only, while other kinds of genetic differences are disregarded. In addition, non-coding regions of DNA, long thought to be non-functional junk, contain many kinds of genetic regulatory elements, some of which are species-specific. These species-specific regulatory elements make up a very small proportion of the total count of differences, but have a significant effect on how our genome works. For example, many of these regulatory elements are known to affect gene expression in the brain. Taken together, these species-specific genetic differences contribute to our anatomical and physiological differences with chimps. In addition, there is not enough evolutionary time for all these coordinated changes to have happened by the mutation-selection process. Thus the evidences for common ancestry put forward by various scientists are not as solid as they might seem. The more we learn about our human genome, the more it seems to be brilliantly and uniquely designed. Introduction. In 2007, Science magazine published an essay titled Relative Differences, The Myth of 1%, by John Cohen. The essay challenged the received knowledge of our culture that we are genetically nearly identical with chimpanzees. Cohen detailed how our genetic differences are significantly higher than 1%. But this news did not penetrate the culture. Numerous articles, books, newspapers, and TV science shows still use the 1% figure. Even the major scientific journal Science used it as recently as 2012. The underlying concept that gives this figure its persistence is common descent. This concept is something that most biologists treat as axiomatic, untouchable, and completely self-evident, which is why the figure of 1% has such staying power. Even if they disagree with the neo-Darwinian account of evolution, and some scientists do, see Chapter 8, most scientists, including most theistic evolutionists, still accept our common ancestry with chimpanzees. The question at hand, then, concerns the steps that supposedly took us from being ape-like to being human. Are we fancy apes, with all the evolutionary baggage that comes along with that, or are we unique beings with capabilities far beyond the reach of animal species? Because we do not have access to much information about our supposed common ancestor with chimps, we will address this question by examining how we differ from chimps in our genetics, physiology, anatomy, and more. 2. Definitions. Before we begin, we need to define a few basic terms. Others will be defined as we go along. DNA is composed of two very long complementary strands of nucleotides, called A, C, T and G, with each nucleotide from one strand paired with one from the other strand. They always pair together in the same way, A with T and C with G, see Figure 15.1 for an illustration. Nucleotides. Nucleotides are the chemical building blocks out of which DNA and RNA are made. DNA expression. When DNA is copied into RNA and used to make something, we say that it has been expressed, just as reading a text out loud can be said to be expressing the text. Chromosomes. Our DNA is segmented into separate chromosomes, which function as units of inheritance. We typically have 46 chromosomes in each cell. Eggs and sperm have 23 chromosomes each. SNPs. When DNA is compared between two copies of a chromosome from different individuals, for example, differences are usually found. Often they are single nucleotide changes, as in Figure 15.2, and are called single nucleotide polymorphisms, SNPs. We can also compare a chimpanzee's chromosomes with ours. They are very similar enough to know which chimpanzee chromosome matches each of ours. What differences there are between our chromosomes and theirs can be identified. The differences can be as small as an SNP, or they can be rearrangements, duplications, deletions, or insertions of DNA. Gene. A gene is defined as DNA that carries information about how to make one or more proteins or RNAs. RNA. RNAs are strings of nucleotides that are copied from genes and are used in a variety of ways by the cell. RNAs express the information in the DNA in order to carry out its function. Amino acids. Amino acids are the molecular units that comprise a protein. Proteins. Proteins are the building blocks from which the cell is primarily constructed, and they do the work of the cell, carrying out chemical reactions necessary for the cell to function. Each protein is made of a long string of amino acids whose sequence, order in the string, is specified by its corresponding coding sequence in the DNA. Protein translation. DNA carries the information for how to make proteins, but the information carried in DNA needs to be interpreted by the cell. DNA is to be copied into RNA, DNA is expressed, and then translated into protein. Copying DNA into RNA and then translating it into protein is like translating the information in one language, the nucleotide sequence of the DNA, into another language, the amino acid sequence that makes a protein. Genetic code. The genetic code determines how the DNA is turned into protein. Each set of 3 particular nucleotides, such as AAA, AAC, AAT, AAG, ATA, ATC, ATG, or ATT, for example, specifies one particular amino acid. There are roughly 20 amino acids, and 64 possible nucleotide triplets, called codons, that specify them. Because of this, most amino acids are specified by more than one codon. Said another way, a DNA string contains the information for making protein in the form of a digital code made up of triplet nucleotides. This code must be read and translated in order to make protein. 3. 1% similarity? Sequencing the genome. Chimpanzees are our closest living relatives, according to standard evolutionary theory. It is assumed by the scientific establishment that we can look at their behavior, physiology, and genetics in order to learn both how we are the same and what makes us different. We can compare genomes, the whole complement of an organism's DNA, to find out which genes were crucial in making us who we are. We can identify differences in our DNA that perhaps make us prone to disease, or discover genes that may protect against disease. This was the rationale for sequencing the chimpanzee genome, and the project was begun in 2003, shortly after our own genome sequence was published. Published in September of 2005, the chimpanzee genome sequence was primarily done using the human genome as a scaffold, a framework for piecing the chimpanzee genome together. The chimpanzee DNA sequences, which had been deciphered in pieces, were ordered and assembled according to the arrangement of our human DNA in any places where the assembly was unclear. This means that the actual chimpanzee sequence could be different in order or sequence than has been reported, and we might not know. Because the sequencing process is prone to errors, each read of the sequence needed to be done multiple times. The preferred standard is at least 12-fold redundancy. Each nucleotide must be read at least 12 times. But the published chimpanzee genome has only a 3.6-fold sequence redundancy, which is problematic for determining which SNPs are genuine and which are errors in the sequencing process. Because of this low redundancy, the published chimpanzee genome sequence is generally referred to as a draft genome, not a definitive one. Even the canonical human genome sequence, which was completed in 2003, continues to be refined and annotated. Nevertheless, the consortium that sequenced the chimpanzee genome claimed that the sequence reads were high quality and could be used for analysis. We must await another chimpanzee genome sequence assembled de novo, without a human scaffold, and with higher multiplicity to know for sure. Comparing genomes. In the paper reporting that first sequence, the estimate for the percentage of SNPs in the human genome that differs when compared to the chimpanzee genome is 1.23%, 1.08% when variation between humans is taken into account. This low percentage difference is one of the reasons it is claimed that we share common ancestry with chimpanzees. At this level we appear very similar. But there are other kinds of differences between chimpanzee and human DNA. Insertions and deletions are 2 to 4%, different studies use different samples and techniques, so this variation in numbers is not surprising. Differences also exist between chimpanzee and human DNA in the number and location of repeating genetic elements. About 0.4% of the genome is comprised of repetitive genetic elements unique to humans, according to the chimpanzee sequencing consortium. In addition, the Y chromosomes of chimpanzee and human males are very different from each other. The Y chromosome represents 1.8% of the genome. Among protein-coding genes, copy number variations represent a large number of differences. Human sequences may have 2 or more copies of particular genes, while chimpanzees have only 1, for example. According to Demuth et al., our results imply that humans and chimpanzees differ by at least 6%, 1,418 of 22,000 genes, in their complement of genes, which stands in stark contrast to the oft-cited 1.5% difference between orthologous nucleotide sequences. This genomic revolving door of gene gain and loss represents a large number of genetic differences separating humans from our closest relatives. All told, based on current knowledge, there is at least a 5% difference in our DNA, and that does not count rearrangements in the DNA, where segments of the DNA appear flipped end-to-end in relation to chimp DNA, or where one segment of human DNA is in a different location than in chimpanzees. In truth, though, counting raw difference is not the best way to calculate how different we are genetically speaking, and there are several reasons why it is not surprising that we share 95% of our DNA sequence with chimps. First, our basic building blocks, the proteins out of which our cells are made and the enzymes that carry out cellular metabolism, are very similar to those of chimpanzees, almost identical in many cases. One can think of our genes as being like the bricks and mortar, nails and wood, shingles and wires out of which houses are made. 2 houses may look different but be composed of the same basic building blocks. By analogy, the building blocks out of which we are made, the genes, are very similar for chimps and humans, even if our bodily forms are different. Second, the vast majority of our DNA does not code for protein but functions like an operating system, determining what files, genes, should be used when, and where. The routine processes of life are carried out by this operating system, and we share these basic routines with chimps. Thus in many respects our operating systems are the same as those of chimps. Last, it is the things that are different that matter. As we will show, small sections of DNA can have a large effect on how things come together. 4. Differences in how our DNA is used. Back in the 1970s, when people were first beginning to get the idea that the human genome and chimpanzee genome were very similar, despite our obvious anatomic, physiological, and behavioral differences, Mary Claire King and A. C. Wilson proposed that the differences that matter might not be in the sequence of the DNA but in how it is put to use. At that time, there was not sufficient experimental precision to identify individual genes nor to detect the kinds of differences we will outline below. We now know that when, where, and how our DNA is used matters much more than an overall count of nucleotide differences. Human-specific differences in gene regulation, as we will see, are what make us unique. Human-specific genes. We have 20,000 or so genes. We now know that some are unique to us. Estimates vary as to how many. One report says there are about 300 human-specific protein-coding genes, mainly due to new duplicates of existing genes. Another paper reports over 600. These new genes have been linked to disease, meaning their overexpression or underexpression can affect the progress of disease. They tend to be expressed in the brain and the testes. As many as 60 de novo genes, not arising by duplication of existing genes but genuinely new, have also been reported. These genes may be disease-associated also, meaning they are likely to have function. It is worth noting that even one novel, functional gene is a remarkable thing from an evolutionary point of view. It used to be accepted that getting new genes was very hard, but now new genes are being found in every genome that is sequenced, genes that appear to be unique to that species. This was a surprise for evolutionary biologists. From a design point of view, however, the existence of new genes for each species makes sense. Multipurpose genes. It was discovered in the late 1970s that protein-coding genes can be used to encode many different proteins. The genes are broken up into segments called exons, and after the genes have been transcribed into RNA, the RNA can be spliced into different arrangements of exons. Say a gene has exons 1 to 5. The RNA can be spliced to include exons 1 and 2, or 2, 3, 4, or 1, 3, 4, or 4, 5, for example, figure 15.3. There can also be genes expressed in the opposite direction, for example 5, 3, 2. So although we have only about 20,000 genes, we have potentially many thousands of different protein-coding RNAs made from those genes, with each unique RNA making a different protein. There is an important role for alternative splicing in establishing differences between humans and chimpanzees, one study reports. In fact, 6 to 8% of genes that have been studied display splicing differences between chimps and humans. That means that our genes may be used to make different proteins, even though they appear to have the same DNA sequence. Differential gene expression. Different tissues and organs in the body are made of specific cell types, and each cell type expresses its own set of genes. The pattern of expression of genes affects what kind of cell it becomes. So where, when, and what genes are expressed all matter to the development of the body, and for its continued functioning. It follows that, in order for us to develop into humans and not chimpanzees, it is likely that differences in gene expression are involved. Significantly, there are substantial differences in gene expression between humans and chimpanzees, particularly in the brain. There are multiple kinds of elements involved in regulating differential gene expression. The most well-known are protein transcription factors, roughly 1 to 3% of them human-specific. The transcription factors recognize and bind to particular DNA binding sites, and influence the activity of their neighboring genes, figure 15.4. Some increase gene expression, some decrease it, some are tissue-specific, and some are general enhancers of gene expression. Some silence genes completely. 1 to 3% of our transcription factors are unique to us. This is significant because a single transcription factor can change the expression of multiple genes, thus having a greatly magnified effect. Those unique transcription factors can have a significant effect on how our genome is used, even though they account for very little of the total DNA themselves. Non-coding DNA and its functions. Only about 2% of our genome carries the information to make protein, an astonishing fact. The remainder is non-coding, it does not specify how to make protein. Roughly half of that non-coding DNA is composed of repeating elements, sometimes called mobile genetic elements. This is another astonishing fact, that so much of our DNA should be composed of repeating DNA. Many of these repeating elements resemble a certain kind of viral DNA that can copy itself and insert in new locations in the genome, though most of these mobile genetic elements are inactive. For a number of years, many scientists assumed that the non-protein coding DNA was junk, the detritus of evolution, like that junk drawer at home where bits of miscellany with no obvious purpose end up. This was based on the idea that mobile elements had copied and inserted themselves into new locations, accumulating over time, and elements that had become broken were inefficiently removed. Broken genes also accumulated. We now know that at least some of this junk DNA is involved in modulating the behavior and structure of chromosomes, regulating the expression of genes, as well as perhaps providing ways for organisms to respond to their environments genetically, as will be discussed below. The change in view is exemplified by the about face on junk DNA by Francis Collins, director of the National Institutes of Health. As Marvin Oleski reports at World Magazine. Collins claimed on page 136 of his book The Language of God that huge chunks of our genome are littered with ancient repetitive elements, Rs, so that roughly 45% of the human genome is made up of such genetic flotsam and jetsam. In a talk given in New York, he claimed the existence of junk DNA was proof that man and mice had a common ancestor, because God would not have created man with useless genes. Last year, though, speaking at the J.P. Morgan Healthcare Conference in San Francisco, Collins threw in the towel. In terms of junk DNA, we don't use that term anymore because I think it was pretty much a case of hubris to imagine that we could dispense with any part of the genome, as if we knew enough to say it wasn't functional. Most of the genome that we used to think was there for spacer turns out to be doing stuff. We should be clear, though. Collins has not given up his position that chimps and humans have a common ancestor. We focus below on specific kinds of junk DNA because they make up a large portion of our genome, and have in many cases been shown to have function. Signs and lines. Short interspersed nuclear elements, signs, are a type of mobile genetic element that represents about 12% of the genome. Among other things, signs help to specify in which cells genes should be expressed. 7,000 ALUS, a kind of sign, are species-specific, present in humans but not in chimpanzees. Signs tend to be present near genes of particular functional types, and affect their expression, often in cell-type specific ways. Human-specific ALU elements, as they are called, are also involved in RNA editing, a process by which RNA is changed after it has been copied from the DNA. RNA editing can affect many aspects of RNA processing, such as splicing and stability. This sign-induced RNA editing is most notable in the human brain, and is largely species-specific. Of course, the brain is one of the areas where we differ most from chimpanzees. But RNA editing is not just in the brain. It is essential for development, and improper editing results in diseases like cancer and psychiatric disorders. Other mobile genetic elements, called long interspersed nuclear elements, lines, are about 17% of the genome. Many, about 1,800, are species-specific. They have a role in regulating the way chromosomes behave, where they cluster in the nucleus, how they are packed, and they inhibit gene expression. Thus they play an important role in the organization of the nucleus. They also tend to flank groups of genes, so as to control the expression of RNAs from different strands of the DNA. In other words, they delineate units of gene expression. Lines also help direct different kinds of cells to develop in the brain. Lines move to new locations in the genome of particular brain cells as they develop, landing near different genes involved in neuronal development. That means many of our brain cells literally rewrite their genetic instructions during the process of becoming mature neurons. The same is true in other tissues of the body as well. To reiterate, many lines and signs are species-specific, meaning we have a unique set of these elements in our genomes compared to chimpanzees. From all we have said, it should be apparent that these species-specific elements can make a profound difference in our anatomy, physiology, and behavior. Not to mention our brains. Long non-coding RNAs. 70 to 90% of the genome is transcribed into RNA but does not produce any protein. Called long non-coding RNAs, lncRNAs, these RNAs can be nested within genes going in either direction, or they can come from DNA with no protein-coding genes. They often originate from or contain signs and lines. Figure 15.5 illustrates the kinds of lncRNAs that are possible. These RNAs have been a matter of some controversy. They tend to be species-specific. The evolutionary biologists like to say they are not conserved across species, and so they have been called junk, the product of indiscriminate transcription like so much background noise. Yet when examined, they do have functions. They often act by changing the DNA architecture. Some link different stretches of DNA together, looping the DNA into functional domains, or causing changes in gene expression. Others tether the DNA to the nuclear periphery when it is not needed. Still others act as buffers to prevent gene expression from different segments of DNA from interfering with one another. This is important because overexpression can lead to diseases such as cancer. Many of these lncRNAs are also tissue-specific. For example, there are human-specific lncRNAs expressed in the brain. HARs. Human accelerated regions are human DNA segments whose sequences differ substantially from related mammalian sequences. The mammalian sequences are conserved, meaning they differ very little among species. Yet the human HARs are quite different. The evolutionary interpretation is that there has been a burst of evolutionary change in these regions, after the separation of the chimpanzee and the human lineages. HARs could also be different by design. Interestingly, these HARs tend to be located near developmental genes, transcription factors, and genes expressed in the central nervous system. More differences. All non-human primates, in fact most mammals, carry endemic infectious retroviruses, a kind of virus that integrates itself into the host genome in order to replicate itself. There are two kinds, simian foamy viruses, SFVs, and simian infectious retroviruses, SIVs, that are shared among most non-human primates. Among humans, we have only the newly introduced HIV and human T leukemia virus, and no others. If we were descended from a common ancestral population with chimpanzees, we should carry the SIV and SFV's viruses also, but we don't. Either we were purged of the viruses or we never had them. The organization of our DNA sequence differs considerably. We have different recombination hotspots than chimpanzees. These hotspots are places where homologous chromosomes recombine, shuffling segments of DNA into new combinations. As a result, our DNA is organized into different shuffled blocks. As for rearrangements of the DNA, stretches of DNA are inverted in humans, compared to chimpanzees, contributing greatly to sequence divergence. Insertions of 10 to 15,000 bases of DNA account for 32 million bases of human-specific DNA. Insertions of greater than 15,000 bases contribute 8 million bases of human-specific DNA. Finally, our chromosome 2 differs from that of chimpanzees. See the section on chromosomal fusion near the end of this chapter for more on this. Genetic networks. Transcription factors, signs, lines, and lncRNAs all act to regulate genes in a sort of genetic network of interactions. Some genes interact with other genes, which in turn interact with still other genes. Within this network of gene interactions, some genes act as hubs of connectivity. If the expression of these hub genes is shifted, they affect the expression of many other genes. Thus, changing the expression of a few genes can have a large effect. For example, an estimated 17% of the neural network in the cortex of the brain is unique to humans, even though our total genomes may differ from chimpanzees by only 5%. Other parts of the brain show other differences also. This means that small changes in gene regulatory elements like the ones mentioned above can have large effects on our gene networks, and thus on our physical and behavioral characteristics. The genome as an operating system. If the genome sounds complicated, it is. It needs to be complicated in order to enable the timing and location of differential gene expression. Genes are grouped into folders, and then into superfolders. DNA is organized into loops, then megafolders. Chromosomes are arrayed in the nucleus, each with its own territory, so that sets of genes can be expressed in a coordinated fashion. Genes in active folders take various inputs and combine them into a single output, functioning like logic gates. The output of all the cellular logic gates combines, and the outcome is carefully regulated cellular behavior, and ultimately, a functioning human being. We may share 95% or even 99% of our DNA sequence with chimpanzees, but it's how the DNA is used that matters. Thus, one can say that our genome functions like a very complex operating system, more complex than anything we have ever built. Our genome and its coordinated expression are a wonder of intricacy, responsiveness, and beauty, far, far more sophisticated than anything we have dreamed of. Looking down into the depths of that complexity is truly dizzying, leaving one with a sense of awe. Physiological and anatomical differences. Given all the genetic differences and regulatory complexity described above, it should come as no surprise that our physiology and anatomy is different from that of chimpanzees. We do not have the same reproductive biology. Our teeth develop more slowly after birth than chimpanzees' teeth do, and our young are born profoundly helpless and require prolonged maternal care. Our brains are larger and continue to develop long after birth, forming neurons and connections at a rapid rate. In fact, new neurons continue to form even into adulthood. Our musculature is weaker, with smaller bone insertion points. Our thyroid hormone metabolism differs. We get AIDS and malaria and chimpanzees don't. Our immune systems differ. Our diets differ, and our intestines reflect that difference. We shed tears, chimpanzees don't. We can swim and have a diving reflex, but chimpanzees can't swim. We have chins, chimpanzees do not. We walk and run upright. Our feet are different, ours are designed for walking and chimpanzees for climbing. Our necks are longer, and the skull is set on top of the spine for balance and to be able to look forward. Our rib cages move freely from side to side to accommodate our gait, and expand to allow deep breathing during running. Our shoulders are designed for throwing, while chimpanzees are designed for climbing. Our pelvis and hips are oriented so as to permit upright walking. Our legs angle in so that our feet are underneath us. Our inner ear canals are oriented differently to increase our sense of balance. Our hands are designed for tool use, not knuckle walking. We have a poorer sense of smell but a greater tactile sensitivity in our fingertips. We have greater fine motor control, and our thumbs can touch the far side of our hands. Finally, there are all the cultural and behavioral differences. We plan. We think about the past and the future. We make intentional decisions. We can delay gratification for long periods. We engage in long-range trade. Adults play. We dance. We make music. We have language and communicate symbolically, and we write novels and poetry. We have mathematics and art. We domesticate animals and engage in agriculture. We wear clothing. We engage in hospitality. We control fire and we measure time. We practice religion and bury the dead. We have empathy for others and altruism on a scale unknown in the animal world. We care for the infirm and the elderly. Some may say that these traits are inherited from our common ancestors, that they are all honed by natural selection to help the individual compete. But we see nothing like the human scale of behavior in chimpanzees. Our culture is exceptional, even unique, by any standard of the animal kingdom. It is orders of magnitude more sophisticated than anything chimpanzees do. A paper titled Comparing the Human and Chimpanzee Genomes, Searching for Needles in a Haystack, by Ajit Varkey and Tasha Althide, describes literally 100 or more physiological and behavioral differences, some of which we have cited here. Bramble and Lieberman, in the journal Nature, list the changes necessary for long-distance running. They include 26 anatomical and physiological innovations. The most striking trait that distinguishes us from chimpanzees is our intellect. There is no evolutionary explanation for its appearance. Varkey et al. State. It is difficult to explain how conventional natural selection could have selected ahead of time for the remarkable capabilities of the human mind, which we are still continuing to explore today. An example is writing, which was invented long after the human mind evolved and continues to be modified and utilized in myriad ways. Explanations based on exaptation, the adaptation of a trait originally used for some other purpose, seem inadequate, as most of what the human mind routinely does today did not even exist at the time it was originally evolving. Experts in human evolution or cognition have yet to provide a truly satisfactory explanation. One can find simplistic accounts of our evolution on the internet and in museums. Favorite genes are listed as causal agents for our evolution from ape-like ancestors. But in order for our common descent from an ape-like ancestor by purely natural means to be true, many of these features had to arise at the same time in a coordinated fashion, meaning some number of mutations also had to happen at the same time. For example, an enlarged brain would require changes to the jaw and teeth as well as to the brain case. This significant and defining anatomical change would have needed multiple mutations to occur. In this regard, an article from the Associated Press discussed a scenario where a single genetic mutation, found in humans, was proposed to have reduced the size of our ancestors' jaw muscles, thereby reducing jaw size, and permitting large brain cases and large brains to evolve. Critics disagreed, saying evolution does not work so neatly. The first early humans with the mutation probably would have had weaker mouths, but still had large teeth and jaws. Many additional mutations would have been needed. The mutation would have reduced the Darwinian fitness of those individuals, said anthropologist Bernard Wood of George Washington University. It only would have become fixed if it coincided with mutations that reduced tooth size, jaw size, and increased brain size. What are the chances of that? Clearly, many mutations would have had to occur together, simultaneously, to accomplish the kind of coordinated change necessary for our evolution. They could not have been just any mutations, for by themselves or in the wrong combinations they would have been deleterious or even fatal, thereby wiping out any progress. 6. Time enough for change? Let's ask the question, then, how long would it take to acquire a specific mutation, or even two mutations, to accomplish a particular anatomical change? The general assumption by population geneticists is that mutations arise at a regular rate, though this may not be true. Once a mutation has occurred in one individual, it must spread through the population over succeeding generations to make a lasting change in the species. When the mutation is finally the only version of a particular gene in the population, we say the mutation has become fixed in the population. How long this process takes depends on several factors. The first is whether the mutation is beneficial by itself or not. Second, the size of the population matters. Third, the generation time also is important. Finally, of course, the mutation rate itself has an important effect. When these factors are taken into account, it is possible to build mathematical models to estimate how long it would take a particular kind of mutation to appear. The estimates of these factors most commonly given by population geneticists are that we supposedly came from a population of about 10,000, with a generation time of 10 years, apes have shorter generation times than we do, and a mutation rate of 10 to 8 mutations, that is, 1 in 100 million, per base per generation, at the time we last shared common ancestry with chimpanzees. A number of mathematicians and biologists have used these or similar estimates to calculate the length of time it would take to obtain a binding site in the DNA within 1,000 bases of some gene, in order to change its gene expression. The reason for this calculation? It is assumed that changing a binding site in the DNA would change gene expression, and thus change that individual's behavior or anatomy in some way. The mutation would then need to spread through the population and become fixed. Here's the problem: to get a single mutation in a DNA binding site and have it become fixed would take anywhere from 1.5 million years to 6 million years, depending on whose calculations are used. If 2 mutations are needed to get a change in behavior or anatomy, it would take approximately between 84 and 216 million years, once again, depending on whose calculations are used. Other research using different methods merely confirms the problem. Yet we have only 6 million years since we supposedly diverged from chimpanzees. One proposed solution is to allow the search space to include 1,000 bases near any gene in the genome. That means that for a 5-base binding site there are a maximum of 20,000 x 205 base non-overlapping sequences or 4 million places available for a new binding site to appear. That reduces the waiting time considerably, to 7,500 years on average for a 5-base binding site. The problem is that very few binding site mutations are likely to be beneficial. Not just any old binding site will do. And most beneficial traits will require more mutations than just one. If even one essential trait required 2 specific coordinated mutations, the evolutionary process would stall completely, because 216 million years is too long to wait. The neo-Darwinian process cannot accomplish what is needed to explain our origin in the time available. 7. Does similarity indicate common descent? We have ruled out the Darwinian mechanism as being sufficient to account for our origin, but we have not addressed the question of common descent, namely, could a designer have used the evolutionary process to make human beings? We already know that it would have to be a guided process, because of the problem of the amount of time required to accumulate all the necessary coordinated mutations. So now the question is, do we indeed share common ancestry with chimpanzees? Are our bodies the product of guided evolution from an ape-like ancestor? Or do we have a unique created origin as human beings? In this section we will examine several arguments that are typically made by evolutionary biologists in support of common descent, and consider the responses of design biologists. Assumptions. Before we examine the evidence, though, it should be mentioned that there are some basic differences between the way evidence is approached by evolutionary biologists and design biologists. The chief assumption made by evolutionary biologists is that the genetic changes responsible for evolutionary change are random, and therefore, if a group of species share a trait in common that is not found in other related species, it is presumed that the common ancestor of the group developed that trait, and they all share it because of common descent. On the other hand, if genetic change is directed rather than random, the trait is most likely shared because the organisms use similar solutions to a physiological need. This beautifully explains convergent evolution, where unrelated organisms share a trait in common. It should also be noted that many arguments about common descent are circular, in particular those concerned with pseudogenes, supposedly broken genes of no use to the organism but still in the DNA. If organisms are not thought to be closely related, then the existence of a particular pseudogene is claimed to be due to functional requirements. On the other hand, if organisms are thought to be closely related, that pseudogene is taken to indicate common descent. To say that pseudogenes prove common descent when the organisms involved are closely related, but prove a functional constraint when the organisms in question are unrelated is to assume common descent from the very beginning, not to prove it. Logicians know this as the fallacy of begging the question. Evolutionary biologists tend to assume randomness and attribute any similarity to common descent. Design biologists tend to assume order and purpose and assume that any similarity is functional, not necessarily due to common descent. Darwinian evolutionists tend to assume that many poorly understood aspects of our genomes, example, pseudogenes, synonymous codons, or junk DNA, see below, do not have important functions and are the product of random mutations and common descent. In contrast, proponents of intelligent design predict that, if our genomes were designed, then many of these mysterious features will turn out to have important functions. The problem is to distinguish between the two viewpoints. 8. Arguments used to support common descent. Pseudogenes. Because pseudogenes appear to make defective protein, or none at all, they have been thought to be junk, remnants of the evolutionary process. Because they tend to be located in the same place and have the same errors in humans and in chimpanzees, they are taken as evidence for common descent, for example by Francis Collins and Dennis Alexander. Pseudogenes have not received much attention in the scientific literature because they are assumed to be junk. But that is changing rapidly. Where pseudogenes have been carefully studied, they are often found to be functional, and in some non-standard ways. Part of the problem is that a pseudogene may be active in specific tissues only during particular stages of development, making identification of their functions difficult. Nonetheless, researchers in the field are confident that continued research will yield more evidence of functionality. As one group states, we believe that more and more functional pseudogenes will be discovered as novel biological technologies are developed in the future. Definitely, the so-called pseudogenes are really functional, not to be considered anymore as just junk or fossil DNA. Surely, many functional pseudogenes and novel regulatory mechanisms remain to be discovered and explored in diverse organisms. There are several ways in which pseudogenes have been found to work so far. They can fuse with adjacent genes to form chimeras that produce either coding or non-coding RNAs, and they can form RNA duplexes with their parental gene and prevent its expression or signal its degradation. One sign pointing toward their likely functionality is that their sequence is very similar in many different species. There are more than 8,000 processed pseudogenes in the human genome, 60% are very similar in mouse and human. The fact that their sequences are so similar means that they are likely to have a sequence-dependent essential function, so that their sequence cannot be changed without harm. That degree of similarity is not something that would be expected if pseudogenes served no biological function. If they do have a function in both chimpanzees and humans that is highly dependent on their precise sequences, the similarity between chimpanzee and human sequences would not be surprising. They could be performing the same function, requiring the same sequence in both genomes. As an example, a pseudogene in the beta-globin cluster of genes in humans shows evidence of sequence similarity within and between the human and chimpanzee populations. The similarity is not due to protein coding but appears to involve regulation of developmental changes via chromosomal interactions. Synteny refers to how well chromosomal sequences from different species align with one another, how similar the arrangement of genes along their chromosomes is. If they align well, it is assumed to indicate they came from a common ancestor. But there is a possible functional explanation. Chromosomal structure has a profound effect on gene regulation, and where genes are located in the nucleus affects their expression. Long and short-range interactions between genes, the way the DNA loops, whether genes are sequestered or not, all these things are affected by chromosomal order. Rodley et al. Say, the association of chromosomes with each other and other nuclear components plays a critical role in nuclear organization and genome function. Genomes are highly ordered yet dynamic entities in which chromosomal positions, structures and interactions are controlled in order to regulate nuclear processes. This sophistication is a strong argument for design. Thus, it is possible that any similarity similarity in gene order might be a function both chimps and humans need. If the sequence similarity is due to function, then it need not indicate common ancestry. Synonymous codon use. One of the evidences offered in favor of common descent is the reuse of synonymous codons. Some codons, triplet nucleotide sequences in DNA, specify the same amino acid. For example, lysine may be encoded by 4 different codons in RNA, CUU, CUC, CUA, or CUG. The assumption has been that these codons truly are synonymous and ought to be able to be used interchangeably. Yet when there is a choice among possible codons, chimpanzee and human genomes almost always use the same codon. It is therefore argued that this is because the sequences share a common ancestor. However, it has since been discovered that codons are used for multiple purposes. Which codons are used changes the DNA sequence, which in turn can affect gene expression, protein degradation, the controlled destruction of proteins by the cell, or how proteins fold into 3-dimensional shapes. Codons may therefore be identical because they have to perform multiple jobs that are specific to one codon, none of the others will do. The fact that both humans and chimpanzees use the same codons may be due to this rather than to their ancestry. In addition, this is a strong argument for design. The sophistication required to have codes within codes would not arise by simple mutation and natural selection. Chromosomal fusion. When chimpanzee and human genomes are compared, our chromosome 2 appears to be a fusion of 2 chimpanzee chromosomes. The argument is made that this demonstrates our common ancestry with chimpanzees. However, the junction where the supposed fusion took place is not made of typical telomeric sequences. Telomers are special sequences found at the end of chromosomes. Instead, degenerate sequences are found elsewhere in the genome but not associated with breaks or fusions. The human chromosome 2 may have always been as it appears now. There is no particular reason to propose there was a fusion event except under the assumption of common descent. Therefore it cannot be used as an argument for common descent. The designer as a deceiver. This argument goes something like this: if there is an intelligent designer, then why did he make it look like things evolve? That makes him a deceiver. There is a logical flaw here as well. It is stated as fact that things look like they evolved by natural processes. But things do not look like they evolved. As has been shown, many good reasons to believe things were designed are found in molecular biology. There are also many examples from the design of larger-scale structures like the eye or a bird's wing. Even the complementary and interlocking nature of the biosphere all give evidence of design. In fact, biologists are continually told that they must remember that things only look designed, they really aren't. That clearly means the designer is not a deceiver. He has made it so that everyone can detect his design. 9. Population genetics and Adam and Eve. In recent years there has been much discussion about whether the genetic data indicates that humans could not have descended from an initial couple. Evolutionary biologists often cite population genetics models in this regard. Based on these models, current estimates are that there were about 10,000 individuals in the population when we first split from chimpanzees. 10,000 individuals are too many to allow for a unique origin with Adam and Eve. However, the basic assumptions used in population genetics may be flawed, and there is good reason to think that an alternate model for our origin is possible. This argument is presented in the next chapter. Conclusions. The preponderance of the evidence says that we humans have a unique origin. First, arguments from chapters 1 through 14 show that neo-Darwinism cannot explain the information-bearing, complex, and specified nature of life, and that common descent is a flawed hypothesis. Then in this chapter we have demonstrated that neo-Darwinism cannot account for the specific patterns we see in our genomes, and that the common ancestry of chimps and humans is questionable. The amount and kinds of differences in our DNA cannot be accounted for in the proposed evolutionary time available. The apparently purposeful placement of repeated elements like signs and lines, plus their important roles in species-specific differences, argue against an unguided process of common descent. The sheer, amazing complexity of gene regulation and chromosome interactions argue for brilliant design. Our anatomical and physiological differences would require many specific coordinated changes that could not happen without guidance. But more importantly, if things like synteny, shared codon usage, and pseudogenes can be explained functionally, the argument for common descent deteriorates, and a unique origin becomes more likely. The chief question asked about human evolution is this: are we descended from an ape-like ancestor, or are we unique, with a distinct origin? In the context of this book, of course, this question is paramount. One of the reasons people tend to become theistic evolutionists is precisely because they consider the evidence based on genetic similarity for our descent from an ape-like ancestor to be incontrovertible. We have presented arguments here that indicate the evidence for chimp and human common ancestry may not be so incontrovertible after all, and that in light of our basic ignorance of the way our genome functions, and in light of the increasing evidence for human-specific design, the rewriting of theology is a bad idea. Let us now review the overall argument we are making and where we are so far, in brief. Chapter 14 shows that the fossil record does not support the idea of a continuous evolution between a chimpanzee-like ancestor and us. This chapter shows that our genetic differences are far greater than have been commonly reported, and that they reveal many functionally significant, uniquely human changes. There are too many such changes to have happened by random mutation and selection. In the next chapter we will examine whether the claim that we had to come from a starting population of at least several thousand is necessarily true. We will report ways to distinguish between that idea and the possibility that we may have come from a single pair, Adam and Eve, and we will describe a new population genetics model that may allow us to test the two possibilities, to see which best corresponds with the actual genetic data we have.